Okay, Kavita?
All right, are we ready? Okay.
Yes.
All right, today's April 17, 2024. We will now convene the PNT committee meeting. I'm Kavita Chawla, the chair of the PNT committee. I will read off the names of the participating attendees, so please say here when I call your name. We'll start with the PNT committee members. Peter Barquette.
Here.
Hi. Laura Bestie.
Here. Good morning.
Morning. Michael Corsellis.
Here. Good morning.
Good morning, Michael. Kevin Flynn.
Here. Good morning.
Morning, Kevin. John McKay.
Here. Good morning.
Morning, John. Christy Weiland.
Good morning.
Morning, Christy. Zoe Taylor.
Hello.
Hey. And Gregory Hudson.
Here.
Here. Do you prefer Greg or Gregory?
Greg is fine. Thank you.
Okay. All right. And then our Healthcare Authority members, Nania Connor.
Hello. Hi, here.
And Luke Dearden.
Luke won't be here today.
Okay, got it. Lita Vasquez.
Here.
Amy Irwin.
Yeah, she might be joining later.
Okay. Ryan Pistoresi.
Morning.
Morning. Donna Sullivan.
Hi, it's Donna. Amy won't be coming.
Oh, okay.
Okay. Marissa Tabile.
Here. Good morning.
Morning. Ryan Tekatomo.
Hello, good morning.
Good morning, Ryan. Joy Zarate.
Here.
Hi. Our labor and industry members, Jamie Mai.
Here.
Great. And then our DERP presenters, Shannon Kugley.
I'm here, thank you.
Hello. Wesley Lindsay. Okay. Rachel McCausland. And Courtney Cooper.
They might
Here,
be
good
coming on
morning.
closer to their presentation time.
Okay, got it. All right. And then our Magellan Medicaid Administration presenter, Umang Patel.
Hi. Good morning.
Good morning. And then our managed care organization representatives are Greg Seamus from Molina Healthcare, Heidi Goodrich from Molina, Petra Eichelsdorfer, United Healthcare, Omar Daoud, Community Health Plan of Washington, and Jeffrey Natividad, Community Health Plan of Washington. Now, Nania, we'll go over meeting logistics.
Sorry, Dr. Chawla, can I just ask for one thing?
Please.
I just wanted to quickly introduce Nina Nguyen. She is my colleague here at Prime Therapeutics. Just to let the committee know that I will be slowly phasing off of the Washington account, and Nina will be slowly taking over as well. And so this is her first meeting. I will still be presenting, and she's just kind of observing to get a lay of the land.
All right. Hi, Nina. Welcome.
Hello. I look forward to working closely with you all.
Thank you. Thanks for that, Umang. All right, Nania, go ahead.
Yes. Hi. Good morning, everyone. The committee and presenters can mute and unmute themselves at any time, but please mute yourself when not speaking so as to limit background noise. Presenters, please share your webcams while presenting. And also committee members, please share your webcams during discussions and motion consideration. For stakeholder participation, the chair will read the list of stakeholder names who pre-registered to speak. We will unmute you. After, the chair will ask if there are any other stakeholders. If there are, please raise your hands and we can call upon you and unmute you. You can also use the Q&A box. We'll address your questions during the stakeholder time. If you do not fill out the stakeholder conflict of interest form, please answer the questions we'll post on the screen. Your 3 minutes will start after you answer the questions. Thank you.
Thank you, Nania. And any other logistics or operational updates from Lita? Okay, I'll take that as a no. All right, with that, let's get started. So I see Shannon, you're up first from DERP.
Yep.
Take it away.
Okay, great. Can you hear me okay? Great, okay. Go ahead and share my screen. Okay, can you see the presentation?
Yes, we can.
Great. Okay, I'll get started. Hi, everyone. So I will be talking to you about targeted immune modulators for plaque psoriasis, psoriatic arthritis, and generalized pustular psoriasis. This is an update of a systematic review. And my name is Shannon Cugley, and I'm from the RTI UNC Evidence-Based Practice Center. So here's an overview of today's presentation. And here we have a list of the abbreviations that are used in the presentation. And as you know, this report covers plaque psoriasis and psoriatic arthritis. New to this report is generalized pustular psoriasis, or GPP. And TIMs are drugs that selectively block the immune response. These drugs were initially FDA approved for use in these conditions in 2002. In 2022, the FDA approved spesolimab for treatment of generalized pustular psoriasis. Here in this figure are the drugs that are included in the review. We have them organized according to their mechanism of action. So the mechanism of action is listed in the dark green box, and the drugs in the gray-blue boxes are approved for both plaque psoriasis and psoriatic arthritis. Those drugs in the light green boxes are approved for plaque psoriasis only, and those in the yellow boxes are approved for psoriatic arthritis only. There's just one agent in a peach shaded text box, which is approved for patients with GPP. An asterisk indicates that biosimilars are available and approved for use. So new drugs with this update include the IL-36 inhibitor, spesolimab, which was approved by the FDA in September of 2022 for treatment of GPP, as well a new topical PDE4 inhibitor, roflumilast, was approved by the FDA in July 2022. However, the oral formulation is not yet FDA approved. Since the prior report, the tyrosine kinase 2 inhibitor, deucravacitinib, and the IL-17A inhibitor, bimekizumab, was approved for plaque psoriasis. And here are the PICOs for this updated review. The criteria were similar to the prior report, except that we added GPP and we excluded cohort studies. Our KQ for the update was number 1 on the comparative effectiveness, and the second on the comparative harms, the third on variation in effectiveness or harms by subgroup, and the fourth on ongoing studies. Our updated search was conducted to cover the literature that was available through the end of August 2023, with active surveillance through November of 2023. We conducted individual study risk of bias assessment, and we calculated risk ratios and confidence intervals where possible. We applied the grade methodology to rate the overall certainty of evidence. Lastly, we searched clinicaltrials.gov to identify ongoing studies. Here's the DERP approach to risk of bias. So low risk of bias indicates clear reporting and mitigation of any potential bias due to conflicts. Moderate indicates some incomplete information on methods or that unmitigated meaningful conflicts of interest exist. High is for studies with flaws that introduce serious bias. And here's a brief overview of the grade certainty of evidence. We graded disease remission, clinical improvement, quality of life, adverse events, and serious adverse events outcomes for this review. As a reminder, our CT bodies of evidence start at high certainty of evidence and can be downgraded for problems of consistency, precision, risk of bias, and reporting bias. On this slide is our study flow diagram. We screened just over 1,450 citations, and we assessed 82 full-text articles. We identified 11 new articles, of which 6 were new studies. We carried forward 34 studies that were reported in 55 publications from the prior report. Thus, the total for the current review is 66 articles representing 40 studies. The report included data from 30 studies of TIMS for plaque psoriasis, 10 for psoriatic arthritis. We found no eligible studies of TIMS for treating GPP. That is because studies had to be head-to-head comparisons to be included, and the studies of spesolimab were not eligible because they were placebo-controlled. This figure depicts selected study characteristics. Of the total 40 studies, 6 are new to this update and 34 were included in the prior report. 36 are moderate risk of bias and 4 were high risk of bias. 30 were conducted among participants with plaque psoriasis, 10 among individuals with psoriatic arthritis. 37 studies were head-to-head comparisons of FDA-approved TIMs, and 3 were comparisons of a pipeline TIMs to an FDA-approved TIM or placebo. As a reminder, this figure characterizes the unique studies included in the report. We refer to publications that report additional outcomes or longer-term follow-up for an included study as a companion. In the subsequent slides, I will present the review findings, first for plaque psoriasis and then for psoriatic arthritis. Within each condition, I will describe findings for the comparative benefits, including variation by subgroups, followed by comparative harms, and lastly, briefly report on the comparative benefit and harms of pipeline drugs. On this slide, we list the most common outcomes used in the studies. ACR is a measure of arthritis clinical improvement and disease remission. DLQI is a quality of life measure specific to dermatologic conditions, where a score of 0 or 1 indicates remission. PASI is a measure of clinical improvement and disease remission for psoriasis, and the PGA, IGA, patient GA are global assessment items to measure clinical improvement and disease remission. I will now share the findings for the comparative effectiveness of RCTs for plaque psoriasis. Here is an overview of the studies we identified addressing the comparative effectiveness of TIMs for plaque psoriasis. As I noted, we included 30 RCTs representing 23 unique head-to-head comparisons. Nearly all studies enrolled participants with moderate to severe psoriasis based on at least a 10% BSA, a PASI score, or both for at least 3 to 6 months duration. Some, but not all studies required participants to be naive to biological agents. 4 RCTs are new to this update, including 2 RCTs of deucravacitinib versus apremilast, 1 of guselkumab versus secukinumab, and 1 RCT of risankizumab versus apremilast. In addition, we identified 1 new publication associated with the previously included RCT of guselkumab versus adalimumab, and 1 companion study reporting results of subgroup analyses from a previously included RCT of risankizumab versus secukinumab. Regarding the risk of bias ratings across the entire body of evidence, all the included RCTs were sponsored by industry and had extensive industry involvement in study design, execution, and reporting, and were therefore downgraded from low to moderate. Furthermore, we assessed 2 RCTs as high risk of bias, one due to insufficient blinding and switching of treatments during the study, and a second because it was open label. Over the next series of slides, I will share evidence for each comparison. Here we see an example of the summary of the findings for KQ1. The comparison in the number of studies and participants are in the green text box. We found 1 RCT, including 166 individuals comparing apremilast with etanercept. The outcome category and grade assessment are in bold text. In this example, clinical improvement and quality of life were reported. The findings are noted along with the specific measure that was used to obtain the result and the time point of follow-up. This study reported no difference in clinical improvement or in quality of life as measured by the PASI 75 and the DLQI respectively at 16 weeks. We graded each outcome as low certainty. For this presentation, we include only the outcomes that we graded. Other outcomes are described in the full report. For previously included comparisons without new data, I will summarize the findings, but I will provide more details on outcomes for new comparisons or companion papers with new data. In the interest of time, I will not discuss the rationale for individual grade assessments, but I can tell you that across the evidence, imprecision accounted for most of the downgrades of 1 to 2 levels. Downgrading for other reasons was less frequent. For details on the grade assessment, please refer to the full report. We did not identify any new studies or data of mimikizumab versus adalimumab or versus secukinumab. Of note, bimekizumab was a pipeline in the prior report. Bimekizumab was more effective than adalimumab for disease remission and for quality of life. We assessed outcomes as moderate certainty. Bimekizumab was also more effective than secukinumab for disease remission and quality of life, also assessed as moderate certainty. We did not find any new studies or data for bimekizumab versus ustekinumab. Bimekizumab was more effective than ustekinumab for disease remission and quality of life. We assess the certainty of evidence as moderate for both outcomes. We did not identify any new studies or data for the comparisons on this slide. Brodalumab was more effective than ustekinumab for disease remission and quality of life, and we assess the evidence as high for both outcomes. The higher dosage of certolizumab pegol, 400 milligrams, was more effective than etanercept for clinical improvement. There were no differences in clinical improvement between the lower dosage, that is 200 milligrams, and etanercept. The certainty of evidence was moderate. Deucravacitinib versus apremilast is a new comparison for this update. We included 2 RCTs comprised of more than 1,200 participants. Deucravacitinib was more effective than apremilast for clinical improvement at 16 weeks, as measured by the PASI 75. The relative risk was 1.7 with a 95% confidence interval of 1.3 to 2.1 in one study, and 1.3 with a 95% confidence interval of 1.1 to 1.6 in the second study, favoring deucravacitinib for clinical improvement. We assessed the certainty of evidence as high. Deucravacitinib was more effective than apremilast for quality of life, as measured by achievement of a 0 or 1 score on the DLQI. At 16 weeks, the relative risk was 1.4, with a 95% confidence interval of 1.1 to 1.9, and was 1.6 with a 95% confidence interval of 1.3 to 2.1 in the second study. At 24 weeks, the relative risk was 2.0, with a 95% confidence interval of 1.5 to 2.7 in one of the studies, and was 1.9 with a confidence interval of 1.5 to 2.5 in the second study. We did not identify any new data or studies for etanercept versus infliximab or etanercept versus ixekizumab. For both comparisons, etanercept was less effective for disease remission as measured by PASI 75 and was less effective than ixekizumab for improving quality of life. Certainty was very low for the outcomes in the first comparison and high for etanercept versus ixekizumab. We did not identify any new studies or data for etanercept versus secukinumab. Etanercept was less effective than secukinumab for achieving disease remission and improving quality of life. Certainty was high and moderate for those two outcomes, respectively. We did not identify any new studies or data for etanercept versus tildrakizumab. Etanercept was less effective for disease remission at 12 and 28 weeks. We rated certainty of evidence as high. Etanercept was also less effective than tildrakizumab for improving quality of life, and we assessed certainty of evidence as moderate. This comparison also had a subgroup analysis, which showed no difference based on having or not having metabolic syndrome. We did not identify any new data or studies comparing etanercept with tofacitinib. We identified one RCT that compared etanercept with 2 doses of tofacitinib, either 5 milligrams or 10 milligrams twice daily. Of note, tofacitinib is not approved for plaque psoriasis, and there is no indication that it will be resubmitted for consideration for plaque psoriasis. Etanercept was more effective than a 5 milligram dose of tofacitinib for disease remission, clinical improvement, and quality of life outcomes. There was no difference in outcomes for etanercept versus tofacitinib 10 milligrams. Certainty of evidence was moderate for all outcomes except for quality of life for the 10 milligram tofacitinib comparison. We did not identify any new studies or new data for etanercept versus ustekinumab. Etanercept was less effective than ustekinumab for disease remission. We assess the certainty of evidence as low. Guselkumab was more effective than adalimumab for achieving disease remission and improving quality of life over 16 weeks, and this was based on high and moderate certainty of evidence, respectively. We included one new companion paper with data from this previously included RCT of guselkumab versus adalimumab. The new companion paper reported that guselkumab was superior to adalimumab for improvement in work productivity loss. However, work productivity loss was not an outcome for which we assess certainty of evidence. We identified one new high risk of bias RCT for this comparison. Based on the one previously reported RCT that reported disease remission, guselkumab was more effective than secukinumab at 48 weeks. Guselkumab was not inferior to secukinumab for the combined endpoint of 12 and 48 weeks. The response for guselkumab was numerically lower than secukinumab for disease remission response at 12 weeks. We assess certainty of evidence as moderate. In a separate article, authors reported on this comparison by subgroups based on age, weight, BMI, severity of disease, body area affected, and prior medication use. Guselkumab remained more effective than secukinumab at 28 weeks in each strata of every subgroup identified. The newly identified RCT compared guselkumab with secukinumab in 40 participants with a PASI score less than 10, which is not moderate or nor severe disease, and with a single treatment refractory plaque. Guselkumab was less effective than secukinumab at 16 weeks for clinical improvement. We rated this study as high risk of bias, and the certainty of evidence for clinical improvement was very low. No new studies were identified for ixekizumab versus guselkumab or for ixekizumab versus secukinumab. Ixekizumab was more effective than guselkumab at 12 weeks for disease remission. There was no difference between the agents by 24 weeks. Findings were similar for quality of life. We assess outcomes as moderate certainty of evidence. There was no difference between ixekizumab and secukinumab for disease remission or clinical improvement at 24 weeks in a study of individuals with genital psoriasis. Certainty of evidence was moderate for both outcomes. We found no new studies or new data for the comparison of ixekizumab versus ustekinumab. Ixekizumab was more effective at achieving disease remission and improvements in quality of life at 12, 24, and 52 weeks, and we rated this evidence as moderate certainty. We did not identify any new studies or data comparing risankizumab to adalimumab. Risankizumab was more effective at achieving disease remission and improving quality of life compared with adalimumab at 16 weeks. We assessed outcomes as moderate certainty of evidence. We identified one RCT of a new comparison of risankizumab versus apremilast with 352 participants. Risankizumab was more effective than apremilast for disease remission as measured with the PASI 90 and for clinical improvement measured as PASI 75 at 16 weeks. We assessed the certainty of evidence as moderate for both outcomes. We did not identify any new studies of risankizumab versus secukinumab, but we did identify one new companion paper. Risankizumab was more effective than secukinumab for achieving disease remission at 16 and 52 weeks. We rated this evidence as moderate certainty. The newly identified companion paper reported results of subgroup analyses from the previously included RCT. There were no significant differences in outcomes based on post-hoc subgroup analyses for age less than 40 years versus 40 years and older, male versus female, white versus non-white, BMI of less than 25 versus 25 to 30 versus 30 or higher, disease severity at baseline, prior biological use versus no use, presence versus absence of psoriatic arthritis, current versus former versus never smoker, and disease duration of less than 15 years versus 15 years or more. We did not identify any new RCTs or data for risankizumab versus ustekinumab. Risankizumab was more effective than ustekinumab for disease remission and quality of life at 12 to 16 weeks. We assess the certainty of evidence as high for both outcomes. We did not identify any new studies or data for the comparison of secukinumab with ustekinumab. Secukinumab was more effective than ustekinumab for disease remission and quality of life at 16 and 52 weeks. We rated both outcomes as high certainty of evidence. All RCTs included for KQ1 also reported harms. In the next slide, I will review these comparisons that had a significant difference in either the AEs or SAEs and with at least an evidence rating of low. In one RCT, apremilast was associated with a lower incidence of AEs than etanercept. We rated this evidence as low certainty, and there was no significant differences in the incidence of serious AEs. In one RCT comparing etanercept with a 100 milligram and 200 milligram dose of tildrakizumab, AEs were reported in two separate time periods, weeks 1 through 12 and weeks 13 through 28. There was a higher incidence of AEs with etanercept versus the 100 milligram dose of tildrakizumab for both time periods. However, compared with the higher dosage, the difference was significant in the second period only. Certainty of evidence was moderate. In a study new to this update, the incidence of overall AEs was lower for risankizumab versus apremilast, with a relative risk of 0.68 and a 95% confidence interval of 0.54 to 0.86. There was no significant difference in the incidence of AEs. We assess the certainty of evidence as moderate. In one of the three studies comparing risankizumab to ustekinumab, AEs were reported in two time periods, weeks 1 through 16 and weeks 17 through 52. A lower incidence of AEs was observed for risankizumab during weeks 17 to 52, and a lower incidence of SAEs was observed for risankizumab during weeks 1 to 16. No significant differences were observed for AEs or SAEs in the other two RCTs. We assess certainty of evidence as low for AEs and low for SAEs. Next, we'll review the evidence for TIMS for plaque psoriasis. On this and the next two slides is a table that summarizes the evidence for clinical improvement, disease remission, and quality of life, as well as overall AEs and SAEs for the 23 comparisons for plaque psoriasis that were included in this update. The comparisons in blue text are either new to this update or have new data for this update. The certainty of evidence is indicated by the number of filled circles within each table cell. The direction of effect is indicated by cell shading. Blue means that the first TIM of the comparison is more favorable. Purple means no difference between the two agents, and red means that the second TIM listed was more favorable. The cells shaded in gray indicate unable to determine the direction of effect. Because the table of comparators is long, this information is split across 3 slides. One pattern to observe is in this table is that there are very few differences between agents with respect to harms. Differences between agents are observed for clinical improvement, disease remission, or quality of life. However, with so many comparisons, it's difficult to identify clear patterns about effectiveness. We have the most evidence for etanercept, which was represented in 8 comparisons, and for ustekinumab, which was represented in 6 comparisons. Etanercept was inferior to certolizumab, infliximab, ixekizumab, secukinumab, tildrakizumab, and ustekinumab. Etanercept was more effective than the lower dose of tofacitinib and was similar to apremilast. Ustekinumab was less effective than all comparator agents it was assessed against except for etanercept. This includes bimekizumab, brodalumab, ixekizumab, risankizumab, and secukinumab. Agents other than etanercept and ustekinumab are represented in fewer comparisons, so identifying patterns for any given agent is challenging and requires a more complex analysis than what we can do in this present update. We found no eligible studies of pipeline agents for plaque psoriasis. Next, we will move on to psoriatic arthritis. We identified a total of 7 RCTs. As you can see from this evidence map, 6 of them compare a TIMS agent to adalimumab. The 7th study compares ustekinumab to the TNF-alpha inhibitors. However, the specific one was selected by the participants and their providers, so the study was not blinded, and this was the reason that it was assessed as high risk of bias. The other study that we assessed as high risk of bias compared adalimumab to etanercept and infliximab. It was not well described and allowed for dose adjustments of infliximab during the trial. We identified 3 companion publications reporting data from previously included RCTs. 1 new companion publication reported a subgroup analysis, and 2 reported additional outcomes. We did not identify any new studies or data for this comparison. Clinical response was similar across agents. No statistical significant testing was done. We assessed the evidence as low certainty. Very low certainty. We did not identify any new studies or new data for adalimumab versus tofacitinib. Adalimumab has had numerically lower response rates on ACR20 compared with both doses of tofacitinib. Adalimumab had a numerically lower response on PASI 75 for skin disease compared with the higher dose of tofacitinib, and a similar response to the lower dose of tofacitinib. Quality of life outcomes were roughly similar across the groups. The study did not conduct statistical testing. We assessed all outcomes as low certainty of evidence. We did identify 1 new companion paper reporting a subgroup analysis, but no new trials for ixekizumab compared with adalimumab for psoriatic arthritis. There were 2 RCTs of this comparison. In 1 study, no statistical testing was done for ixekizumab versus adalimumab, as the primary aim of the study was to compare ixekizumab to placebo. Ixekizumab every 2 weeks and every 4 weeks was slightly better than adalimumab for joint disease improvement at 24 weeks. We rated this evidence as moderate certainty. Ixekizumab was superior to adalimumab for skin disease improvement in both studies, with a moderate certainty of evidence. We assessed clinical improvement as high for a composite ACR50 and PASI 100 response, and for sustained PASI 75 response at 52 weeks for ixekizumab versus adalimumab. The newly identified companion paper reported a post-hoc analysis of a subgroup of our participants with psoriatic arthritis and moderate to severe plaque psoriasis from one of the previously included trials. The subgroup included 100 of the 556 trial participants. Ixekizumab was more effective than adalimumab at 24 and 52 weeks for the primary endpoint, which was simultaneously achieving ACR 50 and PASI 100, and for most of the other outcomes. Findings were mixed for quality of life outcomes. We did not identify any new studies or data for secukinumab versus adalimumab. 1 RCT reported no difference in arthritis clinical improvement at 52 weeks. Secukinumab was more effective than adalimumab for clinical improvement in skin disease. We assess the certainty of evidence as moderate for both outcomes. We identified new data for 1 study that was previously included for the comparison of upadacitinib versus adalimumab. A larger proportion of individuals taking upadacitinib 30 milligram dosage showed improvement in arthritis at 12 weeks as measured by the ACR20. Upadacitinib 30 milligrams remained more effective than adalimumab at 56 weeks. The relative risk was 1.1, with a 95% confidence interval of 1.001 to 1.2. There was no difference in clinical improvement with the lower 15 milligram dose of upadacitinib. We assessed the certainty of evidence for clinical improvement as moderate. For quality of life, the authors reported change in HAQ-DI at 12 weeks. Upadacitinib for 15 and 30 milligrams were better than adalimumab. The newly included companion paper reported quality of life at 56 weeks. Upadacitinib 30 milligrams was more effective than adalimumab for quality of life, with a relative risk of 1.2 and a 95% confidence interval of 1.01 to 1.3, and was measured as the proportion of participants who achieved a change in HAQ-DI score of 0.35 or higher. There was no significant difference for the 15 milligram dose. We assess the certainty of evidence as moderate for quality of life. We did not identify any new studies for this comparison. A small RCT with 47 participants with active enthesitis, a narrow subset of overall psoriatic arthritis, compared ustekinumab with various TNF-alpha inhibitors. We graded the evidence as very low certainty for ustekinumab being more effective for enthesitis and skin disease remission, but no difference in arthritis remission. Six of the 7 studies that were included for KQ1 for psoriatic arthritis are also reported harms. As before for harms, we are only presenting comparisons where a significant difference in AEs or SAEs was reported and when the certainty of evidence was at least low. We identified one new companion paper with data from a previously included study that compared either 30 milligrams or 15 milligrams of upadacitinib versus adalimumab. More AEs were found with upadacitinib 30 milligrams with a relative risk of 1.1 and a confidence interval of 1.02 to 1.20. We assess this as moderate certainty of evidence at both the 12 and 56-week time points. The relative risks were 1.1 and 1.3, and both 95% confidence intervals exclude the null results. There were no differences in AEs for the 15 milligram dosage for either time point. Furthermore, there were no differences in SAEs at 12 or 56 weeks for either dosage, with low certainty of evidence. And here's a summary of the RCT evidence for benefits and harms of TIMS for psoriatic arthritis. We did not identify any new comparisons of an FDA-approved TIMS for psoriatic arthritis, but we did identify new companion studies with additional data for previously included studies. As you can see, the evidence is mixed. For the comparisons with at least low confidence of evidence, low certainty of evidence, which is everything but the top and bottom row, you can see that compared to adalimumab, ixekizumab, and secukinumab are more effective at improving skin disease, but there are no differences for arthritis symptoms and no differences in AEs. Upadacitinib is more effective than adalimumab at improving both skin disease and arthritis. However, it is associated with more AEs. On this slide, we summarize the pipeline treatments. As a reminder, bimekizumab has been approved for plaque psoriasis, but it is not approved to treat psoriatic arthritis, thus it is listed as a pipeline agent. We identified 2 new RCTs reporting on bimekizumab. One new RCT was for a comparison that was included in the prior report, bimekizumab versus placebo. This RCT also included a head-to-head comparison of bimekizumab with adalimumab. The other new RCT compared bimekizumab with placebo. Limitations of this evidence base include, for some comparisons, that direct evidence is still lacking. So there are many possible comparisons for which we did not find any studies. There's limited long-term efficacy and safety data available. Also, manufacturers sponsored nearly all RCTs. Studies were not powered for harms outcomes, and our review did not include RCTs with a duration shorter than 12 weeks, nor did we include data from conference abstracts or press releases. We included studies published in English only and not in other languages. We identified 10 ongoing studies, 2 RCTs in participants with plaque psoriasis, and 8 RCTs in participants with psoriatic arthritis. Drug manufacturers are funding 9 studies, and a university is funding one of these studies. We found no eligible ongoing studies of TIMS for individuals with GPP. In conclusion, for plaque psoriasis, the largest body of comparative direct evidence is for etanercept and ustekinumab, compared with other TIMS agents. For clinical improvement or disease remission outcomes, etanercept is less effective than the following comparators based on moderate or high certainty: certolizumab, ixekizumab, secukinumab, tildrakizumab, ustekinumab. Ustekinumab is less effective than the following comparators based on moderate or high certainty: bimekizumab, brodalumab, ixekizumab, risankizumab, and secukinumab. Beyond the patterns we observed for etanercept and ustekinumab, several TIM agents demonstrated superior effectiveness in pairwise comparisons. You can refer to the heat map I showed earlier in the presentation for details. Lastly, few differences in harms among TIMS agents were observed based on very low to moderate quality evidence. In the future, a network meta-analysis or a review of such analyses may provide insights into patterns of effectiveness and harms across the many available TIMS agents. For psoriatic arthritis, there are limited head-to-head comparisons. From the evidence we reviewed, we can conclude that upadacitinib may be more effective than adalimumab for improvement in arthritis and skin disease, and that ixekizumab and secukinumab are no different than adalimumab for improvement in arthritis, but are more effective for improving skin disease. Any questions? And I thank you for your time.
Thank you, Shannon. Any questions from committee members? Okay, and then are there any stakeholders for this section while we're waiting? I don't see any hands raised and I don't see any listed, none yet. Sorry, you're muted.
I thought I unmuted myself, but no, there are no one listed and there are no hands raised.
Excellent. Okay. I suppose then we could go ahead and review the motion.
And right now I'm just going to copy from the previous one and I'll make the edits while you guys discuss.
I think the main sanity check would just be, which I haven't been able to do from reviewing the motion, is that all the drugs that were reviewed are actually on that list. Do you know if this is an updated left column for the drugs reviewed, Rania?
Yes, they're updated.
Awesome. Okay, great. All right, I will invite the committee members to come back on camera. Oh, I see most of you are. Yeah, any comments, updates, or suggestions?
So are all the ones in the left column supposed to also be in the paragraph?
This is Ryan. Yeah, we'll need to add in a few of them.
Yeah, they're not all in there.
And remove some. So the ones with the asterisks are the ones that they're drugs that were carried over from previous review. So I'm just removing them right now.
Okay.
So this is Ryan. So just to remind you that the TIMS class has more than just plaque psoriasis, psoriatic arthritis. It also has the rheumatoid arthritis report, which is a separate report, and then the Crohn's and ulcerative colitis report, which is a separate report. So depending on how these drugs are approved for their different indications, the list on the left includes all of the drugs within the TIMS class. And then for this, we were going to have it be the ones that were in this report. So if you recall earlier on in the presentation where they had that slide that had all the list of the different drugs and they were different colors and they were kind of in the different columns for the IL-17s and the IL-23s and the TNFs. Yeah, so we're just trying to make sure that what was presented today is what's here in the motion.
as Nadia works on that, any comments from the committee regarding the rest of the language? I think right now it says the PDL must include a drug approved for treatment of the following FDA indications. So RAAS, Crohn's, ulcerative colitis, and I guess because psoriatic arthritis is at the very top in the title. Any amendments to the language?
So we copied this from a statement about a different disease, right? So we need to change out all those diseases, I think, to the ones that we're talking about now. Because
Ryan,
copied and pasted from the paragraph below, which is about different diseases.
Exactly right.
So
Yeah.
So this is Ryan. So the last time that we reviewed this drug class, we did rheumatoid arthritis, ankylosing spondylitis, Crohn's disease, and UC. So we will want to
We'll replace all that parenthetical with what's in the title.
Right, plaque psoriasis and psoriatic arthritis. Yep.
We'll just give them a second to get to that.
Yeah.
A lot of changes.
Do we need to review which ones are preferred, or since we're not saying you have to try two preferred agents within this text, that doesn't apply?
So this is Ryan. So this is for the Washington preferred drug list, not the Apple Health preferred drug list. And so when you move to the DUR board in the latter half of today's meeting, you will be doing more of reviewing what's on the Apple Health preferred drug list and looking at the policies. For the Washington PDL, you're making the recommendations of, how should we do our cost analyses to then select the preferred and non-preferred. So that happens later with the cost analysis.
Thank you.
And this is right. So if you would like to see it, we can pull up the Washington PDL, but I think we would just need a moment to get that brought up.
Okay. It looks like I've made the edits to the drug names, and so I'm ready to slice and dice.
So the Nanya, at the bottom where there's the conditions listed in the parentheses, the rheumatoid arthritis, that whole section needs to be replaced by the conditions in the title.
Okay.
And I think to Laura's point, the piece here is that they cannot be subject to therapeutic interchange on the PDL is probably, I think, the most relevant here. Washington PDL, I don't think we've done that previously, to the best of my memory. Are any of the committee members interested in reviewing that? And if not, if anybody wants to propose the motion.
Oh, sorry, before you propose, do you want me to make this change here?
No, that part's perfect. Yeah.
Okay.
Yeah.
Any other changes to this paragraph?
Yeah.
So the list of drugs in the paragraph should match what was on the slide earlier. Okay, and we're sure that it does.
Yes,
I
and I
think there
remember.
might be some missing there. Do you want to see the slide? Would that help? Or I mean, I think guselkumab, for example, you'll want to add.
Which one is missing?
Guselkumab after Golem.
Let me see here.
I almost wonder, like, do we need to? Anyway, yes, go ahead. I don't know if anybody has a clever idea of how to do a check of all of the names and whether they're comprehensively included or not. Kevin's got one.
Sorry, you said am I missing it in the paragraph or am I missing it in here?
In the paragraph, so the guselkumab is not in the paragraph,
Oh,
but it's
okay.
in the left-hand column.
Let me see.
Um,
Below, after the golimumab.
Yeah.
I suppose if you want the entire list, do we just like copy paste that list in there and just remove the spaces after the drugs? Would that go faster?
You could.
Could you also just refer to the medications listed in the table? I mean, do you have to write them out all over again?
That's a great point too, yeah. Would that be okay I guess to Ryan and Nanya as a question? The drugs listed in the left column or something like
But
that.
the drugs in the left column are all of the TIMs. We only want to list here the ones that are for the indication at the top.
Correct.
So it can't just reference the column. So we have to double check from Shannon's slide that we have all the drugs listed that are for this indication. And then also this paragraph still says at the top, rheumatoid arthritis, ankylosing spondylitis, Crohn's disease, and ulcerative colitis instead of saying the new indications.
Oh, I'm sorry. What on the top where?
Before we list the medications.
Oh, thank you for pointing that out. Let me.
Okay, Shannon also makes a point that the drugs in the column are not the drugs reviewed. So sorry, I misunderstood that earlier.
You'll just want to highlight starting at rheumatoid arthritis.
Oops.
Just from rheumatoid arthritis through to the period and then replace that. Yeah. Perfect. And then Shannon, do you want to just read out the drugs?
Oh yeah, that might be a good way to do
If
it.
you can read them in alphabetical order too, then we can make sure that
I'll
they're
do my yes, this will be
okay. Amazing.
a fun challenge.
Sorry.
No,
Or we could do split screen. Those are sort of the two
it's
options.
okay. Let's see. Okay, I'll try to do this. Okay, so adalimumab, you have abatacept, I think you have.
Yeah.
You got certolizumab. Sorry, that's out of order. We need
No, that's fine.
brodalumab and bimekizumab. Got the 2B ones.
Brodalumab is not.
Brodalumab.
Yeah.
Yeah.
I'm sorry, which one am I missing?
The brodalumab. BRODA, right where your cursor is. Oh, and
Oh.
Nanya, can you bring up the presentation again, the slides?
Yes.
I think it might be best to.
And will you go to
yeah.
the beginning where it lists the drugs that were included in this report?
So the list is that slide that you see. They're ordered by mechanism of action. I don't have right at that. I can get you an alphabetical list if that would help. Like I can just go off mute and make a list and copy and paste it in the chat if that would help.
No, that's fine. Can you just say in your motion the drugs listed on slide 5 of the TIMS for plaque psoriasis presentation, and then we can fix the motion later.
That sounds awesome.
This is going to happen probably for all of these, so we're sorry we were not prepared for breaking it out by indication with the past motion.
that sounds reasonable to me. And do the other committee members agree? Can we use that? Yes, I see thumbs up. Okay, Nanya, does that sound good to update the motion language? So I think after I move that, I would probably remove the name of the drugs and just say
all the drugs on slide 5, and then I will update everything.
yes. So do you want to remove the drug names from the paragraph and
yeah, remove the drug names from the paragraph. Insert the drugs listed in slide 5 with the presentation name. So we know which, just the drug names.
Oh, sorry.
Go ahead, John.
You need to call out anything in regards to biosimilars at all?
We don't, the way, and I'm speaking for the center, if it's a biosimilar, I don't think we consider that as a different drug. So if it's adalimumab, XYZ, we don't list them all individually.
This is Ryan. That is correct. So we do have several of the adalimumabs already on the Washington preferred drug list. So we don't break them all out here because otherwise that list would get much longer than it already is. So if you see adalimumab, you can consider all 7 or 8 of those approved biosimilars to be within that.
Great, thank you.
This is Laura Bestie. Along those lines, for sake of completeness, should it say the medications plus their biosimilars? Or is that not even an issue?
So this is Ryan. In the past, we haven't considered that an issue. Like we don't do that for the generics. If there's an extended release of another drug, we don't necessarily have to then, include that. So some of the older motions, you may notice that we do have certain dosage forms, but we've tried to reduce that in these newer motions.
All right, great. So I'm just reading through here and make sure we're not missing anything. So after considering the evidence of safety, efficacy, effectiveness in special populations with the use of TIMS for their FDA-approved indications relative to the listed conditions, I move that the drugs in slide 5 of the TIMS for name of the slide deck are efficacious for their approved indications, including their applicable biosimilars when available. The PDL must include a drug approved for the treatment of these FDA indications and must include a self-administered agent if indicated. These medications cannot be subject to therapeutic interchange in the Washington PDL. Okay, so that's the language. How's that sound to the committee? I see some nods. Okay, then whenever we're ready to propose the motion.
This is Kevin Flynn. So after considering the evidence of safety, efficacy, and effectiveness in special populations for the use of targeted immune modulators for their FDA-approved indications relative to plaque psoriasis, psoriatic arthritis, and generalized pustular psoriasis, I move that the drugs listed in slide 5 of the targeted immune modulators for plaque psoriasis, psoriatic arthritis, and generalized pustular psoriasis update slide deck are efficacious for their approved indications, including their applicable biosimilars when available. The PDL must include a drug approved for the treatment of the following FDA indications: plaque psoriasis, psoriatic arthritis, and generalized pustular psoriasis, and must include a self-administered agent if indicated. These medications cannot be subject to therapeutic interchange in the Washington preferred drug list.
This is Laura Bestie. I second the motion.
Thank you. All in favor, please say aye.
Aye.
Any opposed? Okay, with that, the motion carries. Thank you. Okay, with that, I think we move on to Wesley Lindsay for the atypical antipsychotics session. Do we have Wesley online? Oh, I see the name.
Good morning to all. Can you hear me?
Yes, we can. Good morning, Wesley.
Welcome from Alabama. How is everything in the state of Washington today?
Sunshine, surprisingly.
Oh, I'm so sorry. Wesley, before you start, I just noticed that there was a hand raised in the attendance list from the previous. Okay, it's down now. So never mind. Sorry. Go ahead.
All right, Wesley, take it away.
All right. So am I driving the slides? Is that the preference here, or do you have a designated driver on those?
You can drive the slide, no problem.
Sure. All right, so hopefully I'm sharing my screen. Does everybody see the appropriate slides, please?
Yep.
Yes, we
see them.
Okay, great. And is there a preference whether I stay on camera for the presentation or off camera? What's the typical process?
Whatever you would prefer.
All right. Well, I'm going to stay on just in case. So I will, first of all, thank you so much for the opportunity to join you this morning. I had the opportunity to present this obviously a couple of weeks ago to the full collective of DERP, and I'm excited that you're interested in this topic. This obviously was a pretty large report and something that we put a lot of effort and energy into. I think we got some great information here, and I'm excited to share it with you this morning, or if you're in the Eastern time zones this afternoon. So today I'm going to be talking about atypical antipsychotics and their use as adjuvant treatment for major depressive disorder, the clinical evidence, and this is a systematic review. We also performed a meta-analysis with this, and you'll see some of the results of that as we proceed. Our overview for today. So this is going to be the typical process that we follow for systematic reviews. So we'll talk a little bit about background and table setting just to kind of get us the context and the clinical importance of this particular topic. We'll go through our PICOs to assess or to report the populations involved and the other elements that we evaluated. We'll take a look at our key questions. So looking at the efficacy, looking at the harms, and any specific parameters to that you're interested in. We'll go through our methods so that you can get a sense of how we performed this systematic review and meta-analysis. Probably what you're all here for, the findings. So we'll take some time to walk through the findings and what we saw with these particular agents and their efficacy and their harms in this particular clinical situation. And then obviously get to our discussion and kind of how we pulled it all together and how we can take this information forward as an actionable item for you as a state. So for our background, major depressive disorder has a lifetime prevalence of about 21% in the US, and it is ranked as the leading cause of disability worldwide. Part of the reason that we took on this topic, 50% of those with depression will experience recurrent episodes. So we need something that's going to be effective for those that are treatment resistant or potentially getting recurrent issues. So the guideline-based strategies to achieve remission, they include lithium or an atypical second-generation antipsychotic, which is what we'll talk about today, along with an antidepressant in those patients who demonstrate treatment-resistant depression, which will abbreviate as TRD through the rest of the presentation today. And SGAs appear to be a preferred treatment strategy, particularly when you compare to first-generation antipsychotics, and we can probably imagine why with the side effect profiles of first-generation antipsychotics and all of the risk and complications that come along with those agents. The populations we evaluated for this review. So we assessed adults with major depressive disorder. The interventions, just for the early part of this presentation, we broke them up into two groups. We have a group that actually does have an FDA indication for adjuvant therapy for major depressive disorder. So those 5 agents are aripiprazole, brexpiprazole, cariprazine, olanzapine and fluoxetine, and quetiapine. Interventions that do not have an FDA-approved indication, we also assess those, but the agents that fall into that category are acinapine, clozapine, iloperidone, lumateperone, lurasidone, paliperidone, pimavanserin, risperidone, and ziprasidone. So it looks like I got the better end of the deal today. Instead of having to present all of the biologics, it looked like that you were talking about in the previous report. So I know that that's always kind of an adventure working through all of those drug names. The comparators that we evaluated for this, we looked at another listed intervention. So this is the long way to say head-to-head studies, and we did actually find one of those. But I wouldn't get my hopes up. If you're really excited about a head-to-head study, you might be a little disappointed when we actually get to that study and discuss it. We also evaluated standard of care and placebo. But as a note that is a little bit later in the presentation, all of the patients that were in these studies were on some type of antidepressant therapy. So nobody was getting no treatment. They at least had some type of antidepressant monotherapy throughout. Outcomes that we evaluated, we wanted to look at depression severity, and there are obviously a number of different assessment tools that we can use for this particular outcome, and we'll talk about some of those as we go through. We wanted to assess quality of life. We assessed function, and that could either be social function, sexual function, any kind of quality of life or issue of life to improve function. Suicidal behavior or risk, adverse events and serious adverse events, and the study design that we evaluated for this review was a randomized controlled trial. For our key questions, we obviously wanted to assess the effectiveness of these agents. We also wanted to take a look to see if there were any variations about patient characteristics, such as age, duration of major depressive disorder, severity, etc., that might impact the efficacy of these agents. We did the same with harms. So what are the global harms? And then are there any patient characteristics that might make somebody more susceptible to a particular harm, such as age, duration of major depressive disorder, etc. We also evaluated the characteristics of ongoing studies and selected pipeline agents such as pimavanserin and lumateperone. So now to the methods for our project. So if you're familiar with the DERP process, we evaluated using the DERP typical clinical evidence sources. So we searched Ovid Medline, as well as the Cochrane Library, DuckDuckGo, Google Scholar, etc. For ongoing studies, once again, the usual sort of resources there for clinicaltrials.gov, FDA resources, etc. You can see the others there on the screen. For additional sources, we looked at review articles. So any review article that was included in our searches, we evaluated the reference list and bibliography to see if there were any additional studies that might not have been captured in those earlier evidence searches. So we searched the clinical evidence sources from inception to October 20th of 2023, and that's when we began screening and moving through the rest of the process. As I mentioned, we examined the reference lists for systematic reviews. Once we had all of our studies, we assessed the risk of bias for all of the included original trials. We then used the grade approach for the overall certainty of evidence for critical clinical outcomes that we'll talk about in a moment. And once again, clinicaltrials.gov and those other resources for our ongoing studies related to this particular topic. So the risk of bias assessment scheme that we use, if you're familiar with the DERP process, there are 3 levels to the risk of bias assessment. A study can have a low risk of bias, moderate risk of bias, or a high risk of bias, just depending on the criteria that we evaluate for each of these. And there is a rubric that we use to assess the risk of bias for the individual studies. And evaluating the grade certainty of evidence, there are 4 levels to the grade. You can see high is obviously the highest grade that we can recommend or that we can evaluate the evidence with. We have moderate, low, and very low. The outcomes that we focused on for our grade ratings for this particular project, we looked at the Montgomery-Åsberg Depression Rating Scale, or MATTRS scale. That was one of the most common that was used to assess the depression severity throughout the studies. We also looked at the HAM-D17, the Hamilton Depression Rating Scale. This also is a common rating scale used in depression. We looked at CGI-I, which is the Clinical Global Impressions Scale Improvement. So this is a clinician-rated scale to evaluate patients as they progress through treatment. We assessed response. So response is typically going to be defined as a 50% reduction in symptoms from baseline. For harm outcomes, we evaluated the BARS, which is the Barnes Akathisia Rating Scale. So this is something, as the name implies, assesses akathisia and looking for unwanted movement disorder or unwanted movement adverse events for a patient. And lastly, change in body weight, because that can be one of the more significant quality of life or patient acceptance issues whenever they're started on therapy. And so this can be something that can be a barrier to therapy. So we wanted to make sure that we address that as part of this project. As mentioned, we also performed a meta-analysis on the evaluable studies, and so these were assessed with RevMan 5.4. It does also need to be noted that not all study results could be reported and incorporated into the meta-analysis. So on the grade rating, you might see a larger number of studies than what you might see in the meta-analysis graphic that we'll talk about as we move through the presentation today. For the meta-analysis, we focused on those outcomes that were assessed using the grade rating. And if you would like to spend a little more quality time with those meta-analysis figures, those are available in Appendix C. That is part of the report that is available to you from DIRT. So now the good stuff. This is what you came for. So we're actually going to approach the findings we're going to look at all of these agents and we're going to use them or just approach them alphabetically. So, oh, I'm sorry, not yet. We'll get there. And so for the study flow diagram, we wanted to give you an idea of sort of the project flow. So initially we had about 1,402 documents screened. Once we went through the process, we ended up with 96 publications that were eligible for this particular project. And so we had 47 of these that were original studies and then 49 which were additional analyses. So these might be post hoc or secondary analyses, or it might be pooled analyses of similarities within a particular subject or particular agent. Measured in the ones that we'll talk about today, I mentioned the MATTRS and the HAM-D scale are the ones that we'll look at for depression severity. For overall improvement, we'll look at clinical global impressions, response, and remission. Although, admittedly, we didn't spend a lot of time, particularly at least on our presentation, talking about remission. That's a pretty difficult benchmark to meet, especially with as short as many of these studies are that we'll talk about. So there is additional information in the report on remission, but we're not going to talk about remission very much today. And then for adverse events, again, we'll look at the Barnes Akathisia Rating Scale and also another movement adverse event scale called the AIMS, or the Abnormal Involuntary Movement Scale. So some overall context for the findings that we have. So in total, as I mentioned, we've had 47 original studies and 96 publications. Looking at those that have an FDA approval versus those that don't, we had 38 studies that were performed and those that do have an FDA indication for adjunct therapy in major depressive disorder, and 9 of those studies were performed in agents that don't have an FDA approved indication. The population, so we were looking at adults in general, but wanted to note that we actually had 2 studies that looked at older adults. So one study, I believe, 55 and older, and the other 60 and over. The rest of the studies, the other 45, just looked at 18 and over as adults. The comparators that were assessed, so 46 of these studies looked at placebo or monotherapy. And as I mentioned, we had one head-to-head study that evaluated multiple agents within the class. So again, giving kind of a high-level overview of the number of studies and publications. So for those that are FDA approved, for aripiprazole, there were 12 randomized controlled trials. Study sizes range from 52 to 1,522, and you can see a total in of just under 5,000 total participants for these studies. On the right-hand column, you can see study durations, and it ranged from 6 to 12 weeks. Bexpiprazole had 5 randomized controlled trials. You can see the study size ranges there with a total of just under 3,000 total participants in the studies, and study durations 6 to 26 weeks. Cariprazine had 5 total RCTs with just over 3,000 participants with study durations of 6 to 8 weeks. The olanzapine fluoxetine combination product had 5 RCTs with just over 2,000 total participants. And quetiapine had 10 total studies, and you can see again just over 2,100 total participants for quetiapine XR, and study durations of 6 to 12 weeks. So for those that had an FDA-approved indication, we had 37 total studies in this case, in a total of 78 publications, just under 15,000 total participants, and relatively short study durations. And you're going to see that's a theme and a limitation we come back to of 6 weeks for most of them, but up to 26 weeks. So I know some of you are saying, well, wait a minute, you said in the previous slide there were 38 publications in those that were FDA approved. So here is that one lone head-to-head study that we have. So we have aripiprazole versus olanzapine versus lithium. So we have one study and one publication, a grand total of 30 participants in this study, and a total study duration of 4 weeks. So I hope that I lowered your expectations earlier efficiently so that you weren't terribly excited about what we're going to learn from this particular study. For study characteristics for those that did not have an FDA-approved indication, we had 2 RCTs for pimavanserin, and you can see 501 total participants with study durations of 6 to 10 weeks. Risperidone, we had 5 RCTs with just under 1,000 participants with studies in 4 to 24 weeks. And for ziprasidone, we had 2 RCTs, a total of 203 participants, and these studies both lasted for 8 weeks. So as noted here, we didn't find any published studies for the use of acenapine, clozapine, allopurinol, lumateperone, lurasidone, or paliperidone as adjuvant treatment for major depressive disorder. All right, now, as promised, I mentioned that we would get to these alphabetically. So just like Alabama is the first state in the United States alphabetically, we're going to start in the A's with aripiprazole. So just some overall study characteristics. We had 12 RCTs, an additional 23 additional publications with aripiprazole. So 8 of those were secondary or post-hoc analyses, and 15 of those publications were pooled analyses. And again, this had the 2 RCTs that were performed in older adults. And most of these studies did have a run-in period to confirm that the participant did have treatment-resistant depression. Looking at the grade ratings for the outcomes associated with this particular agent with aripiprazole. So this is going to be a comparison to placebo or to ADT monotherapy, ADT standing for antidepressant therapy. So for our efficacy outcomes, we had for MATTERS, the Montgomery-Åsberg Depression Rating Scale, we had 9 RCTs with just under 2,800 participants. We assessed this as a grade of high, and we saw that MATTERS scores typically improved 2 to 3 points during initial treatment. And to give that a little bit of additional context, for most of the studies that for aripiprazole and that we'll talk about for almost all the agents, baseline MATTERS scores was somewhere between 25 to 30. So a 2 to 3 point improvement is fitting in that context of 25 to 30 at baseline, so a roughly 10% improvement from baseline score. For the CGI-I scores, we had 8 randomized controlled trials reporting this outcome. And we assessed this as having a grade of a high. And so we did see some modest improvement in CGI-I scores, typically about 1 point throughout the course of treatment. For response, so that's a 50% improvement in symptom score from baseline for a participant. 9 RCTs evaluated that outcome. We assessed this as having a grade of moderate. And so we did see that aripiprazole showed higher rates of response, and that was about a 10 to 28% absolute change from baseline. And typically you see anywhere from 20 to 40% of total participants in the aripiprazole achieving response as an outcome. So giving a little bit of visual to what I've just mentioned. So this is the meta-analysis plot. And so you can see on the left-hand side, you have the different studies, you have the different differences seen within the individual study. And then on the right-hand side, you can kind of see this plot in the visual. So the top of the plot with the green boxes and lines are the results of that individual study. And then at the bottom, you'll see the black diamond, which indicates the cumulative effect of the pooled, the meta-analysis for those evaluable studies. So you can see in this particular finding for MATTERS, One of the studies didn't achieve significance, but when you pooled all the studies together, you did see an improvement overall in participants that were assigned to aripiprazole. For CGI-I scores, again, you can see the individual studies and their contribution to the meta-analysis. You can see at the bottom, the black diamond isn't particularly prominent down there. It's actually pretty narrow, but it does, again, indicate improvement overall with those taking aripiprazole. But you can see from the individual studies, several of them didn't achieve significance on their own. And finally, for matters response, once again, you can see the individual study contributions and where they fell in terms of their statistical significance. And once again, you can see when combined that we did see some overall improvement with aripiprazole as part of the meta-analysis. Harms findings, so looking at the bars, that akathisia rating scale, 7 RCTs evaluated that, and we saw a grade, or we assessed it as having a grade of moderate, and we saw that aripiprazole showed modestly higher scores, which means they had a slight increase in akathisia. For change in body weight, 11 RCTs assessed this outcome, and we had a grade rating of high. And aripiprazole typically showed between 1 to 1.5 kilogram increase in body weight within the first 6 weeks of therapy. A couple of subpopulations that we wanted to note. So factors that were noted in some of the secondary and post hoc analyses. And so factors that improved the rate of response or remission for individuals was noted that if they were employed or if they had less severe symptoms at enrollment, that those tended to have higher rates of remission and response. A couple of factors that didn't seem to impact response or remission were age or baseline anger or hostility scores. Moving to our next agent, brexpiprazole. So we had 5 RCTs as part of this particular agent and had 12 additional publications, all of those being pooled analyses. And again, most of the studies had a run-in period to confirm that these participants did indeed have treatment-resistant depression. Looking at matters scores for brexpiprazole, so we had 5 RCTs evaluating this outcome. We assessed this as having a grade or certainty of evidence of high, and matter scores typically improved 1.5 to 3 points during treatment. And again, same sort of baseline typical profile of matter score of 25 to 30 at baseline. For the CGI-I scores, you can see 4 RCTs assess this outcome. We had this as a grade of moderate, and we did see some modest improvement in CGI-I scores. However, there were inconsistent results between the 4 studies. For response, we noted 5 RCTs evaluated this outcome, tested as having a grade of moderate, and we did see that brexpiprazole showed higher rates of response, with only a 4 to 12% absolute change within the agent. Once again, assessing the meta-analysis, so giving a visual to what we've just noted. For change in matter scores, we did have 2 evaluable studies for this outcome. And you can see that they're, when combined, that they did show an improvement towards brexpiprazole. Looking at response, so again, that 50 improvement from baseline. You can see, once again, we noted inconsistent results, and you can see that sort of visually on the top 4 studies. Only one of those achieved significance, but once again, when combined all together, you can see a tendency toward an improvement with brexpiprazole in that combined analysis. Brexpiprazole versus placebo in our harms. For the BARS assessment, we had 3 randomized controlled trials, and we assessed this as having a grade of high. We saw that brexpiprazole again had modestly higher scores, so an increase in akathisia. And for change in body weight, 5 RCTs noted this. Again, we had a grade of high on this, and brexpiprazole typically showed a 1 to 1.6 kilogram increase in body weight through the first 6 weeks of therapy. Moving along to cariprazine. Looking at the efficacy outcomes with cariprazine, we had 5 RCTs evaluating the matters score. So we assessed this outcome with a certainty of evidence of high. And MATTERS scores typically improved 1 to 3 points during initial treatment. Once again, typically having a 25 to 30 MATTERS score at baseline. For CGI-I, 5 RCTs assessed this outcome. We had this as a grade of moderate, and so we did see maybe some modest improvement in CGI scores, but they typically weren't significant within the individual studies for this agent. For response, we saw, again, 5 RCTs. We assessed this with a certainty of evidence of high. Cariprazine showed response rates of 1 to 10% absolute change, but they were typically not significant within the individual studies. So looking at the meta-analysis for these outcomes, so cariprazine versus placebo in change of MATTERS. Once again, you can see most of the individual studies didn't achieve statistical significance as the bar crosses that point of no difference of 0, but when combined all together, you can see some improvement in the total combined analysis. For CGI-I, you can see that none of the studies achieved significance. And I know whenever I put up this slide, for those of you that are reading this and seeing it, you're probably a bit skeptical that this slide is correct, but we triple checked it. And yes, just based on the variances and sample sizes, this is how this particular analysis shook out. So kind of just some interesting viewing there. Saw a bit of an anomaly with this slide, but yes, this is correct. But once again, you can see that there was improvement when combining all of these studies together with cariprazine. Looking at response, once again, you can see most of the studies didn't achieve statistical significance on their own, but when combined, we do see some favor toward cariprazine in rates of response. Looking at harms and the bars assessment, we had 5 RCTs assessing this, and we assessed this with a certainty of evidence of high. And once again, cariprazine showed modestly higher scores, so an increase in akathisia. And for the change in body weight, we assessed this as having a greater certainty of evidence of high. And cariprazine showed typically 0.4 to 0.9 kilogram increase in body weight through the first 6 weeks of therapy. So less weight gain than what we've seen in the previous set of agents. Moving along to olanzapine and fluoxetine, so a combination product. You're probably kind of getting into the rhythm now. So for our MATTERS scores, we had 5 RCTs evaluating this outcome. We assessed it as having a grade of high, with olanzapine and fluoxetine improving scores typically 3 to 5 points throughout the studies. For response, we had 4 RCTs reporting this result with a grade of moderate, and olanzapine fluoxetine combination showed inconsistent results ranging from 1% to 18% absolute difference. You can see from the meta-analysis here, so looking at the change in MATTERS scores, we had 2 evaluable studies, and when combined, you can see that there is some favor towards olanzapine fluoxetine combination. Looking at response, you can once again see that most of the individual studies did not achieve statistical significance, but the overall combined analysis did achieve statistical significance just by the barest of margins, but it is significant. Looking at the harms for olanzapine and fluoxetine, looking at the bars assessment, we had 4 randomized controlled trials, but assessed this as having a grade of low. And we didn't really see a much increase in the scores during treatment for this particular assessment. For change in body weight, this was one that was kind of notable. So we actually had this as a grade of high, but olanzapine and fluoxetine actually showed up to a 6 kilogram increase in body weight at the start of therapy. So this was the one obviously associated with the most weight gain among the class that we're talking about today. So our head-to-head study, olanzapine versus aripiprazole versus lithium. So as noted, we only had 1 head-to-head study, a grand total of 30 participants. They were divided evenly, so 10 in each of the treatment arms for olanzapine, aripiprazole, and lithium. And there was no significant difference between the therapies at week 4. And so a very, very short study for this particular trial. So for quetiapine versus placebo, looking at the MATTERS score, so we had 5 RCTs assessing this, and so we had this as a certainty of evidence or grade of moderate. And MATTERS scores were typically improved around 3 points during initial treatment, and significance was inconsistent. So you're probably seeing the theme now for these second-generation antipsychotics. We're seeing roughly about a 3-point improvement in MATTERS scores throughout the class. For CGI-I, we had 6 RCTs. We assessed this as having a grade of high, and we saw again a relatively modest 1-point improvement in the CGI-I scores. For response, we evaluated this as having a certainty of evidence of high, and we did see that quetiapine showed consistently higher rates of response and a 10 to 13% absolute change. Looking at the combined analysis for quetiapine and change of MATTERS, you can see that in the combined analysis, this is one that did not achieve statistical significance even when combining the groups. In response, you can see that the 2 individual studies both achieved significance as well as the combined analysis between them. For our harms outcome for quetiapine versus placebo, for our BARS assessment, we assessed this as having a grade of low and that there were no significant differences reported. For change in body weight, we assessed this as having a grade of high, and quetiapine typically showed about a 1 kilogram increase in body weight in the first 6 weeks of therapy. So we did have an assessment or 2 here for quetiapine versus lithium as a different pharmacologic agent. So we had 2 RCTs that assess this particular matchup. And so for efficacy, we saw in the MATTERS score, we had this as a grade of low. Quetiapine showed a significant improvement in MATTERS in one study and no difference in one study. For CGI-I, we assessed this again as having a grade of low. Same thing, we saw a significant difference in one study, but not the other. And for response, we assessed this as having a very low, and there was no difference between the groups, with both groups reporting sort of unusually high response rates. So it seemed out of the norm from what we saw from the other studies. For harms in quetiapine versus lithium, we had 1 RCT assessing change in body weight. They didn't actually report how much weight that the participants actually gained, but they just said that more participants reported weight gain as an adverse event in the quetiapine group. And we assessed this as having a certainty of evidence of low. So moving along to risperidone. So risperidone versus placebo for our efficacy outcomes for MATTERS. We assess this as having a grade of low, and that MATTERS scores improved between 1 to 7 points during initial treatment. For HAM-D scores, they actually reported those for these agents. We assessed this with a grade of low, and we saw inconsistent improvements in the HAM-D17 scores. For response, we had 2 RCTs that assessed this. For grade, we had it as moderate, and we did see risperidone showing high rates of response with 15 to 22% absolute difference between risperidone and placebo. Again, putting the visual to our findings, you can see here for change in MATTERS scores, when we assessed or we combined all of these, the assessment was that there was no significant difference between risperidone and placebo. For change in HAM-D, same finding that we didn't see a significant difference within the pooled, or excuse me, the combined analysis for this particular outcome. Harm outcomes for risperidone. For BARS, we had 2 randomized controlled trials, but assessed this as a certainty of evidence of high. And risperidone did not significantly worsen BARS scores, so we didn't see an increase in akathisia. In change in body weight, once again, we graded this as high, but they didn't report the amount of weight gain. They simply reported that participants in the risperidone reported weight gain as an adverse event, but again, didn't specify how much weight was gained. All right, so for today's presentation, we're to everybody's favorite drug with ziprasidone. So why is it everyone's favorite drug? Because it's the last one. It starts with Z. So our final drug we'll talk about today is ziprasidone versus placebo. So for our matters, we had one RCT with 64 participants, and we assessed this as having a certain of evidence or grade of very low. And so matters scores improved by 4 points, but this was not significant. For CGI-I, we had 2 RCTs and a grade of moderate, and we saw that ziprasidone did show an improvement in one study and no improvement in the other. And for the HAM-D, we assessed this as having a grade of moderate. Once again, ziprasidone showing significant improvement in one study and no improvement in the other. Due to the low number of studies, we weren't able to perform a meta-analysis on those outcomes. And for bars, for the akathisia rating scale, one study, a grade of very low, and there were no clinically relevant changes that were reported in this particular study. For the ongoing studies that are relevant to this particular topic, we actually identified 13 ongoing studies evaluating SGAs as adjuvant therapy for MDD. So we can see through here, we have 2 studies of aripiprazole. Comparators of relevance are bupropion, venlafaxine, and escitalopram. And completion dates anywhere from April 2021, so the results haven't been posted yet, to December of 2025. We have 5 studies with brexpiprazole, comparators of relevance for placebo, citalopram, and escitalopram. Once again, we have estimated completions where some of these studies may already have been completed but haven't reported results all the way through April of 2029. We have 2 studies with cariprazine comparing placebo, compared to placebo with a completion date of September 2021. Again, results haven't been posted for that one yet. Additional studies, 3 with lumateperone, comparing it to placebo. Estimated completion is February of 2024 through May of 2024. And 1 study with quetiapine evaluating it against amantadine and pramipexole, sample size of 150, and it's estimated to be completed in September of 2024. So now to our discussion and bringing all of this information together. So to kind of tie it all up, SGAs are a guideline recommended addition to ADT in patients that have treatment-resistant depression. As noted, most agents we saw typically a 2 to 3 point improvement in MATTR scores during the first 6 to 8 weeks of therapy. Response rates were somewhat inconsistent overall between this group of therapies we've talked about today. Movement adverse events were typically slightly higher, as noted by the bars and AIM scores, but it's not really known if these would improve with continued therapy. So you see an initial worsening, but as the patient continues therapy, they adapt, and this is something that resolves with continued treatment. Weight gain is a significant concern, and it was consistently reported. We saw usually about a 1 kilogram increase, although we did see a much larger increase with the olanzapine fluoxetine combination of up to 6 kilograms. So something that we need to be mindful of if we want to start patients on these particular agents so that we can counsel them appropriately so they have appropriate expectations. Grade ratings were generally high to moderate and consistent results between the study groups with aripiprazole and brexpiprazole. But I think one of the things that consistently came up throughout the project was the clinical efficacy. I'm going to come back to that in just a moment. Grade ratings were a bit more variable with other therapies within this class, and I think you noted that particularly with those that had fewer numbers of studies and smaller studies. A consistent limitation that we encountered evaluating these studies was the short duration. Most of them were 5 to 8 weeks. So assessing initial response, we got some information there, but assessing long-term therapy in terms of both harms and efficacy, we really just didn't get a great sense of that throughout the studies. A lack of head-to-head studies. Again, we only had 1, and it wasn't particularly impressive. And a lack of long-term follow-up. So once again, seeing if efficacy is maintained for the long term or if adverse events or harms stay, resolve, or even get worse with continued therapy. So in the clinical efficacy debatable part, one thing that one of our clinicians here at Auburn University who does specialize in psychiatry, so talking about when we see about a 3-point, we'll say on average, improvement in these patients, she said that while you're still looking for response, so cutting in half those improvements are cutting in half the symptoms from baseline. A lot of times what they see clinically in their practice is that even though the rating scales don't improve a whole lot, a patient will just sort of holistically say, well, I feel better. And so clinicians tend to, if the patient is reporting that they feel better, even though they can't necessarily quantify it, the clinicians are somewhat hesitant then to remove the agent from the patient. So the clinical efficacy part, again, depending on practice, might have some variation there. So with that, I'll be happy to take any questions from the panel.
Thanks so much, Wesley. That was an excellent comprehensive review. Committee, oh, I guess I'll ask for stakeholder input first. I don't see any listed.
I'm
Yeah.
sorry, this is Greg. Real quick for Wesley, a
Yes.
quick question. I didn't see any noted on your slide deck, but I was just wondering if you saw any up and coming comparative studies.
When you say comparative studies, are you talking about like head to head?
Yes, excuse me, head to head studies between different agents, such as, you know, aripiprazole versus lithium, anything like that coming up.
I did try to note that in the comparators. No, most of there was only the one with the amantadine and pramipexole comparing to quetiapine. That's the only head-to-head study that was currently in the pipeline based on our searches.
Okay, thank you. I'm just trying to note those as you are.
Other questions from committee members? No? Okay. And I don't see any hands raised from stakeholders. I guess, Nanya, if you can take us to the motion.
Mhm. Okay. I kind of start copying this in here, and I'm going to make an update on the lists of drugs and then slice and dice.
So yeah, I guess the older ones, the clozapine, iloperidone, probably need to be removed. The lurasidone, yeah. Perfect, thank you.
The top, it has the has different indications, just like the last one.
Yeah,
So no, it says autism spectrum disorder will say treatment for MMD.
Yeah.
Oh, yeah.
Let me.
I think she's still cleaning up the
Okay.
the list of meds on the paragraph. But yes, thank you for calling that out.
Thank you. Okay.
Not to interrupt, but this is noted in the report, but I didn't mention it today. I noticed that you had pimavanserin off to the side. So the makers of pimavanserin actually abandoned their effort to get an indication in this particular space due to lack of efficacy. Again, that's in the report, but I didn't mention that as part of the presentation today.
I appreciate that, Wesley. Thank you. So maybe we can just remove it from the list altogether on the left. Yeah, perfect. You can remove that. Oh, no, we can leave the major depressive disorder.
I would just keep going and remove all the way through disruptive behavior disorders and then replace it with the top, what it says on the top, as adjuvant therapy for the treatment of MDD is what it probably should say.
So right there.
Yeah.
Okay.
No, keep
No.
going all the way. No, that's fine. Just also keep going with erasing.
All the way.
Keep going all the way through disruptive behavior disorders. Perfect.
Oh, thank you.
And then we could say for treatment as adjuvant therapy for the treatment of MDD. Or I don't know if that's redundant.
This? No.
Just the part as adjuvant therapy for the treatment of MDD. Yes, please. We'll see how that rolls. Okay. After considering the evidence of safety and efficacy for the treatment, for the adjuvant treatment, so I guess you would remove the treatment. No, sorry. The treatment comes up twice. So if you can just remove. Yeah. For the adjuvant therapy of the treatment of DD.
We need to call out that olanzapine wasn't used in isolation in terms of like with fluoxetine, or can we just leave that out?
So this is Ryan, and typically when we see drugs with combinations from two different Washington PDL drug classes, we tend to not use them within the motion. So if you think about the asthma COPD, we do have a lot of those combinations there. For other drug classes, like with diabetes drug classes, for some of the statins, we typically don't have those combinations listed in the motion. Also, one of the ways that those studies were done is it was olanzapine and fluoxetine versus placebo or fluoxetine monotherapy. So essentially, a lot of those studies are looking at the effect of adding in olanzapine. So we opted to just leave it as olanzapine here in the motion, knowing that it was studied in association with fluoxetine.
Thank you.
Go ahead, Peter.
Thanks. I'm trying to recall the language that we typically use, but just past halfway in the motion that's showing on the screen, there's a sentence that says something like these medicines should be on the Washington State Preferred Drug List. And I thought we usually use the language are eligible to be on the preferred drug list. And I'm wondering if maybe I've got that wrong, or maybe this class is being treated differently as a protected drug class. And so
This
all
is
of them.
Ryan. Yes, yeah. For most drug classes, we typically have language that says and are eligible to be preferred. I believe years ago, the P&T committee made the recommendation to the agency to try to have as many of these drugs be preferred, so that way when a patient is starting therapy, that the providers do have the option. In fact, looking at the current Washington preferred drug list, all of these drugs are listed as preferred, and so that's typically how we've reviewed this class in particular as a protected class for antipsychotics.
But this is Donna, but we do require generics first, and this is old language taken from a different motion, from a different review. So I think it is due time to update the language to refer how these drugs are, that they're all eligible.
Yeah, I mean, I think it's reasonable to copy the language that we have for our other motions about eligibility, and then leaves a little bit more flexibility. I don't feel super strongly about it, but I think for consistency's sake, and I think it would make sense. Curious to hear if other members of the committee feel strongly one way or another.
Other comments from the committee members? I think that's a great point. And I assume yeah, go ahead.
This is Donna. I mean, all of these drugs are already on the PDL in the antipsychotic class. I don't think to be honest with you, I don't even think it's necessary unless you feel strongly about using these in major depressive disorder. We don't usually prefer drugs based on indication. So these drugs are already preferred on the PDL. So I don't know what this motion is going to do to the status of these drugs on our PDL.
Okay. That's helpful, and that's a good point, that these medications are preferred or not preferred, period, not really by indication. Other comments?
So this is Laura Bestie. Donna, are you recommending then that the line that says should be preferred on the Washington preferred drug list just be removed?
This is Donna. I'm questioning whether we even need to make a motion. I mean, and just take the presentation that we saw as information only, because unless you're going to say that these need to be preferred for a major depressive disorder, there's no policy. We don't require diagnosis for these. If they're being used for depression, then they are on label, then they're just approved based on, their preferred status as they are today.
Okay, so I'm hearing that maybe we don't even need a motion. We can move forward, unless there are questions.
I think you need to have a motion of whether or not you need a motion.
Why don't we just do the motion because we already wrote it and it seems fine. It's basically just, certifying that we agree that all of these are still effective. I don't know if there's any use to having this sentence about pregnancy too, but if that's sort of like what's usually written, I feel like we might as well just keep it.
Did we want to remove that? This is Laura Bestie, the pimavanserin from the side on the
Yes.
left column.
Yeah, great point. Let's do that. Nanya, if we can remove the grayed out medication on the left column, the pimavanserin.
This is Christy Wyland. Donna, I'm curious if we choose to move towards a motion, but it's not necessary. Is it more work on the back end for you guys, or if we don't actually need this?
So Ryan, this could be similar to how we do some of the other surveillance reports where we just present, here's the update in the class, here's the evidence, and then we can do a cost analysis on the back end and update the PDL as needed.
Okay, so with that, Ryan, do we need to change the language at all, or should we move forward with the motion as written? If the committee members agree.
This is Laura Bestie, or do we just update the surveillance accepted as adequate column?
Yeah, we could update that to say the report accepted as adequate and reiterate the prior motion.
Yeah, Kavita here. I think the difference in the language is like, what was it? A systematic review versus a surveillance, because this was not just a surveillance report. So I don't know if we'll have to then change all of that too. So, thoughts about moving forward with the motion as is, and then that way we can just approve the systematic review as well.
Probably going to spend more time just talking about whether or not we need a motion than just passing a
Exactly.
motion. So I would say let's just make the motion and continue on.
All right, who'd like to go for it?
This is Laura Bestie. I make a motion that after considering the evidence of safety, efficacy, and special populations for the adjuvant therapy of the treatment of MDD, I move that aripiprazole, brexpiprazole, cariprazine, olanzapine, quetiapine, risperidone, and ziprasidone are efficacious for their approved FDA indications and should be preferred on the Washington preferred drug list. Second generation antipsychotics cannot be subject to therapeutic interchange in the Washington preferred drug list. The preferred drug list should include at least one medication that is considered safe in pregnancy.
This is Peter Barquette. I'll second the motion.
All in favor, please say aye.
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 Any opposed? And the motion carries. Thank you all. And great. We are not only on schedule, but a touch ahead of schedule. Do we want to keep going or should we take a break?
We can
All
go
in
ahead
favor
and
of a break. Go ahead.
I was going to say we can go ahead and take a break.
Okay, sounds good. So we'll be back in 15 minutes. So 10 past 11.
Sounds good.
All right. Thank you. Thank you, Wesley. We can make sure we have quorum. Looks like we do. All right, so going into the next part of our morning is the multiple sclerosis report by Rachel McCausland and Courtney Cooper. Are you on?
Hi, yes, good morning.
Hello, good morning, welcome.
Hi, good morning.
The floor is yours.
Great, thank you. My name is Rachel McCausland. I'm one of the researchers from the center, and I'll be presenting this update along with my colleague Courtney. We're presenting on disease-modifying therapies for multiple sclerosis, and this is an update of prior reports. Next slide. Just briefly, an overview. We'll start with the background of MS and disease-modifying therapies. We'll go through our PICO criteria, the key questions that we were looking to answer, our methods for this review, findings, and finally a discussion of those findings. Next slide. We have a pretty extensive list of abbreviations that are used throughout this presentation. A lot of these are standard for reviews in statistics, and I won't go over them, but there are a few that are specific to this topic that I want to touch on. Starting with DMT, that's for disease-modifying therapy. And then we have MS, which is multiple sclerosis. We have 4 types of MS. There's CIS, clinically isolated syndrome, RRMS, which is relapsing remitting MS. SPMS is secondary progressive MS, and PPMS is primary progressive MS. We'll go over those in further detail later. These are just the acronyms. And then we have 2 outcomes measures, EDSS, which is the Expanded Disability Scale Score, and MSFC, which is the Multiple Sclerosis Functional Composite Score. Next slide. As I mentioned, this is an update. This report the prior report was completed in 2020. But as you can see here, the topic has been reviewed and updated many times in the past. The original report dates back from 2007, and that report and all subsequent updates are archived in the DERP clearinghouse. Next slide. So now I'm going to go into a little bit of the background of multiple sclerosis and disease-modifying therapies. Next slide. Myelin surrounds and insulates neurons and allows efficient transmission of nerve impulses. In multiple sclerosis, the body's immune system attacks the myelin. This leads to neurologic dysfunction. Symptoms of MS include sensory issues such as numbness, muscle weakness or spasms, vision problems, dizziness, and trouble walking or speaking. Next slide. Multiple sclerosis is the most common immune-mediated inflammatory demyelinating disease of the central nervous system. The prevalence of MS has been increasing over the past 5 decades. A 2019 population-based estimate found that 1 million adults were estimated to be living with MS in the US alone. MS is the most common disabling neurological disease of young adults, with symptom onset typically occurring between the ages of 20 and 40 years. Next slide. MS is currently categorized into 4 main types, although the pattern and course of MS is unpredictable and can vary substantially from person to person. First, we have clinically isolated syndrome, or CIS, and that's the first episode of neurologic symptoms caused by inflammation and demyelination in the central nervous system. Then there's relapsing remitting MS, or RRMS, and RRMS is characterized with clearly defined attacks of new or increasing neurologic symptoms, followed by periods of partial or complete recovery, also known as remission. RRMS can be further characterized as either active, with relapses evidence of new MRI activity over a specified period of time, or both. or it can be not active. Then we have SPMS, or secondary progressive MS, and that's progressive worsening of neurological function or disability accumulation from RRMS, with or without occasional relapses, minor remissions, and plateaus in severity. Finally, we have primary progressive MS, or PPMS, and that's characterized by neurological function worsening or disability accumulating from disease onset, with occasional plateaus in severity, temporary minor improvements, or acute relapses. Progressive relapsing MS was an older term that has now been eliminated. People who were previously diagnosed with progressive relapsing MS would now be considered to have primary progressive MS, and so that's not listed here in our types of MS. Next slide. For some people, visual helps to understand, so that's what we have here. And starting on the left, you see RMS, which has clearly defined attacks, those are the relapses, of new or increasing neurologic symptoms. In remissions, all symptoms may disappear, or some symptoms may continue and become permanent. However, during those periods, the disease does not seem to progress. And in the middle, you see SPMS, the secondary progressive MS. And some people diagnosed with RMS eventually go on to have a secondary progressive course in which neurologic function worsens progressively or disability accumulates over time. In SPMS, the person may have occasional relapses as well as periods of stability. And there on the right, you see PPMS, that's a primary progressive MS. That's the neurologic function worsening or disability accumulating as soon as symptoms first appear. PPMS can have those brief periods of stability and periods of increasing disability with or without a relapse or new MRI activity. CIS, a clinically isolated syndrome, is not pictured here because it is just the first case of neurologic symptoms. MS is unpredictable, can vary substantially from person to person. These are the descriptors that are currently in use, but an international committee organized by the National MS Society and the European Committee for Treatment and Research in Multiple Sclerosis have proposed a new way of classifying MS as a continuous disease process. A change in classification will take years, however, so this report uses the current system of dividing MS into different disease types, since that's what is currently used in the available research. Next slide. At the time of this report, the FDA has approved 19 disease-modifying therapies for multiple sclerosis and clinically isolated syndrome. The aim of these DMTs is to reduce the number of relapses, delay progression of disability, and limit new MS disease activity as seen on MRI. Next slide. Over the next 3 slides, we have a table with the disease-modifying therapies that are included in this report. There's 17 DMTs that are indicated for people with relapsing forms of MS, which includes clinically isolated syndrome, relapsing remitting MS, and active secondary progressive MS. DMTs that are prescribed for people diagnosed with CIS, those that are experiencing the first episode of neurological symptoms, have the goal of preventing or delaying a second attack. That second attack is also known as conversion to MS. Next slide. The second line down, you see siponimod. In March 2019, the siponimod labeling was the first to explicitly describe that relapsing forms of MS include CIS, RMS, and active SPMS. All sponsors of other DMTs approved for the treatment of relapsing forms of MS have since updated their indication statements to conform with this contemporary nomenclature. It's important to note, though, that DMTs approved for relapsing forms of MS are not indicated for people with non-active secondary progressive MS. There are 2 DMTs, cladribine and alemtuzumab, you can see those here on the screen, that are indicated for people with relapsing remitting MS and active SPMS who have had an inadequate response to other DMTs. Unlike the other DMTs that are approved for all relapsing forms of MS, cladribine and alemtuzumab are not approved for people diagnosed with CIS. And then one of these DMTs, ocrelizumab, the third one down on the screen, is also indicated for primary progressive MS. This is the only DMT currently FDA approved for PPMS. Next screen. This is just the final listing from that table. Of note, there are 19 FDA-approved DMTs for MS, but 2 of those, metoxantrone and adalimumab, are not included in this table because they were excluded from the prior DERP reports on DMTs for MS and have remained excluded from this review. This table is also included in the complete report, and you can look over it in detail there if you need to. Next slide. Our PICO criteria. Population that we focused on was adult outpatients aged 18 years and older with multiple sclerosis, including relapsing remitting, secondary progressive, and primary progressive. We also looked at adult outpatients with clinically isolated syndrome, those having their first demyelinating event or symptoms. The interventions that we looked at were those listed DMTs in the previous table with FDA approval for the treatment of multiple sclerosis and clinically isolated syndrome. Next slide. Our comparators that we looked at were any other listed intervention. We only looked at head-to-head comparisons. The landscape of DMTs for multiple sclerosis has rapidly evolved over the past 20 years. The previous report did look at placebo-controlled. However, because there are so many different FDA-approved DMTs as treatment options, we wanted to limit this evidence review to the comparative effectiveness and harms of DMTs for MS. So placebo-controlled comparisons were excluded. Except for RCTs evaluating DMTs for clinically isolated syndrome, placebos were included as a comparator. Outcomes that we looked at were relapse, disability, quality of life, functional outcomes, persistence, or for those with CIS, conversion to MS diagnosis, which is the second instance of an attack or the first relapse. We also looked for adverse events, overall adverse events, serious adverse events, withdrawals due to adverse events, and specific adverse events such as hepatotoxicity. Next slide. Study designs. We looked at randomized controlled trials, placebo-controlled trials for CIS only, and retrospective and prospective cohort non-randomized studies comparing an intervention type with another for outcomes on harms only. We did not look at efficacy in those cohort studies. We required a 12-week study duration or longer for all included studies, and for those prospective cohorts reporting on harms, we also required a minimum total sample size of 1,000. Next slide. Key questions that we answered. KQ1, what is the effectiveness of DMTs for multiple sclerosis? KQ2, what is the effectiveness of DMTs for people with clinically isolated syndrome? KQ3, do DMTs differ in harms by indication, meaning those with MS or CIS? Do the effectiveness and harms vary by subgroup, was KQ4. Those subgroups, for example, would be participant characteristics, use of prior DMTs for MS, subtype of MS, presence of comorbidities, or antibody status. And finally, KQ5, what are the characteristics of ongoing studies of DMTs for MS? Next slide. Our methods. Next slide. We limited searches of evidence sources. We searched for clinical evidence sources. We limited the searches of those evidence sources to citations published after the previous DERP report searches, which were done on October 15th in 2019, with an updated Ovid Medline search on February 3rd, 2020. For newly approved DMTs that were not included in the previous DERP reports, we searched from database inception. We checked all the studies from previous reports against our inclusion criteria. So specifically, we looked for those placebo-controlled trials that were previously included, and we excluded them from this updated report. We assessed the risk of bias of individual studies. We combined the studies using Review Manager for major outcomes. We used the grade approach for major outcomes. And finally, we searched at clinicaltrials.gov for ongoing studies through the end of 2023. Next slide. As I mentioned, the risk of bias of individual studies was assessed. We used the standard DERP risk of bias assessment for low, moderate, or high, low being clear reporting methods and mitigation of potential bias and conflict of interest, up through high with clear flaws that might introduce serious bias. Next slide. We use the grade approach for certainty of evidence across the key outcomes, which were relapse, disability progression, change in disability as assessed using the EDSS, change in function assessed using MSFC, persistence, and serious adverse events. Just as a little reminder, GRADE's approach to rating the certainty of evidence is based on a 4-level system. There's high, moderate, low, or very low. The GRADE approach classifies randomized controlled trials as initially starting at high certainty and observationals as initially starting at low certainty. By assessing the 5 domains, which are risk for bias, inconsistency, indirectness, insufficient precision, and publication bias, that certainty can be rated down, or in the case of large effects, existing dose-response relationships, or plausible confounders, it can be rated up. Next slide. Our findings. Next slide. So as I've mentioned, this was an update, and we brought forward 109 studies that were included in the previous report. Combined with database searches, this resulted in 3,614 unique records. We screened 741 at the full text stage, of which 612 were excluded. If you want to see exclusion reasons by studies, those are included in Appendix D of the full report. If you direct your eye down to the bottom left in the green square, for this update, we included a total of 38 RCTs, 45 cohort studies, and 39 sister publications. This includes newly identified studies and the studies that were pulled forward from the previous report. Next slide. So here we can see another breakdown of those final inclusions. There were 122 total inclusions, 98 were previous inclusions, and 24 were new studies. Of those 24 new studies, we had 6 RCTs, 15 cohort studies, and 3 sister publication or ancillary studies. Of the 6 RCTs that we included, all 6 included populations with MS. We did not identify any new studies looking at clinically isolated syndrome. And for those 6 RCTs, there were 4 different head-to-head comparisons. Next slide. Now we'll go into those findings in a little bit more detail, starting with the comparative effectiveness and harms for multiple sclerosis. Next slide. So this is a diagrammatic representation of the RCT evidence that was eligible for this report for the MS population. I realize that there's a lot of information on one page, so in the next slides, we'll zoom in on some important parts in just a moment, but I wanted to give you a high-level overview of all of the evidence. You'll see here that the newly approved DMTs are in green. The 4 new head-to-head comparisons are shown with bold connections, and the other evidence shown here was identified in the 2020 report. Next slide. So here, zoomed in, we're focusing on the 4 new head-to-head comparisons. there were 3 newly approved DMTs, ofatumumab, ponesimod, and ublituximab, and they were each compared with teriflunomide. We also identified a comparison of glatiramer acetate with fingolimod. Although fingolimod and glatiramer acetate were included in the previous report, this specific head-to-head comparison is new. Next slide. If you'll direct your attention to the right, you'll see that there are 5 FDA-approved DMTs, diroximel fumarate, cladribine, monomethyl fumarate, siponimod, and peginterferon beta-1a that we did not identify any eligible RCTs for. Next slide. And here we see the portion of the diagrammatic representation that focuses on the previously reviewed head-to-head comparisons and different dosing schedules comparisons that we did not find any new evidence for. Next slide. The previous report that we just saw in that diagram included 11 head-to-head comparisons. There were 23 RCTs and 4 different dosing schedule comparisons. We did not identify any new evidence for these previously reviewed head-to-head comparisons. The findings from the 11 previously reviewed head-to-head comparisons have not changed since the previous report, and because of that, we will not be discussing those specific studies in this presentation. The findings from specific studies remain included in the report, and the slides from the 2020 presentation with the findings from those specific studies can be found at the end of this presentation. Next slide. As previously mentioned, for patients with MS, we identified 4 new head-to-head comparisons evaluating the efficacy and harms of DMTs in patients with MS. 3 new DMTs, ofatumumab, ponesimod, and ublituximab, were each compared with teriflunomide. And we also identified a new head-to-head comparison of fingolimod with glatiramer acetate. 3 of the studies included participants with both relapsing remitting MS and secondary progressive MS. 1 trial was limited to only relapsing remitting MS, and 2 trials included relapsing forms of MS. The studies ranged in length from 12 to 30 months. Next slide. The first comparison that we'll be discussing was the ofatumumab 20 milligrams versus teriflunomide 14 milligrams. We identified 2 RCTs with a total sample size of 1,882, evaluating the efficacy and safety of subcutaneous ofatumumab compared with oral teriflunomide in patients with RRMS or SPMS. So it's the Asclepios 1 and Asclepios 2 trials. They were identically designed, conducted concurrently, and reported together in one publication. Each trial was powered for the primary endpoint, the annualized relapse rate, and the combined trials provided the sample size and power for the pre-planned meta-analysis of disability worsening. Participants in the subcutaneous ofatumumab group also received oral placebo, and participants in the oral teriflunomide groups also received subcutaneous placebo, corresponding to the active drug in the other group. We assessed the Asclepios trials to both be at moderate risk of bias because of concern around author conflicts of interest and sponsor involvement in the study design and analysis. Next screen. A meta-analysis of the 2 Asclepios trials found that ofatumumab significantly reduced annualized relapse rates at 30 months. The pooled analysis of the 2 trials found that ofatumumab significantly reduced the risk of disability worsening at 6 months, but there was no significant difference in confirmed disability improvement at 6 months. There was a moderate certainty of evidence for both of these findings. Next slide. Study completion, which is an indirect measure of persistence, was found to be significantly higher for participants in the ofatumumab group, also a moderate certainty of evidence there. There was no significant difference in the rate of serious adverse events. However, this was rated with a low certainty of evidence. Disability progression and changes in function with the MSFC were not evaluated in the Asclepios trials. Next slide. We identified one RCT with a total sample size of 1,133 participants, evaluating the safety and efficacy of oral ponesimod compared with oral teriflunomide in patients with relapsing remitting MS or SPMS. This was the Optimum trial, and we rated it at moderate risk of bias because of concerns around attrition, author conflict of interest, and sponsor involvement in all aspects of the study. Next slide. In the Optimum trial, it was found that ponesimod significantly reduced relapse rates at 108 weeks, so right at 2 years, with a moderate certainty of evidence. Changes in disability. There was no significant difference between ponesimod and teriflunomide at 12 weeks. But there was a very low certainty of evidence. Next slide. Similarly, there was no significant difference in study completion, which is that indirect measure of persistence, and no significant difference in rates of serious adverse events. These were both rated at a low certainty of evidence. Disability progression and changes in function were not evaluated in the Optimum trial. Next slide. We identified 2 RCTs with a total sample size of 1,094, evaluating the efficacy and safety of intravenous infusions of ublituximab compared with oral teriflunomide in patients with relapsing MS. This was the Ultimate 1 and Ultimate 2 trials, and similar to our previous discussion, these ones were identically designed, conducted concurrently, and reported together in one publication. The authors analyzed results separately for primary outcomes in the 2 trials, but they conducted a pre-specified pooled analysis for selected secondary and tertiary outcomes. Participants in the intravenous ublituximab group did receive an oral placebo, and participants in the oral teriflunomide group received an IV placebo, corresponding to the active drug in the other group. We assessed the Ultimate trials both at high risk of bias because of author financial conflicts of interest and a high level of sponsor involvement in study design, analysis, and publication. Next slide. A meta-analysis of the Ultimate 1 and Ultimate 2 trials found that ublituximab significantly reduced relapse rates at 96 weeks with a moderate certainty of evidence. The pooled analysis of the 2 Ultimate trials found that there was no significant difference between ublituximab and teriflunomide for changes in disability at 12 weeks. Next slide. There was no significant difference in study completion and no significant difference in rates of serious adverse events. The persistence was rated with a very low certainty of evidence, and the serious adverse events with a low certainty of evidence. Change in function, as measured by the MSFC, was reported in the Ultimate trials. However, it was reported as a tertiary endpoint from which no conclusions could be made. Disability progression was not evaluated. Next slide. Finally, we identified 1 RCT with a total sample size of 1,064, evaluating the safety and efficacy of oral fingolimod 0.25 milligrams and oral fingolimod 0.5 milligrams, each compared with subcutaneous injections of glatiramer acetate in patients with relapsing remitting MS. This was the ASSESS trial, and we evaluated it to be at moderate risk of bias because of concerns around the number of enrolled participants, lack of participant blinding, and author conflicts in interest. The ASSESS trial enrolled fewer than half the planned number of participants, which affected the power calculations, and randomization ratios that were requested by the FDA were not possible. Although the trial was rater blinded, a double-blind design was not used for this trial. Investigators stated that they did not use a double-blind design because it would have required double dummy treatment and thus daily sham injections for the 2 fingolimod arms. Next slide. Fingolimod was found to significantly reduce relapse rates that's the fingolimod 0.5 milligrams was found to significantly reduce relapse rates at 12 months. There was no significant difference with fingolimod 0.25 milligrams and glatiramer acetate at 12 months. This was a moderate certainty of evidence. For changes in function, similarly, the fingolimod 0.5 milligram significantly improved functional disability at 12 months, but there was no significant difference with fingolimod 0.25 milligrams and glatiramer acetate at 12 months, with moderate certainty of evidence as well. Next slide. Study completion, which is that indirect measure of persistence, was higher for both fingolimod groups. And of those participants who completed the study, higher proportions of the fingolimod groups received the study drug until completion. There was no significant difference in rates of serious adverse events, low certainty of evidence there, and disability progression and changes in disability were not evaluated in the ASSESS trial. Next slide. KQ2 and 3, the comparative effectiveness and harms for clinically isolated syndrome. Next slide. So the previous report identified 5 placebo-controlled comparisons in 8 RCTs and 1 dosing schedule comparison for people diagnosed with CIS. We did not identify any new evidence for those previously reviewed comparisons. So similar to what we discussed previously for the MS population, the findings from the previously reviewed comparisons have not changed since the previous report, and we will not be discussing the specific studies in this presentation. Findings from those specific studies remain included in the report, and slides from the 2020 presentation with the findings from specific studies can be found at the end of this presentation. Next slide. KQ4 was looking at variations by subgroup. Next slide. We looked for variation by subgroup across all 43 RCTs reviewed, including the 6 new for this report. Participant factors such as age and prior treatment may change the effectiveness of treatment, but subgroup analyses are not reported consistently across studies, which limits our ability to draw robust conclusions. The presence of neutralizing antibodies does not appear to be associated with a reduction in the effectiveness of treatment. Next slide. And from here, I'll be passing the mic over to my colleague, Courtney Cooper.
Thank you, Rachel. We will revisit key question 3, do DMTs differ in harms by indication when considering evidence on people with MS and CIS? And this time we'll be reviewing the eligible cohort studies. So in contrast to the RCT evidence identified for this updated report, we identified new cohort study evidence that evaluated some of the same comparators and outcomes as the cohort study evidence identified in 2020. Therefore, we have integrated the newly identified evidence with findings that have remained unchanged from the 2020 report where applicable. So in summary, we identified 45 cohort studies, 15 of which are newly identified for this update. The majority of studies were assessed as moderate risk of bias because of concerns about author conflicts of interest and industry funding. The rest were assessed as high risk of bias due to additional concerns about adjustment for confounding. 35 cohort studies, 11 of which are newly identified for this update, reported on treatment discontinuation or switch. 12 cohort studies, 6 of which are newly identified for this update, reported on serious adverse events. There is an overlap of 2 cohort studies which reported on both treatment discontinuation or switch and serious adverse events. And additionally, we did not identify any direct comparison of harms by indication. As mentioned on the previous slide, 35 cohort studies, 11 of which are newly identified for this update, reported on treatment discontinuation or switch. Overall, from the evidence reviewed, treatment discontinuations or switches appear to be significantly lower with fingolimod and dimethyl fumarate. Notably, a number of cohort studies investigated treatment discontinuation or switch of oral DMTs compared with the injectable DMTs. However, not every FDA-approved DMT was included in the reviewed studies, and the reported outcomes are not generalizable to all categorically oral or injectable DMTs. Therefore, uncertainty does remain regarding the risk of treatment discontinuation or switch for oral DMTs compared with injectable DMTs. Additionally, 12 cohort studies, 6 of which are newly identified for this update, reported on serious adverse events. It's important to note that serious adverse events are higher in some DMTs. Specifically, the risk of liver injury was higher for interferons, alemtuzumab, teriflunomide, and fingolimod. The risk of PML was higher with fingolimod and dimethyl fumarate. The risk of infection was lower with interferon beta and glatiramer acetate. And lastly, based on the evidence reviewed, uncertainty remains regarding the association of cancer risk and DMTs. Multiple cohort studies reported association among various DMTs and an increased risk of cancer. However, only one identified study reported a statistically significant association. In this study, interferon beta, dimethyl fumarate, and fingolimod were significantly associated with cancer reporting. Key question 5, what are the characteristics of ongoing studies of DMTs for MS and CIS? We identified a total of 12 ongoing studies. And these ongoing studies have the potential to fill important evidence gaps, particularly where we did not identify published comparative RCT evidence for a DMT. And this is specifically the case for cladribine, diroximel fumarate, monomethyl fumarate, PEG interferon beta 1A, and siponimod. However, it is important to note that all the information about these ongoing studies is based on posted eligibility criteria and not on published results. So these findings represent our best estimation for current ongoing trial information of interest to DERP. 6 ongoing studies are comparative RCTs. Notably, every single one of the DMTs eligible for inclusion in this DERP report have ongoing comparative RCT trials. Across these trials, there is a trend towards comparing against groups of DMTs rather than one DMT against another DMT. For example, 2 of these trials are investigating ofatumumab versus first-line DMTs and other approved DMTs, categories which are published in the registry and defined by trial investigators. 2 additional trials have grouped the DMTs as early aggressive therapy versus traditional therapy, and early highly effective therapies versus escalation therapies. And these categories are once again defined by the trial investigators and are not necessarily reflective of the evidence we identified. Lastly, we identified 2 trials comparing several eligible DMTs of interest to DERP to a stem cell treatment that is not currently FDA approved. These trials were included given the possibility of subgroup analyses comparing DMTs of interest for this report. This table contains additional information for the 6 comparative RCTs that were discussed on the previous slide. So I'll walk through this briefly. In terms of study populations, one trial includes individuals with relapsing MS, and the rest include individuals with relapsing remitting MS. Estimated enrollment numbers range from 100 to 900 participants, and estimated completion dates range from March 2024 to April 2030. Finally, there was also an ongoing comparative RCT that was identified in the previous 2020 report that has been completed but has yet to publish results. This trial was completed almost 3.5 years ago at this point and was investigating PEG interferon beta 1A compared with interferon beta 1A or 1B and had a study population of 80 participants. The study is still of interest given that no comparative RCT evidence on PEG interferon beta 1A was identified for this report. We also identified 2 ongoing placebo-controlled RCTs that include adults with primary progressive MS and relapsing MS. Both trials are comparing ocrelizumab against a placebo. One of them has the potential to investigate a population with CIS, which would meet eligibility criteria for this report. The other trial is looking at a population with primary progressive MS, which is of potential interest as ocrelizumab is the only DMT currently FDA approved to treat individuals with PPMS. So there's a gap in terms of head-to-head RCT evidence currently available for this population. Lastly, we identified 3 ongoing prospective cohort studies in which we emphasize comparative evidence. As with the comparative RCTs, these cohort studies hold potential to provide evidence on all our DMTs of interest. However, we will not be able to know this with certainty until results are published. One trial that included any DMT was completed in September 2023 with 1,250 total participants who either had relapsing remitting MS or relapsing secondary progressive MS. Trial results had yet to be published at the time of our search. The other 2 trials, both of which are evaluating diroximel fumarate, are only including pregnant women with MS. This table contains additional information for the 2 ongoing placebo-controlled RCTs and 3 ongoing prospective cohort studies. For the 2 placebo-controlled RCTs, estimated enrollment numbers are 175 and 1,000, and estimated completion dates are December 2027 and August 2028. We touched on the details of the completed prospective cohort study on the prior slide. So of the 2 prospective cohort studies that are not yet completed, estimated enrollment numbers are 908 and 1,178, and estimated completion dates are January 2031 and July 2032. And we'll conclude this presentation with a brief discussion and a summary of our findings. In total, we identified 38 RCTs, 6 of which are newly identified for this update. 15 of these are head-to-head comparisons in people with MS, 4 of which are newly identified for this update. And these were evaluated in 29 RCTs, 6 of which are newly identified for this update. We did not identify any new evidence for this update in terms of RCT evidence on comparisons of different dosing schedules for people with MS, comparison of different dosing schedules for people with CIS, and placebo-controlled comparisons for people with CIS. All of the findings on this information remains unchanged from the 2020 report and entails 4 comparisons of different dosing schedules for people with MS evaluated in 9 RCTs, 1 comparison of different dosing schedules for people with CIS evaluated in 1 RCT, and 5 placebo-controlled comparisons for people with CIS evaluated in 8 RCTs. Furthermore, we also identified 45 cohort studies, 15 of which are newly identified for this update. As a reminder, there are 17 total FDA-approved DMTs included in this report. 13 of them received FDA approval prior to the publication of the 2020 DERP report on this topic. And 4 DMTs received FDA approval since the prior DERP report on this topic was finalized in 2020. These approvals were made between April 2020 to December 2022. All 4 of these DMTs, which are ublituximab, ponesimod, ofatumumab, and monomethyl fumarate, are approved for adults with relapsing forms of MS. These DMTs cover a variety of different routes of administration as well as administration frequencies. Notably, 2 ongoing comparative RCTs projected to complete by February 2026 are specifically interested in ofatumumab. And 3 of these new DMTs, ublituximab, ponesimod, and ofatumumab, were evaluated against teriflunomide in 5 of our 6 newly identified comparative RCTs. We did not identify any RCT evidence in this report for monomethyl fumarate. Monomethyl fumarate was FDA approved based on bioequivalence with dimethyl fumarate, and notably it's the sole active metabolite detectable in the plasma of dimethyl fumarate. And we'll briefly summarize findings for the 29 RCTs, again, 6 of which are newly identified for this update, comparing DMTs for people with MS. As mentioned previously, we only found comparative RCT evidence for 3 of the 4 newly approved DMTs, ofatumumab, ponesimod, and ublituximab against teriflunomide. When compared against teriflunomide, the newer DMTs all significantly reduced relapses. And compared with teriflunomide, ublituximab was associated with increased serious adverse events. However, ofatumumab and ponesimod were not. In a newly identified comparison in which fingolimod was compared with glatiramer acetate, fingolimod significantly reduced relapses. And when comparing the older DMTs directly, alemtuzumab, fingolimod, ocrelizumab, and teriflunomide significantly reduce relapses and are not associated with increased serious adverse events compared with other DMTs. This finding remains unchanged from the 2020 report. Considering that same set of 29 RCTs, 6 of which are newly identified for this update, it is important to note that we did not identify head-to-head trials for every possible comparison of relevant interventions, so we're not able to state conclusively that other therapies are any more or less effective overall. We did, however, identify a recent and relevant network meta-analysis drawing comparisons that is summarized in the full report. Furthermore, we did not identify any eligible RCTs for 5 FDA-approved DMTs, and these are diroximel fumarate, cladribine, monomethyl fumarate, siponimod, and PEG interferon beta-1A. Lastly, subgroup analyses are not consistently reported across studies, so we are limited in our ability to draw robust conclusions about these. From what was identified, the presence of neutralizing antibodies does not appear to reduce effectiveness, and patient factors such as age and prior treatment may change effectiveness. Next, we'll briefly summarize findings for the 5 placebo-controlled comparisons in 8 RCTs, none of which are newly identified for this update for people with CIS. Findings that again have remained unchanged from the 2020 report include that the following DMTs significantly reduce conversion to MS in individuals with CIS in comparison with placebo: cladribine, glatiramer acetate, interferon beta-1b, interferon beta-1a, and teriflunomide. Additionally, when compared against placebo, the same group of DMTs did not appear to be associated with serious adverse events. As with the therapies for people with MS, subgroup analyses are not reported consistently across the studies, but there is some evidence that women may benefit more than men from glatiramer acetate and interferon beta-1a. As a reminder, further details are included in the report itself and in the 2020 slides with findings from individual studies, which are included at the end of this slide deck. In total, we identified 45 cohort studies, 15 of which are newly identified for this update. Evidence from the reviewed cohort studies indicates that treatment discontinuations and switches appear to be significantly lower with fingolimod and dimethyl fumarate, and uncertainty remains regarding the risk of treatment discontinuation or switch for oral DMTs compared with injectable DMTs. From this same group of cohort studies, we identified evidence on adverse events. It is important, again, to note that DMTs do have adverse events and differ in their safety profile. Specifically, the risk of liver injury was higher for interferons, alemtuzumab, teriflunomide, and fingolimod. The risk of PML was higher with fingolimod and dimethyl fumarate. The risk of infection was lower with interferon beta and glatiramer acetate, and uncertainty remains regarding the association of cancer risk and DMTs. Overall, evidence is not consistent in terms of which DMTs are compared, which does limit our ability to draw conclusions. Thank you.
Thank you, Rachel and Courtney. That was a very comprehensive review. Committee members, any questions for Courtney or Rachel? Okay, and I don't see any stakeholders that were signed up ahead of the meeting. Any online currently? Okay, I don't see that either. So with that, Nanya, if you can please pull
This
up.
is Lita. There is one hand raised, or
There
two
is.
hands raised now. One hand raised. Hang on just a second. We have Lisa Carmen.
Lisa, you can unmute yourself now.
Okay, can you hear me?
Yes. And let me put the questionnaire on the screen. Just give me a quick second. Let me share my screen. Oh, is it coming along? Can you see? Okay. Looks like he was thinking about that. And let me pull up.
All right, Lisa, if you can please go ahead and answer the questions on here, and then you're ready, Lita will start the timer.
Sure. Hi, I'm Lisa Carmen. I'm representing on behalf of Genentech, and we make ocrelizumab or Ocrevus. I am not a provider or a patient, and the only conflicts of interest is as an employee of Genentech.
All right, go ahead.
Okay, I wanted to talk briefly around kind of the key question number 5 from the DERP review, which really is the open label extension data from our OPERA 1 and 2, our RCTs. We have been tracking outcomes for these patients. We're now out 10 years and have been doing regular updates through the ECTRIMS, focusing on not just relapses, but really impact to disability. So I just wanted to highlight some of those data that has read out. We are tracking 52% of patients who have completed the original RCT that completed out this far in the open label extension. After 10 years, patients who've been continuously treated with Ocrevus, the annualized relapse rate is 0.017, which is equivalent to a relapse almost every 60 years. In addition, after 10 years, almost 8 out of 10 continuous treated Ocrevus patients were progression-free. I additionally wanted to point out, because in this trial, it was a head-to-head against Rebif at the time, and there is a significant difference in disability accumulation. So originally in the trial, for 2 years, they were treated on Rebif or Ocrevus, and later the Rebif arm was allowed to cross over and then continue in the open label receiving Ocrevus. And starting Ocrevus 2 years earlier, so the patients who were not on Rebif, saves 10 years of disease progression. In another way to say it, the years between progression events between the Rebif trial who crossed over to Ocrevus and Ocrevus is 9.5 years. In addition, after 10 years, over 90% did not require a walking aid. I have additional wheelchair data as well. But back to the primary endpoints, after 10 years led to an almost complete suppression of MRI activity, and these benefits were seen in patients once they switched from Rebif to Ocrevus in that arm. I just also wanted to mention, that's kind of the highlights. I have some from PPMS open label as well, but I don't have time to go into those today. A few other additional points. Ocrevus sub-Q we're hoping to hear from the FDA in September. Thought that might be important for you guys to know. In addition, in regards to special populations, we do have some employment data looking at as a continuation of a trial. We have pregnancy data, we have lactation data, and we have newborns that we've tracked. And last, I'll leave you with our 2-year RWE, where we look at events associated with relapse, use, and costs, and looking at first-line versus second-line Ocrevus at all medical costs in patients who were on the first-line Ocrevus were less, and that was attributed to lower inpatient stays on Ocrevus compared to other medications. So thank you.
Thank you, Lisa. Any questions for Lisa from the committee members? Okay. Any other stakeholders signed in? Gonna lean on Nanya to let me know.
No, I don't see any more.
Okay. Okay, then with that, we can move on to the motion.
Can you guys see the motion?
Yes, we can. Thank you, Nanya.
So I've already copied and pasted previous motions, and I've updated it with the list of drugs that were brought up, that were presented today.
Great, thank you so much. Okay, comments from the committee? think one of my main points was the oral agent, so I'm glad to see that that's already called out. Any language that's up for debate? Otherwise, we can move forward with proposing the motion.
This is Michael Corsillis. I think it looks good for me. I'd like to make a motion and pronounce all these names. After considering the evidence of safety, efficacy, and special populations for the treatment of multiple sclerosis, I move that alemtuzumab, cladribine, dimethylfumarate, diroximel fumarate, engolimod, glatimer, acetate, interferon beta 1A, 1A-SC, interferon beta 1B-SC, monomethyl fumarate, peginterferon beta 1A-SC, ocrelizumab, ofatumumab, ozanimod, ponesimod, siponimod, teriflunomide, and obinutuzumab are safe and efficacious. Product that is safe for use during pregnancy should be made available. The MS drugs cannot be subject to therapeutic interchange in the Washington preferred drug list for the treatment of multiple sclerosis. An oral agent should be included in the list of preferred drugs on the PDL.
This is Greg Hudson. I second.
Thank you. All in favor, please say aye.
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 All right, any opposed? And the motion carries. Michael, great job with those drug names. All right, by our schedule, we are scheduled for lunch next. Does that sound good to everybody? Yes?
Yes. Yes, it does.
All right, so 12:07, should we say we'll be back by 12:40 for after lunch?
Yes, that works. Does that work for everyone?
All right, wonderful. Okay. We'll see you in a bit. All right, I think we have quorum. All right, so now, I guess I didn't officially say P&T committee adjourns after the last session and the DUR board now convenes. And the first item on the agenda is with Marissa, who will be taking us through the Apple Health policy for movement disorder agents. Marissa, we have you online.
This is Marissa. Yes, I am ready. So let me go ahead and just tee up my policy, and then we should be able to get started. Sorry, I'm trying to do some reshuffling of some screens here. Okay, I have what I need. Let's share. Okay. Okay, you should be able to see the policy that's displayed.
Yes.
So, okay, perfect. So good afternoon, DUR board. My name is Marissa. I will be going through the movement disorders agents. Particularly, this is a policy around valbenazine or Ingrezza. This is a brand new policy that we are creating, so it's not something that we currently have implemented. It would be implemented if approved after this meeting, a couple months after this meeting. I will go over some very high-level background information, just kind of on the disease state, and then a little bit about the drugs or drug types themselves, just because I don't believe we're going through any kind of clinical review on them in the review of the classes. So just to kind of give you some background, Tardive dyskinesias, or TDs, are involuntary movements of the tongue, lips, face, trunk, and extremities that occur in patients treated with long-term dopaminergic antagonist medications. They are most common, TDs are most common in patients with schizophrenia, schizoaffective disorder, or bipolar disorder who have been treated with an antipsychotic medication for long periods. However, tardive dyskinesia can occur in other patients as well. So people that have fetal alcohol syndrome, other developmental disabilities, and other brain disorders make these particular patients vulnerable to TD. Huntington's disease, on the other hand, is a hereditary disorder that causes nerve cells in the brain to gradually break down. So chorea, or uncontrollable movements, is the defining symptom of Huntington's disease. Huntington's disease chorea is progressive and can affect finger and hand coordination, walking, mouth movements, and swallowing. There are the current treatments available to treat tardive dyskinesia and chorea associated with Huntington's disease are vesicular monoamine transporter 2, or VMAT inhibitors, which are the products that I will be going through in the policy. Those are the products in question today. How VMAT2 inhibitors work is they deplete the transport of dopamine, thus decreasing the release of dopamine and its breakdown by monoamine oxidase. So very high level overview on kind of the drugs and the disease state. So I will just go ahead and jump right in to the clinical criteria. So the first criteria that we have is, well, let me go through it box by box. So the first one is really our blanket statements that we have at the top of our policies, just saying that this policy applies, this drug may be considered medically necessary in patients who meet the criteria below, which I'll get into shortly. And then we have at the top here our blanket statement of really, if all the criteria are not met, but the clinical reviewer determines that it's a medically necessary medication for this patient, it can be approved on a case-by-case basis. And then also the clinical reviewer can choose to use the reauthorization criteria if a patient has been previously established on therapy or is a new client to Apple Health. So that just kind of encapsulates that. For the first indication, I will be going through the chorea associated with Huntington's disease. And actually, let me just give a little background. In this class currently, and it's not on your PDL publication that you were given, but these, the drugs that are in this class are Austedo, Austedo XR, and Ingrezza. And currently they live in what is called our movement disorders Apple Health PDL drug class. And currently, the two preferred products that we have in that class, well, three actually. The three preferred products are, we have tetrabenazine, which is generic, we have brand name Austedo, and then brand name Austedo XR. So all three of those products are preferred without PA on our AHPDL. So really this policy that I'll be presenting today applies to this non-preferred Ingrezza, and patients that are requesting this medication would have to meet all of this criteria in order to get it, with the intention of really having to step through the preferred Austedo XR, or generic tetrabenazine. So that's just kind of some background on what is going on in that drug class and what that looks like. So for this first indication, the clinical criteria is just as follows. We would require the patient to be 18 years of age and older, and that's just really following labeling for these products, for this product specifically. It's prescribed by or in consultation with a neurologist or psychiatrist. The third, the patient should have a diagnosis of Huntington's disease. 4, we would require at least some type of baseline assessment. And what we found was it would be probably using the Unified Huntington's Disease Rating Scale, or UHDRS. And this particular assessment is, from what I have gathered, it's a very big assessment and it's broken up into parts. So whichever part, I guess, either of these, whether that be motor, cognitive, behavioral, functional independence, or total functional capacity, if any of those particular assessments were submitted, then we would accept them. But you know, I understand that's a big test, but even if they submit parts of it, that's really what that is really getting to. For 5, the medication will not be used in combination with another VMAT inhibitor or monoamine oxidase inhibitor, and that's just per labeling as like a contraindication, particularly the MAOIs. And 6, this is what I was alluding to as far as stepping through our preferred product. So treatment with deutetrabenazine or deutetrabenazine ER, which is Austedo or Austedo XR, is contraindicated or has been ineffective or not tolerated for a minimum of 12 weeks. So if they meet all that criteria, we'll authorize it for 12 months or 1 year. And then for the reauthorization criteria, it would just be for number 1, they would just need to meet criteria number 5. So really just stating that the patient won't be using this in combination with another VMAT2 or MAOI. And then 2, documentation is submitted demonstrating disease stability or positive clinical response. So some examples of that would be a reduction in involuntary movements, a decrease in total maximal chorea score. And that is the decrease in total maximal chorea score is actually measured in these functional assessments or part of the UHDRS scale. If they meet both of those reauthorization criteria, we'll authorize it for another year. Getting into the tardive dyskinesia indication, it's pretty similar to above with a little bit of changes. So the first 2 are relatively the same as above. Age indication 18 or older, prescribed in consultation with a neurologist or psychiatrist. 3, they would need a diagnosis of tardive dyskinesia. 4, this is where it's a little bit different just for the baseline assessments. So we would want baseline assessments using one of the following tests. So an AIMS score, so the abnormal involuntary movement scale, or clinical global impression of change, tardive dyskinesia. So at least one of those would need to be submitted. Same as above, it's not used in combination with a VMAT or MAOI. And then 6, at least one of the following treatment approaches was ineffective unless all are contraindicated, not tolerated, or put psychiatric stability at risk. So if the provider switched from a 1st generation to a 2nd generation antipsychotic and that wasn't effective, or B, the patient has a history of discontinuation or dose modification of the offending medication. So they've tried to either discontinue the medication or tried to kind of change the dose and it wasn't effective. So either of those 2. And then 7, kind of same as above, just stepping through our preferred agents. So they've tried Austedo or Austedo XR and it wasn't effective or it's contraindicated, and the trial was at least for a minimum of 12 weeks. So if they meet all the criteria, it'll be authorized for 1 year. And then for the reauthorization criteria is pretty much the same as the other one. We would just require that they are not using it in combination with a VMAT or MAOI, which is number 5 here. And then we would just need documentation that there is disease stability or positive clinical response. So things we would be looking for would be reduction in involuntary movements, improvement in the AIMS score, and improvement in the CGI-TD score. If they meet all of those criteria, the request will be authorized for another year. And then here, these are just the dosage and quantity limits for those. And then it's broken up based off of the indication. We'll go ahead and stop there for a little bit, let you look over that. And then scrolling down a little bit more to the bottom is just the background and the references. And then I'll go ahead and get into the form that supports this. So this is the prior authorization form that helps guide the provider if they're requesting this medication or putting in the PA request. So let me actually zoom out a little bit. This up here at the top is just basic demographic information, patient information, prescriber information, medication information, and direction. Here are the kind of checklist items that we would be looking for. So I'll go ahead and pause. Let me see if there's another page. Okay. I'll pause here, and then I can take any questions from the board.
Hi, Marcia, this is Ryan talking. When I'm looking at the screen, I don't know if other people see it, but it looks like the policy is still up.
Yeah, I need to switch it. Hold on one second. Thanks, Ryan. Let me do my screen. Okay. Can you see the form now?
Yes, we can.
Okay, perfect. Sorry about that.
Right.
Marissa, this is John McKay. I had a quick question for you. So in terms of tardive dyskinesia and treatment failure, moving from a first generation to second generation antipsychotic, what about in terms of failure of two second generation antipsychotics versus going from a first generation to a second generation? Is there any need for maybe just a failure of one second generation to another second generation?
The way that the clinical policy is written right now is that it would just, sorry, I apologize for the scrolling. We would just require, the way that I wrote it was just one second generation antipsychotic. I didn't put switching from like 1 second to another second. Yeah, so I could consider that. If you feel very strongly about adding that type of language in, then I can definitely do that.
I'm just wondering from like a treatment perspective, if they've never tried a first generation and they're on a second generation, but stable otherwise, moving forward.
Yeah, I see what you're saying.
Mm.
I think that's a good call out though. So I could, yeah, consider taking that back.
Yeah, I'd just second John's statement there to simplify that.
Yeah, the term switching, I think, is just confusing. Like, what are you needing to prove that they failed? Is it the first? Is it the second? Is it both? So I think maybe you just want to say, because it says at least one of the following treatment approaches was ineffective. Maybe A is just like 1 second generation antipsychotic is actually what you're getting at.
This is Marissa. So the way that it's written is the intent would be that they would have tried a first generation antipsychotic and then switch to a second one. But I didn't think about it in the way that they could have not taken the first and started with the second generation and then switched to like another second generation. I think, John, is what you were getting to. So it's not written in the way that I believe I'm understanding John is saying. So I could incorporate some type of language into the policy because that's not what is taken into account. So it's not necessarily that they are failing. I'm trying to think of the best way to say this.
Looks like Dawn also has a comment.
Yeah,
Oh.
Marissa, I think if you think of it from a standpoint where they've tried two different second generation antipsychotics and they're still experiencing movement disorder symptoms.
Yeah.
So they're not really failing a second generation antipsychotic. It's just they've tried two and they're still experiencing abnormal movements.
Yeah, and that would kind of be consistent with the language we've had in other policies, which is trial of two agents.
This is Laura Bestie. Would you want to state at least one of the following treatment options caused movement disorders as an adverse effect? So, because like you have was ineffective. Well, it's not really the question whether it's ineffective, it's whether it caused the movement disorders an adverse effect.
Could you just state like continue to experience tardive dyskinesia despite trialing two different?
Yeah, there you go.
Because that's kind of what we're getting at, right?
Exactly.
Yeah, I think that's accurate.
This is Marissa. Sorry. Can you point me to where you would like me to change it and then I can start typing?
Go ahead, Kevin.
I think you could just, because the whole point is not switching, so it just so instead of you just say continue to experience tardive dyskinesia
Or persistent tardive dyskinesia despite trial
of two second generation.
Making it be 2 is stricter than what she had originally put, though. Like, she only put that it had to be 1.
I think the original language was indicating that they've done at least a first generation and a second generation.
But isn't this also
It's that
referred
the
to
first generation like caused it though, and then switching to a second generation didn't fix the problem, right?
This is Donna. You could have abnormal movements on a second generation antipsychotic. Most people aren't started on a first generation anymore, or very seldom. So they're more likely to have started on a second generation, experienced a movement disorder, and then were switched to a different second generation to see if that would resolve the issue. So I think just to, having, they tried 2 second generation antipsychotics and they still are experiencing, abnormal movements.
Go ahead, Peter.
It might be helpful to split this criteria into 2 separate elements. One, an exclusion criteria, because the first generation antipsychotics are more likely to cause this. We would want to say, if you're still taking a first generation antipsychotic, then that would exclude you from getting this therapy because you should have switched. But then just say that you've tried changing at least once, from say 1 second generation to a second pill and a second generation antipsychotic. So it might make sense to just try to do this in 2 criteria elements.
So would that be as she has listed? So she left A as is, which is first gen to second gen, and then this B.
I would make it more generic than that and just say patient has tried changing their antipsychotic, and then I would have another point somewhere below this that would be an exclusion. Continued use of a first generation antipsychotic medication is an exclusion to this use.
Rather than making it more wordy though, could you just reword the whole question or the whole number 6 as far as patient continues to experience tardive dyskinesia after the following approaches: A, switch from first generation to second generation; B, after a trial of 2. So then you're just combining.
That's smart.
The top piece to make it less verbiage. So patient continues to experience persistent tardive dyskinesia after the following, what's the word? Approaches. There you go.
At least one of the following.
Yeah, after the following treatment approaches. And then unless all are contraindicated, not tolerated. That whole sentence.
And just make sure you put at least one in there so that it's not stricter than before.
This is Marissa. Sorry, I'm trying to type and spell at the same time, so I apologize. So then, experience after. So this would be, it would be switching. I think, Laura, what I understand, it would still be like these. It's just I'm like rewording it, switching.
Correct. And then B would just be trial of 2 second generation. Or, I mean, shouldn't A always be the first thing they try though? I don't know how to put that in there. If they're on a first generation, that brings up, I think Peter mentioned it, that it's an exclusion criteria, but that should be always included. So it'd almost be like A plus one of the B and C. I don't know how to verbiage that.
I think if I understood Peter correctly, it's like we just skip on the A because being on a first generation would be an exclusionary criteria separately in the policy. And so we would just start with B. Is that right, Peter? I don't want to speak for you.
Here, now I'm off mute. Yeah, that's what I was thinking.
Yeah, I'm good with that. Do we have a section, Marissa, for exclusion criteria?
No, this is Marissa, sorry. No, we don't have like a section where we include that.
So are you trying to say if they're on a first generation, you would not allow them to be on?
Right. Yeah, because the risk for these movement disorders is so much higher on a first generation, and there's so many choices for second generation medications that if you're still choosing to use a first generation, you, I mean, you shouldn't. It's bad. It's bad clinical medicine, but it would be an exclusion to using one of these meds.
Is there a reason, I don't know, like a first generation would be giving better other symptom control, you know, of like psychoses, delusions, hallucination?
I can't think of one.
I'd
I feel
interject.
like if you have B, you don't need A.
This is Greg Hudson. I mean, I agree with Peter in general that in the vast in general clinical practice, it's not preferred to use a first generation antipsychotic. However, I can think of specific cases where first generations might be, again, these are specific cases, might be preferred or necessary for continued use and continued stabilization of some persistent mental illness. And I wouldn't want to exclude the use of this medication in those populations. So I would not recommend that the exclusionary criteria.
This is John. I would kind of second Greg's concerns there, just in terms of usually we'd see it in like treatment resistant, like schizophrenia, and in the cases of like use of clozaril or some of those other first generation antipsychotics that might be more resistant and not amenable to use of the second generation antipsychotics.
Yeah, I think like this is a case in which this is like a expert, right, who's making this decision. And so I don't think we should like make it so that like a, you know, for if for some reason somebody does have one of these reasons to take this medicine, then they can't get it. I guess like the whole need for 6 when we have 7, I don't understand, because like they're only getting this non-preferred med if they've tried deutetrabenazine anyway. So then in order to get deutetrabenazine, didn't they already have to try all this other stuff? So why are we even making them prove all this stuff in 6 if they can only get this med if they've tried 7 in the first place? Does that make sense? Like, why do you have to re-prove that you did all this stuff in 6 if you already tried 7?
Marissa, is there intent behind
This is Marissa. I mean, it was just to really follow like clinical practice of like what you would expect to see. So like if someone did experience tardive dyskinesia, I mean, clinically you would expect, if they are on an antipsychotic, you probably would expect to see them either switching from something, discontinuing the medication. So it was really just to, you know, go along with the clinical guidelines, but I could see where Dr. Taylor is coming from. But it's just to be inclusive and really be specific, as specific as we could be in the criteria.
Marissa, this is Donna. Do we require, and maybe I missed it, do we require PA for deutetrabenazine that they've tried and failed, or that they've tried 2 different second generation antipsychotics?
This is Marissa. No, we don't have any, that's just preferred without PA, Donna. So I could see where Dr. Taylor is going, because we wouldn't, if there was if we got a claim for it, particularly, like we wouldn't be able to see that info. I mean, you could from claims information, but it's not like we would be looking for the trial and the failure of the antipsychotics in order for them to get the Austedo. So, yeah.
Yeah, then I would be in favor of leaving 6 in, and like even sometimes when there is that other prior auth criteria, it's helpful to put it in, because you can imagine if you get a denial letter and it says, oh, you have to try this other medication, then they try that medication, and then they get another denial letter says you have to try these other things first. So it's important in those, because usually those denial letters include the criteria, right, the policy, and laying out all those steps is helpful so that people don't get trapped in a cycle of denial letters, and they kind of work their way back through all of the criteria. So that would be my 2 cents.
It's a really good discussion. I can see different points of view here. I guess why don't we take maybe a vote on that first? Is that so if I may summarize, the question is like, do we leave the motion as is now, like with those changes that Marissa has noted in the comments, but leave 6 in there versus remove 6 altogether because
I don't think I like withdraw that proposal now that I understand that the other one doesn't require PA. I thought the other one also required PA. I'm fine with leaving 6 with our rewrites. We don't need to vote on it.
Awesome. Great.
I still don't understand the C part though. Which offending medication are you talking about?
It could be whichever. So you would probably be looking for like an antipsychotic, even though that's addressed up here, but I guess whichever other medications could cause tardive dyskinesia besides the antipsychotics too would fall under that umbrella. Or if they've had issues with dosing the antipsychotic, that it would fall under that as well.
Okay.
Yep.
So basically, if you're a patient who's on Haldol and you have a history of discontinuing Haldol, then you're good on 6. You don't need to try a second generation, and you don't need to try 2 second generations because it's just one of the 3 that you need. That's why I'm not sure if that's really what or I'm reading it wrong.
Did we just say and at this Laura Bestie under A, just say this and one of those 2 following, B and C? Or is that like getting too confusing? Instead
I
of
mean,
4.
C is included in A, right? It's like switching from the first gen is the same as discontinuing the first gen.
Well, but this just says you're discontinuing it. It doesn't say you're switching to a second generation under C.
Yeah, true. I guess you need to have you have persistent tardive dyskinesia after stopping the offending agent. Which is I guess what C is getting at.
This is like, yeah, yep, that's what that's getting at. Yep.
Is Kavita here? So is C trying to be a catch-all for things not covered in A and B, like offending agents that are not covered in A and B?
This is Marissa. Yeah, it can be, but really, I think the essence of how it is like if a patient was on an antipsychotic, experiencing tardive dyskinesia, stopped the antipsychotic, but even after stopping, they were still experiencing the tardive dyskinesia. That was how I wrote it with that intent. Yeah.
Okay, it's probably fine. I think it's a little bit hard to parse, but I think it's gonna probably be no matter how we word it. So
This is Christy Weiland. Marissa, I just appreciate that last phrase you said, because if we added at the end offending agent with continued symptoms, that might provide some clarity, because I don't know that I fully understood that until you added that.
This is Marissa. Yeah, I can add that. Let me see. You said with continued symptoms. Did I read that? Did I hear that right?
Yeah.
Correct.
Okay.
Okay, so we've focused nicely on points 6 and 7. Do we want to look at the remaining policy? I guess I'll let you finish there, Marissa, sorry.
Do we need to update the actual, what you've written there, the continues to experience persistent tardive dyskinesia? Because that's not in the actual text.
This is Marissa. So what I have in the note here is what I would be switching number 6 to completely. If I captured that correctly, I just didn't want to do it like all at once and then lose kind of some of the discussion. But the intent is what's in here is exactly what would be translated over to the policy for the new version.
Sure.
So let me know if I missed anything. I think I got the persistent tardive dyskinesia part, Kevin.
Perfect. Thank you.
Okay. Let me know if I missed anything, please.
No, I think you got it, Marissa. You did a great job there. Okay, so maybe looking at, was there any other part of the policy? Should we scroll down further if there's Anything else in there? Criteria. Okay.
This is Marissa, and I can also go back to the prior authorization form as well if you all didn't get a chance to look at that either. I know we might have skimmed over that pretty quickly.
This is Laura. Can we go back to the prior authorization form so we can make sure that the decision we just made on the verbiage is copied over?
I'm trying to get, this is Marissa, I'm trying to get 7 in there. It's a little hard.
Okay, so yeah, so 7 will be reworded a little bit.
It doesn't like when I do that. So apologies that if you were looking, it scrolled down.
I think this is minor, Kavita, here, but would 7 come before 6 in terms of just chronologically? Is that the series of events the physician would go through?
Oh, this is Marissa. Sorry. For this, I was just referring back to the criteria in the policy. I believe it was number 6. That's why I put that there. Yeah. But it does look like the way that this form is written, this is specifically for the tardive dyskinesia, and it doesn't look like we have, yeah, it looks like that's what's specifying. It's a little different. I apologize. My dog is so
Okay.
angry.
And
okay, I see that.
And this is Greg Hudson. And Marissa, if I can ask a question, I just want to make sure I clarify. So for the form with deutetrabenazine or Austedo, is question 7 a part of that requirement for this to be covered as well? I guess I'm wondering if our criteria for covering both of these medications, if they're the same, or if we're adding sort of extra criteria for Ingrezza that's not reflected in Austedo.
This is Marissa. So currently how it would work is Austedo really doesn't have any clinical criteria that like a provider would need to fill out, because right now the way it's on our PDL, it's preferred without any PA. So if you were, if a pharmacy were to fill it, it would hit with no PA required. For this policy really only applies to Ingrezza. If I could just be, I guess that's really the underlying theme. Yeah. So these criteria that you're looking for are really only for Ingrezza.
Thank you.
Yeah. Mm-hmm.
All right, board. Other comments on the policy or the form?
I know some forms like this have like, if yes, go to question blank, if no, go to question blank. And I just want to make sure it's clear that you only have to answer question 7 if the patient has tardive dyskinesia. If maybe there's a way to do something like that or reorder them so that like if you could say like Korea answer questions 3 to 6, tardive dyskinesia answer question 3 to 7. It's not elegant, but just something, because I wonder if it's clear enough that you only have to answer 7. Maybe you could just put it in the body of the question itself, like instead of just at the top, like for patients with, or only answer if, I don't know. I filled out a lot of these for imaging, and sometimes you feel like you're in like a video game where you don't know which way to go next.
This is Laura Vesti. Can you just say if tardive dyskinesia please check boxes for diagnosis, or so then it's just in that box on the very end before question 7.
This is Marissa. So we have just this statement
Yeah.
right here. We haven't traditionally done it on these forms, I believe, where it's like, oh, if you don't meet, if it's not for this diagnosis, skip to question
Oh, okay.
or what have you. I think that might be a good takeaway though for us as far as the form. So I can take that back and see if maybe we can make it in a way where, like, for these particular diagnoses, you would fill out questions like 3, 5, and 7, for example. I totally made that up.
Yeah.
Yeah, but I can take that back to our team and see if we can make some types of updates for that, just so that then it's a little bit clearer as far as like which questions you actually have to answer. Because I can see the providers getting confused as far as like which questions they do answer, which ones they don't answer, or you get, you know, as a reviewer myself, you do get questions that get answered that are completely not relevant to what you're even looking at, you know, because it's just confusing. So I think that's a great call out. I'll go ahead and add that note and we'll take that back.
Kavita here. I think along the lines of what Laura was saying to support Zoe's comment was just for question 7 or for point 7, the question could just begin with if
Yeah.
patient has tardive dyskinesia. So that way it's very clear that you don't need to answer the question if they don't have tardive
Yeah,
dyskinesia.
I agree. I think that would be simple enough for this example.
But
Also,
you have,
oh, sorry.
there's the Huntington's disease section too. So maybe you just have a box that's not applicable that they can check.
Okay. And then I guess just like as a person who like in our system, like somebody would put this form in my box, right? And I wouldn't necessarily have read the policy. I mean, I guess if I was a neurologist, I probably would have, but just like big picture, it's a little bit frustrating when you don't know which is the right answer to get the drug approved. Because if you did know which one was the right answer, then you wouldn't even fill out the form if you were going to put in the wrong answer. You'd be like, oh, whoops, we need to try this instead. So just the reality being that the provider won't always get the policy and the form both. I wonder if you could either link to the policy or have some things on the, I mean, I think it would be a lot more work to put some things on the form, like if no, like this might get rejected, but on a lot of the like imaging ones, they do put stuff like that and it's really helpful.
I think
At
you
least
have a
linking
good
to the
point.
policy.
Yeah. Is that on the top of the form, Marissa? I feel like we made this adjustment early on that there would be a reference website for the
It
referring
says the
to the
preferred
policy.
drug list. I don't know if that's the same site or not.
This is
So,
Marissa. Yeah, it just
this
links
is
the PDL. Go ahead, Donna.
this is Donna. I understand what you're saying, but the policies are posted online. We can try to get there. You need to answer the question truthfully, not just mark the box that's gonna.
I'm not saying that it would be
yeah.
truthful. I'm saying I wouldn't bother submitting it in the
Okay.
first place. That's the thing that wastes people's time, is submitting things that are going to get denied, right? So it's helpful to have a clue, like this is probably going to get denied if you haven't tried this yet. So that's the only.
Maybe Marissa, and we can go back with the ops staff and reword some of the questions to say, you know, must have tried 2 of these, you know, what have you tried?
I think the 30,000 foot view question though that Zoe I think is proposing is, with each of these policies or each of these forms rather, is there a way to include which policy would be linked to it?
When this is Donna, if it's specific to a policy, yes, we could include the policy number, I think, in the title of the form.
Okay.
But
That
it
would be
doesn't get
helpful.
you to the website. We could probably put, Marissa, we need to add a link to the actual policy page. And that way, you know, they can at least copy and paste.
That would be super helpful.
Yeah, and get to the policy page. You'll have to then navigate to the individual policy itself. I think it'll be too difficult to try to give you a specific link directly to the policy.
I think that's reasonable.
Yeah, I think that's reasonable too.
That's a lot of work for us to do, so it won't happen overnight, just to give you some
I
expectation
understand.
management.
No, thank you for taking that feedback. Go ahead, Peter.
I was just going to say, Zoe, I think kind of what you're getting at on the one end of the extreme would be essentially an attestation to the criteria versus providing a bunch of clinical information and then having somebody review it. And with the other health plan that I said in these kinds of committees for, we've looked at that, and it's really difficult to do an attestation because people are so busy that they end up kind of skimming through it or not reading it too closely and just sign their name or click
Yeah,
click a
no,
box.
I agree with that. Yeah, I don't think an attestation makes sense. I just think like a clue that's like, if you clicked no here, like stop. This isn't going to get approved, which a lot of the like HCA imaging, ones have that kind of thing on them, and it's helpful.
Having the link to the policy is nice, but we've actually built that into the EMR for the other health plan that I work at, and nobody clicks on it. I mean, like very few people actually look at the criteria. It's like, I agree, it's really nice to have, and I think we should do it. It's just it doesn't get as much traffic as you would like.
Kavita here. I think it gives the provider at minimum some sense of autonomy that, okay, I have control over this information, whether I choose to access it or not before filling out the form. But okay, great points. Other comments? It's a great discussion. I feel like I'm having deja vu about this policy number thing, though. I feel like we had this discussion like a couple of years ago as well, that the policy number would be included at the top of the form for reference. So I'm glad that we're discussing this. And if not, then we can move forward to the motion for this policy.
Okay, this is Marissa. The motion should be here. Hopefully you can see it.
Yes, we see it.
Okay.
Yes, so whenever the board's ready, yeah.
This is Peter Barkett. I can make the motion. I move that the Apple Health Medicaid program implement the clinical criteria listed on policy 62.38.00.AA-1 as recommended.
This is Kevin
This
Flynn.
is Laura Bestie. I second the motion.
All in favor?
Flynn got it.
Hi.
Any opposed? And the motion passes. All right, moving on. And I think we are going over to Ryan for the anti-hemophilic agent.
This is Marissa. Sorry, hold on one second. I'm just gonna save all of our wonderful work so I don't lose any of it. We have been getting updates to our windows, and I just want to make sure that nothing freaks out on me and I don't lose any of this, even though this meeting is transcribed. Okay, so let me enter that. Okay, and Ryan, I will get your policy up in just a minute. Let me close some of these windows. Is this one. Okay, Ryan, your policy should be displaying. If it's not, let me know, but you should be good to go.
All right. Hi, this is Ryan Takachuma. Yes, thanks, Marissa. So I'll be going over the Hemgenics clinical policy, a little background before we get started. Congenital factor IX deficiency, also known as hemophilia B, is an X-linked disorder that predominantly impacts males and manifests as bleeding from impaired hemostasis and bleeding-related complications. Hemophilia B is categorized by disease severity, which is determined by the amount of clotting factor in the blood. Severe disease is defined as factor IX levels less than 1 international unit per deciliter or less than 1% of normal. Moderate disease is defined by factor IX level of 1 to 5 international units per deciliter or 1 to 5% of normal. And mild disease is 5 to 40 international units per deciliter or 5 to 40% of normal. The current standard of care for hemophilia B is to replace the deficient coagulation factor either through episodic or on-demand treatment given at the time of bleeding or through continuous prophylaxis to prevent bleeding. People who are on a prophylactic regimen may still require on-demand therapy with factor replacement. And so moving on to kind of the Hemgenix. Hemgenix is a gene therapy for hemophilia B, which aims to kind of eliminate the need for factor IX replacement. Hemgenix is given as a single dose by IV infusion that delivers an adeno associated virus 5 vector containing a copy of the gene that encodes for factor IX. The short-term results are promising and demonstrate a reduction in factor IX replacement. However, the durability of the response of this gene therapy has not been established, and patients may need to have factor IX treatment again. The long-term extension trials for this therapy are still ongoing. And so that covers kind of the background, and we can move on to the criteria for this. So etranacogene dezaparvovec Hemgenix may be approved when all the following criteria are met. Criteria 1 is patient is 18 years of age or older, and criteria 2, it's prescribed by or in consultation with a hematologist or specialist in hemophilia. And criteria 3, the patient has not received a prior gene therapy. That's in reference to another gene therapy for hemophilia B. And criteria 4, there's a diagnosis of moderately severe or severe congenital factor IX deficiency defined by factor IX less than 2 international units per deciliter or less than or equal to 2% of normal, as confirmed by blood coagulation testing. And criteria 5 is either a contraindication, intolerance, or history of failure to continuous routine factor IX prophylaxis with greater than 150 prior exposure days to factor IX therapy. Failure to continuous routine factor IX prophylaxis is defined as one of the following: A, current or historical life-threatening hemorrhage, or B, history of repeated serious spontaneous bleeding episodes. And 6 is documentation that the patient is negative for factor IX inhibitor titers. And criteria 7, the patient has a baseline adeno-associated virus 5 antibody titer of less than 1 to 678 measured by ELISA, and factor IX prophylaxis therapy will be discontinued upon achieving factor IX levels of at least 5% following treatment. And criteria 9, documentation is submitted that includes the client's weight and then liver function tests within the past 3 months. And then once all the criteria met, it'll be authorized for 12 months for 1 dose. For the reauthorization, this is a single dose therapy, so there will be no reauthorization for this treatment. And then I think that covers the clinical criteria before just kind of the dosing limits, and then the background. So I can move to the form that has, the form. So this helps facilitate the prior authorization process. That kind of covers it. So I'll give the committee a few minutes to look over the form, and then I'll open up for questions and discussion. Thank you.
We do have a stakeholder for this topic.
Yes, so I see Jonathan Abdul Haq. Are you online?
Yes, Jonathan is.
Can you
Sorry.
hear me?
I'm just gonna pull up the questions and then the timer. Just give me 1 minute.
Thank you. And I believe we do have a completed COI for Jonathan. So I think if we can have once we have the timer on the screen, Jonathan will have you get started.
All right.
Okay, this is done. It should be ready to go.
Okay. Good afternoon. My name is Jonathan Abdul Haq. I represent CSL Behring in medical affairs. I'm not a provider or patient, and my only conflict of interest is as a CSL Behring employee. So I very much appreciate the opportunity to provide a few comments on the proposed eugenics policy. I just wanted, before I get into those, I have 2 comments, but I just wanted to point out we do have a 3-year data readout from our pivotal trial, which is called HOPE-B, and that did show very good durability in terms of factor levels approaching the lower limit of normal. 38.6 was the mean factor activity at 3 years. So we do have good durability data from our pivotal trial out to 3 years. But my first comment on the proposed policy relates to the criteria requiring a factor 9 activity level of less than or equal to 2%. So this requirement was part of the inclusion criteria for our phase 3 trial, which again was called HOPE-B. And the purpose of this requirement in that trial was to facilitate pharmacokinetic analyses that were conducted as part of that study. A factor 9 activity level of less than or equal to 2% was not intended to define any real world population that could benefit from eugenics. And so including this requirement in coverage policies could potentially limit access for patients living with bleeding phenotypes that the FDA has deemed appropriate for this treatment. It's important to note that our FDA approved label, our FDA approved indication, does not require any specific endogenous factor 9 activity level, but rather focuses on adults with hemophilia B who currently use factor 9 prophylaxis therapy or have current or historical life-threatening hemorrhage or have repeated serious spontaneous bleeding episodes. My second comment relates to the proposed criteria requiring an AAV5 neutralizing antibody titer of less than or equal to 678. The presence of neutralizing antibodies to AAV5 was not an exclusion criteria in our phase 3 trial, and patients with pre-existing neutralizing antibodies were allowed to participate in the trial. Our FDA approved label does not require testing for AAV5 neutralizing antibodies, and there's also no FDA approved companion diagnostic for such antibody testing. In addition, there's currently no guidance on an appropriate exclusionary neutralizing antibody titer threshold at this time. Providers are encouraged to test patients for AAV5 neutralizing antibodies and use their clinical judgment in interpreting results according to currently available data. The Athens Gene Therapy Registry will be collecting data for additional understanding in this area. So those are the comments I had to share on this policy. I thank you very much for the opportunity to speak, and I'd be happy to take any questions you may have.
Thank you for your comments, Jonathan. Do we have any other stakeholders? And any questions from the board for Jonathan? Okay, so with that, we can go back to looking at the policy and the form. So I guess as I was hearing you talk about the policy, Ryan, I was wondering, like, that list of tests that need to be done for the patient to get this treatment, is that are those criteria included in the FDA approval? Because I just think about like the cost of the testing and the accessibility of these very specific tests depending on where the patient is.
Hi, it's Ryan Tuxman, and yes, I'm assuming you're referring to criteria 6 and 7. Is that correct?
Yes, 6, 7. Yeah, I think those are the main ones.
Yep, and this is Ryan Takatomo again. So criteria 6 is in the FDA label,
Okay.
that they're required to have a negative factor 9 inhibitor titer. Otherwise, it's recommended not to administer Hemgenix. With criteria 7, with the baseline adeno-associated virus antibody titer, it's important to note that that was like a companion diagnostic that wasn't validated. And so that kind of what that lab, the purpose was to show that if whether or not a patient has some exposure to like wild type virus, and that they might develop some resistance if they were to receive this therapy, that might make the therapy less effective and potentially impact the safety. In the study, there was one participant that had a really high titer, and the Hemgenix was not effective. It's important to note that Hemgenix is a multi-million-dollar therapy, so we want to make sure that when we're giving it, we're trying to maximize the benefit of this therapy, and to make sure we're not putting those millions of dollars to waste. There is guidance from the World Hemophilia Foundation that recommends that they still get this assay completed, and to help use that as guidance for whether or not they may consider prescribing this therapy. And then I think in the HOPE trial, it was up to 18 months. I think at that specific level, there was no correlation with the antibody to factor 9 activity at 18 months after treatment. So that was kind of a level where it might not have an effect. But again, this assay is not validated. There really is no companion diagnostic available yet, but this is all we have. Hopefully that answers your question. Let me know if there's any follow-up.
That does, yeah. Thank you, Ryan. Yeah, great point about how expensive this is. So we should remove any risk of serious effects or any risk of resistance. I hope that this is an accessible test, which I imagine if we're getting to a point where the hematologist is prescribing this, they should have access to it. Other comments from the board? I see John's hand up. Hey, John.
Hi there. This is John McKay. So this is probably for Donna or Ryan. I don't know if it's under the purview of this committee, but I was just wondering, is this going to be a carve-out medication? If so, can you discuss, is this going to be possibility for an outcomes-based reimbursement, kind of given the excessive cost of the medication and the possibility of treatment failures, kind of like one and done?
This is Donna. It is a carve out, from the managed care plan, so it'll be covered through the fee-for-service program. As far as value-based, outcomes-based agreements, I believe CMMI is creating a cell and gene therapy model around this particular gene therapies is this for sickle cell? No, this is for hemophilia. So no, I'm mixing up my gene therapies. So it is a carve out. It's covered under fee for service. The outcomes-based contracting sounds really great on the the front side. They're very difficult to administer, depending on trying to negotiate the actual outcomes, exclusion inclusion criteria with manufacturers. So I'm not sure if we're going to be entering into an agreement at this point in time, but we continue to look at it as resources and are available.
Thank you.
Peter, go ahead. Oh, Peter, I think you're still muted.
There we go. Hopefully you can hear me now.
Yes.
I just want to make sure I understand the point from the industry representative about the factor 9 levels. It sounded like they were saying that was an inclusion criteria in the phase 2 studies, but not the phase 3 studies. And I just wanted to make sure I'm understanding that correctly, or was that also a requirement for the inclusion requirement for the phase 3 studies?
Can you hear me?
Yes, we can.
It was an inclusion requirement for our phase 3 study, but it was an inclusion requirement tied to specific pharmacokinetic tests. So including it in coverage criteria would not be clinically appropriate because the FDA doesn't require any particular endogenous factor activity for in our indication. And as your colleague pointed out, 2% doesn't define any clinical phenotype or medical necessity at all. It was in there to facilitate specific pharmacokinetic analyses. So it's not appropriate to define medical necessity in a coverage policy. And if I could just say one more clarification, the AAV5 neutralizing antibodies, that whole requirement, so that's not in our label at all. So the FDA does not require any testing, period. And the test is offered by us free of charge to clinicians that want to use it in the shared decision-making process. It's the test that was used in HOPE-B. It's going to be updated, so 678 won't even be a valid titer anymore. And requiring the test is what the World Hemophilia Federation suggests, but a specific cutoff, there's no clinical rationale for a threshold. That's why, we offer it to be used in shared decision-making, and clinicians, can decide. Because we had one patient, all the patients of the 54 patients in our trial, 21 had neutralizing antibodies, 33 did not. All patients, regardless, there wasn't a correlation between factor 9 production and having antibodies or not. So that's one point. The other point is all the patients that had neutralizing antibodies were 678 or below, except for one gentleman who was very high, in excess of 3,000, who never produced his own factor 9. But there's no data points between 678 and 3,000. So there's no threshold that anyone's recommending to determine appropriateness. And again, the FDA doesn't even require testing at all. And the Hemophilia Federation suggests doing the test, but there's no threshold that can, responsibly be applied. And again, 678 won't even be a valid number, given that this test is going to be updated by CSL.
Thank you for your comments, Jonathan. All right. So I think with those comments, with that context, how would the committee like to address the language in this section, if at all?
As Peter, this is a very expensive medication, and we're kind of in uncharted territory. And as we, over time, we'll get more experience with the medication and possibly having a chance to address this again. But I'm comfortable with the way that the language is written. Our industry representative said it's basically not appropriate to determine medical necessity beyond compendia use, the FDA-approved indication, and that's just not the way that the industry works. With these very expensive medications, it's absolutely appropriate to determine some higher threshold than simply its FDA-approved indication. That's the whole point of these policies, and I think the policy has been crafted well and it's thoughtful. And maybe we will revise it in the future, but I think for now this looks pretty good.
Thank you, Peter.
This is Christy Weiland. Peter, I appreciate that comment. I feel like how it is written currently seems to mirror the inclusion criteria of the patient population that's been studied, from what I can understand. So I agree with you.
I think most of us are in consensus. Any with opposing viewpoints or feel like any of this language needs to be modified? Looking at your faces too. I'm seeing head shaking. Okay. All right. So with that, let's look at the form again and then move on to the motion. Thank you so much, Marissa. Okay, so this looks pretty much a yeah, it mirrors the policy well. Comments on this from the board? And if not, then we can move on to the motion.
This is Kevin Flynn, and I move that the Apple Health Medicaid program implement the clinical criteria listed on policy 85.10.25.AA-1 as recommended.
This is Peter Barkat. I'll second the motion.
All in favor, please say aye.
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 Any opposed or abstained? And the motion carries. Great, thank you, board. Great discussion. And I think that takes us back to you, Marissa, for the immune modulators.
This is Marissa. Yes, let me go ahead and we didn't have any comments on the Hemogenics, but I'm just making sure I save everything.
Thank you.
And then I will go ahead and get to the thalidomide. Okay. Okay, so this is Marissa again, and I will be going through the immune modulators, the thalidomide analogs, clinical policy. So just to give you very high level overview, these particular drugs can really treat a plethora of different, they have a plethora of different indications. So I'm not going to get very indication specific. I'll just really get into kind of disease and kind of drug just information on it. So as a high level overview, these immunomodulatory drugs are indicated for several different oncology and non-oncology indications. So lenalidomide is commonly used in the first line setting, while pomalidomide is indicated in the relapse refractory setting. The lenalidomide has generally fallen out of favor given newer second generation immunomodulatory agents. When used for the treatment of MM, the lenalidomide analogs are part of the standard 3-drug backbone based on the superiority over 2-drug regimens. The place in therapy for 4-drug regimens is still evolving and should be limited to clinical trials. The lenalidomide is considered a first-line therapy for MDS, but is generally used in the relapse refractory setting for other indications such as follicular lymphoma, marginal zone lymphoma, and mantle cell lymphoma. So different types of oncology and non-oncology indications. So this is a little bit of a long policy, so just bear with me as far as the clinical criteria. The language at the top of the policy is pretty much the same as the other 2 policies that you've seen, but just to mention again, this policy really encompasses the 3 products that you'll see here. So brand names are Revlimid, Pomalyst, and Thalamid. Getting into indication specific. So this is where I said, hold on, because there's quite a bit. The first one is erythema nodosum leprosum, ENL, I'll just call it that. And that indication really just applies to thalidomide. For this one, it will be approved when all of the following criteria are met. So it's prescribed by or in consultation with an infectious disease specialist. The patient, for 2, the patient has a diagnosis of ENL, And the medication will be used for the acute treatment of the cutaneous manifestations of moderate to severe ENL. And if moderate to severe neuritis is present, the medication will be used in combination with corticosteroids unless it's not tolerated or it's contraindicated. Or the medication will be used as maintenance therapy for prevention and suppression of the cutaneous manifestations of ENL recurrence. And these are really just based off of the label. So that's really where some of these criteria came from. If the patient meets all the criteria, we will authorize it for 6 months. And then for the reauthorization criteria, I will say it's pretty standard across the board for all of these indications. It's pretty blanket statement type of reauthorization criteria. So we would just be looking for a documentation of response to treatment defined by improvement or stabilization of disease or symptoms. So very broad language for that. And for the reauth, if they meet that, if we see that they're having clinical improvement or no progression, then we will reauthorize it for another 6 months. So it's a little bit different as far as the yearly approvals that you'll see, at least in like the other policies that we presented today. Well, except for Hemgenics, that was not reauthorized. But typically we tend to go with either 12 months or 6 months. Special occasions, sometimes we'll do less than that. But because there's just frequent monitoring that we want to make sure gets done, that's why we did the 6 months for these. So getting into the next indication, it's follicular lymphoma, or FL. It applies to the drug lenalidomide or Revlimid. For this, it's kind of the same criteria for that. It's just prescribed, same specialists that we would be looking for. So oncologist or hematologist, we would want them to have the diagnosis of follicular lymphoma. And then for 3, this is really mirroring the labeling. And then also we did take into account some of the NCCN guidelines that are written. So that's why you'll see for 3, it's either 3 or 4. But for 3, it would be, if used as first line treatment, lenalidomide will be used in combination with another medication. So you'd probably see a rituximab or obinutuzumab, hopefully I pronounced that correctly, or the patient was previously treated with at least 1 prior regimen for follicular lymphoma. So examples of those kind of treatment cycles that we would be looking for, and these are really pulled from NCCN, like I mentioned. Same reauth duration is 6 months, same reauthorization criteria as above, and the same reauthorization duration. For the next indication, it's Kaposi sarcoma. It applies to pomalidomide. For this one, it's the specialists are a little bit different. It looks like there are oncologists, dermatologists, or infectious disease specialists that we would be looking for. We would, of course, want the diagnosis of Kaposi sarcoma. For this one, the patient has progressed on at least 1 prior systemic treatment. So that would be liposomal doxorubicin or paclitaxel, unless it's contraindicated. And if the patient is HIV positive, the patient remains on a antiretroviral therapy. For this one, same reauth duration, same reauthorization criteria, and same reauthorization duration of 6 months. And then, of course, the disease stability or no progression. For this one, it's mantle cell lymphoma for lenalidomide. This one, pretty simple criteria. Oncologist, hematologist prescribing it, or in consultation, diagnosis of mantle cell lymphoma, and lenalidomide will be used in combination with rituximab. Same reauthorization duration, and then reauthorization criteria. The next indication is marginal zone lymphoma for lenalidomide. For this one, same specialist as the criteria above. The diagnosis of marginal cell lymphoma. For this one, we did also take into account NCCN guidelines as well. So that's why you'll see the "or" here. For this, this is pulled from the label, so it's used as first-line treatment. Lenalidomide will be used in combination with another medication, so rituximab or obinutuzumab, or they've been previously treated with at least 1 prior regimen for marginal cell lymphoma, and these are the examples of the treatments that we would be looking for. Same duration, 6 months, same reauthorization criteria and reauthorization duration. Next is multiple myeloma. This applies to all 3 of those drugs. And just a note, we do have all of these products preferred with PA on our Apple Health preferred drug list. So in order for a patient to get these medications, they would just need to meet these criteria for this. And regardless of what drug they're requesting, as long as they meet them, they could possibly get approved for it. So for this, same specialist, oncologist or hematologist, diagnosis of multiple myeloma. For lenalidomide, specifically or Revlimid, we would be looking for that the medication will be used with dexamethasone as part of a doublet or triplet regimen, or if it's used as a maintenance therapy, the medication may be used as monotherapy. For pomalidomide, we would be looking for the patient has received at least 2 prior treatments for multiple myeloma, including one with lenalidomide and a proteasome inhibitor. And the patient has demonstrated disease progression on or within 60 days of completion of the last therapy. And C, the medication will be used with dexamethasone as part of a doublet or triplet regimen. Or for thalidomide, this one, the medication will be used with dexamethasone or prednisone. Same duration, 6 months, same reauth, and same reauthorization duration. For myelodysplastic syndrome, that's for lenalidomide, same specialist, diagnosis of myelodysplastic syndrome. 3, the patient has lower risk disease, so IPSS lower intermediate 1. These are the different, I guess, risks that we would be looking for. And 4, the patient has transfusion-dependent anemia defined as 2 or more units of red blood cells in the previous weeks. And we would be looking for MDS, so myelodysplastic syndrome, with a del5q abnormality, or if the patient has MDS without the del5q abnormality, we would be looking for these particular lab values. So serum erythropoietin levels are less than 500. And they had a history of an inadequate response to erythropoiesis stimulating agents with or without granulocyte colony stimulating factor, or if they have serum erythropoietin levels greater than 500, we would also be looking for history of failure, contraindication, or intolerance to immunosuppressive therapy, so anti-thymocyte globulin with or without cyclosporin A, or demethylating agents, so azacitidine or decitabine, or a verified SF3B1 mutation. That was a mouthful. Same duration, initial approval duration, 6 months, same reauthorization criteria and duration that we would be looking for, 6 months. And then for this one, it's Poems syndrome for lenalidomide. This one, same specialist, oncologist or hematologist, diagnosis of Poems syndrome. 3, the patient has disseminated disease, so we would be looking for more than 3 bone lesions, and the patient is not a candidate for radiation-only therapy. And 4, the provider attests that the patient is not a candidate for hematopoietic cell transplant. And 5, the medication will be used in combination with dexamethasone. So same 6 months, disease improvement or no progression, and then same reauth 6 months. These are the dosage and quantity limits. It's pretty extensive. Well, I guess it looks a little long, but these are all the indications. And then the background. And then looking at the form, let me pull that up here. Is it? Sorry, it's not pulled. There we go. Okay, this is the form that goes with this policy. And I will go ahead. Let me just see. Okay, I'll try to do it slowly, but I will go ahead and stop for any questions as I slowly scroll through the form for you all.
Thank you, Marissa. While Marissa does that, any questions from the board for Marissa?
Marissa, this is John McKay. I was just wondering if there are any non-oncological indications that we might have to call it like a pregnancy contraindication or anything like that.
This is Marissa. Sorry, so you're
Just
asking if
in
we should include
terms
that
of safety profile for pregnancy, is there any place in there where that would be warranted in terms of monitoring or a contraindication for like the non-oncological indications? Or are they all for cancer?
It looks like for the most part, and I apologize, I did not draft this policy, but for the most part, I believe most of these are the cancer indications. The ones that they're not the cancer indications, I can take a look at that and see if we want to consider adding that to the policy for those non-cancer indications, I think is what you were asking, right? Okay, I just want to make sure I understood. I could take that back and see if we want to consider adding that to the criteria.
The one thing, this has a pretty strict REMS regardless of the indication, so you don't really need, I don't think you need to put that in the policy. There's a lot of paperwork the provider has to do in order to, like, you literally have to write the prescription every month and like check the type of birth control the patient's on, so I don't think we need to be that redundant.
Okay. There's a strict REMS for it.
Yeah, for all of these, where I mean, the Milamide is like the classic drug that we remember from all the birth defects that I'm sure you're referring to.
I think Laura had her hand up for a sec. Okay.
I had the same comment.
Okay. Other questions or comments? And then if there are any stakeholders, there are none listed.
Looking on the list, I don't see anyone's hands raised. No.
Thank you, Nanya.
Mhm.
Kavita here. I guess my question is a basic one. The reason for authorizing for 6 months at a time is because that's the usual length of treatment, or the risks, the adverse effect risks go too high after that? Usually the authorization is for a year at a time.
Yeah, this is Marissa. So we just made it 6 months just because of the continuous kind of monitoring that we would want to be looking for these drugs. So that's why it's a little shorter than what you typically see in most policies where it would be a year. So that's why it's a little bit shortened. We would just want to make sure that that monitoring is being done. And then on our end, we're looking for those monitoring parameters. So that's why. I'm not sure if the treatment duration, I apologize if those are 6 months or not. But it's really just from like a safety and monitoring perspective.
Okay. Other questions or comments? And if not, then we can move to the motion.
This is Michael Corsellis. I'd like to make the motion. Move that the Apple Health Medicaid program implement the clinical criteria listed on policy 20-1 as recommended.
It's Christy Weiland, I second.
All in favor, please say aye.
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 Any opposed or abstain? Okay, and the motion carries. Thank you, board. And I know that with that we would move on to anticonvulsants, but I also realized that our lunch started a little earlier. Is the board okay continuing? I see. Okay, good. I see nodding. So, all right, Umang.
Alrighty, can you hear me?
Yes, we can.
Okay, so I think my video stopped, so I don't know if you can see me, but
yeah, we can't see you.
You can't? Okay.
Yeah.
Okay. Well, that's okay. Marissa's gonna
Sorry,
share.
Umang. I'm gonna go ahead and pull your slides up.
That's okay.
So just give me 1 second.
Yeah.
A lot
No
of
worries.
documents to sift through today.
You're doing an amazing job, Marissa. Thank you.
Thank you. Let me pull everything up here. Get your slides. Okay, I'm almost there. I just got to get the motion slide pulled up and then we should be good to go. So then have everything at once. Okay. Alrighty. Let me share this. All right. All right, Umang, I think you should be good to go.
Okay. Alrighty. Perfect. Well, I will be reviewing the next few topics here. Just a quick reminder to some of the committee members, some that are new or some that may have forgotten, what I'll do is when we review significant clinical updates within the last 12 months or so, I will provide some background, some guideline updates if they are relevant within the last few years. And then we'll go right into the significant clinical updates. So the first one being anticonvulsants. So if we could just go to perfect. And again, just a reminder for some of the committee members, if you see, on the title page of each subclass, we have what the class is in our Magellan Prime system, which is anticonvulsants. And then we also have the Apple Health PDL and how it is listed in the PDL. So anticonvulsants is broken down by mechanism of action, which includes glutamate receptor antagonists, benzodiazepine rescue agents, miscellaneous neuroactive steroid GABA modulators, and succinimides. So moving onward, a little bit of background here. So epilepsy is one of the most common disorders of the central nervous system. It's defined when a person has 2 or more seizures. It affects about 2.2 million Americans with 150,000 new cases diagnosed each year. The risk is estimated to be 1% from birth to age 20 years and 3% at age 75. Isolated seizures may also occur during a febrile illness, after head trauma, or as a result of withdrawal from alcohol or sedative hypnotics. A seizure is traceable to unstable cell membrane or cluster of cells, and excessive excitability spreads either locally, which is defined as a partial seizure, or more widely, which is a generalized seizure. Partial seizures begin in one hemisphere of the brain, and unless they become secondary generalized, they can cause alterations in motor functioning, sensory symptoms, or automatisms. In terms of if there is no loss of consciousness, they are called simple partial. If there is loss or impairment of consciousness, they're called complex partial. And about 70% of patients with epilepsy can be maintained on one drug. Non-compliance and evolving refractory epilepsy are common reasons for treatment failure. And if control is not achieved with one drug, an alternative medication should be attempted before others are added to current therapy. On the next slide here, we'll give a little bit of guideline update. So in 2022, the International League Against Epilepsy developed classifications for epilepsy syndromes. An epilepsy syndrome is defined as a characteristic cluster of clinical and EEG features, often supported by specific etiological features, such as structural, genetic, metabolic, immune, or and infectious. Through a series of publications, the ILAE provides definitions and classifications of syndromes according to categories divided by age of onset. They commonly have age-dependent presentations, features, and specific comorbidities. Diagnostic criteria includes age at onset, neurological exam, genetic studies, and of course, illness. The classification of epilepsy syndromes in neonates and infants focuses on the clinical and lab laboratory features of epilepsy syndromes with onset from birth to 2 years of age, and includes two major groups of syndromes: self-limited epilepsy syndrome and developmental and epileptic encephalopathies. Generally, children with epilepsy developing very early in life experience significant cognitive and behavioral comorbidity and have a higher rate of drug resistance. And the classification of syndromes with an onset between ages 2 and 12 years are categorized broadly as self-limited focal epilepsies, generalized epilepsy syndrome, and developmental and/or epileptic encephalopathies. Childhood syndromes may evolve from syndromes of infancy or may present with severe acute encephalopathy following prior to normal development. Idiopathic generalized epilepsy syndromes and syndromes that occur with a variable age of onset, either in childhood or beyond the age of 18, are further described and categorized by the ILAE. On the next slide, just again, as you can imagine, their anticonvulsants can have a lot of subsections, so I'll do my best here to kind of give you a breakdown. First, we have Lennox-Gastaut syndrome. It is one of the most severe forms of childhood epilepsy and is one of the hardest forms to treat, characterized by mental retardation and multiple seizure types. Patients have seizures daily, sometimes experiencing several seizures within a day. And patients may also experience drop attacks, which is defined as a loss of muscle control, causing the patient to fall abruptly to the floor. Next, we have infantile spasm, primarily consists of sudden bending forward of the body and stiffening of the arms and legs. West syndrome is characterized by infantile spasms, developmental regression, and a specific pattern on the EEG testing called hypsarrhythmia. So it's chaotic brainwaves. The onset is usually in the first year of life, typically between 4 and 8 months, and usually stops by age 5, but may be replaced by other seizure types. Then we have Dravet syndrome, a rare catastrophic form of epilepsy that presents in the first year of life and is characterized by frequent prolonged seizures. Patients may experience multiple seizure types during their lifetime. Infants with Dravet syndrome often experience multiple comorbidities over their lifetime related to the persistent seizure activity, including behavioral and developmental delay. Dravet syndrome is also associated with a 15 to 20% mortality due to sudden unexpected death in epilepsy. The goals of treating epilepsy are to reduce the frequency of seizure occurrence, along with providing the best possible quality of life for the patient. Treatment will depend on the type of seizure. Many different classes of drugs are available to treat the different forms of seizures, and some patients will require more than one drug to control said seizures. Let's see on the next slide here. Okay, we'll go into drug-specific updates. So first, new generics. In March of last year, the FDA approved topiramate, the first generic to the 200 milligram ER oral capsule of Supernus Trokendi XR from Zydus. Then we have methotrexate. In May of last year, the FDA approved the first generic for the 300 milligram oral capsule of Park Davis's Solontin from Novidium. We did have a manufacturer communication April of last year for Fintepla. The DEA published a final ruling declaring the removal of fenfluramine and associated salts, isomers, and salt of isomers from the schedules of the Controlled Substance Act effective December 23rd, 2022. Fenfluramine was previously a Schedule 4 controlled substance since 1973. And UCB Pharma filed with the FDA for a labeling supplement to remove the Schedule 4 designation from the label. And then a new dosing for Vimpat, May of last year. The package insert updated to include loading dose and or higher initial dose during week 1 as an alternative option for initiation of lacosamide in patients 1 month or greater to less than 17 years of age with partial onset seizure for monotherapy or adjunctive treatment, and for patients 4 years of age or older to less than 17 years of age with primary generalized tonic-clonic seizures for adjunctive treatment. This new dosing can be applied for all formulations of the drug and can be utilized when reaching the maintenance dosage in a shorter time frame is indicated. Labeling includes a table with the recommended loading doses according to patient age and weight. Going on to the next slide here. Alrighty, so next we have Poly XR. May of last year, FDA approved a new formulation for the treatment of partial onset seizures in adults and pediatric patients weighing 50 kilograms or more. So there are some warnings here. Fetal toxicity. So use during pregnancy can cause cleft lip and or palate and being small for gestational age. Suicidal behavior and ideations, and cardiac rhythm and conduction abnormalities. In terms of dosing, it is stratified by age and weight. For adults, defined as 17 years of age and older, initial dose for monotherapy is 200 milligrams daily. Initial dose for adjunctive therapy is 100 milligrams daily, and the maximum dose for mono or adjunctive therapy is 400 milligrams daily. And for pediatric patients weighing at least 50 kilograms, the initial dose is 100 milligrams daily. As you can imagine, the availability are extended release capsules in 100, 150, and 200 milligram dosage forms. Righty. And I'm going to pause right there for the committee. Any questions?
Thank you, Aman. On the board, any questions for Aman? And if there are any stakeholders?
It looks like we have one hand raised.
Okay, great. Call on them, Nanya. I don't see it.
Okay, first one we have is Regine Amen, and I'm so sorry if I'm pronouncing your name incorrectly. Regina, you can unmute now.
Can you hear me now?
Yes.
Yes.
Thank you. Yes, I did not fill out the form, so I am not a provider for Washington. I'm not a patient. I don't have any conflicts besides I work for UCB Pharma. I wanted to present the Nasalem, so that's what I'm going to do right now. Good afternoon. My name is Regina Amin. I'm the medical outcome liaison for UCB. I'm here today to discuss seizure clusters, specifically the unmet need, treatment need, and socioeconomic burden, and UCB's product Nasalem, and its request for its placement on the preferred drug list. Seizure clusters are seizure emergencies which are defined as having 2 or more seizures in 24 hours and are different from a patient's typical seizure pattern. Seizure cluster are serious medical events that need to be stopped promptly to avoid potential serious consequences, such as increased risk of status, hospitalization, and greater use of emergency services. For perspective, in the state of Washington, you can anticipate roughly about 74,000 patients with epilepsy, of which 11,000 experience seizure clusters. Nasalem is the first FDA-approved nasal formulation for acute treatment of seizure clusters in the outpatient setting in patients 12 years and older. It is the only midazolam-based option approved for the treatment of seizure clusters. Nasalem is a benzodiazepine indicated for the acute treatment of intermittent stereotypic episode of seizure activities or seizure clusters in patients 12 years and older. It has a Tmax of 17 minutes, followed by a single dose and a half-life of 2 to 6 hours. Nasalem demonstrated efficacy in stopping seizure clusters in a phase 3 double-blind placebo-controlled study of 292 patients, in which significantly more patients received a single dose of Nasalem, experienced treatment success considered placebo, 53.7 Nasalem arm versus 34.3 in the placebo arm. In 81% of Nasalem treated patients, seizure terminated within 10 minutes, and 50% of patients had no risk of recurrence within 10 minutes to 6 hours. Additionally, more patients had a probability of no seizure recurrence for 24 hours after Nasalem treatment. After treatment, return to baseline functionality was assessed by the caregiver, and the median return to full baseline functionality was after treatment with Nasalem was about 90 minutes. Like all benzodiazepine, Nasalem does have a box warning for risk of concomitant use of opiates, risk of abuse, misuse, addiction, and dependence and withdrawal. Please refer to the PI for a full box warning as well as other important information. The most common adverse reaction are somnolence, headache, nasal discomfort, and throat irritation, rhinorrhea. Nasalem is applied in a single dose strength in a box containing 2 5-milligram nasal spray units and does not require weight-based dosing. While the majority of Nasalem patients in the clinical trials did not require a second dose, you can administer a second dose as early as 10 minutes after the first. Thank you for your time today, and please consider putting Nasalem as a preferred status for Washington Medicaid patients. Thank you.
Thank you, Regina. Any questions from the board for Regina? Great. And any other stakeholders, Nanya? Okay.
Hello, can you hear me?
Okay, hi, Amanda.
Hi, how are you?
Well, go ahead. And so if you can answer those questions, and then we'll start your timer.
Yep, my name is Amanda Rendell. I'm an employee of Neurelis, and I'm not a patient, I'm not a provider, and I don't have any other conflicts of interest to report besides being an employee of Neurelis.
Great, thank you. Go ahead.
So we request that Valtoco remained in a preferred position without restrictions. With that, we'd like to use this time to review a few key highlights on Valtoco. Valtoco is an intranasal diazepam spray for emergency rescue treatment of seizure clusters and is the first and only intranasal rescue treatment for adults and children down to the age of 6. Neurelis is currently conducting a clinical trial in patients aged 2 to 5, where we are investigating the safety and pharmacokinetics of Valtoco for label expansion to include these younger 2 to 5-year-old children. Valtoco was designated clinically superior to Diastat, the diazepam rectal gel, by the FDA. Valtoco is the only intranasal rescue medication that allows for customized dosing based on age and weight, with 5, 10, 15, and 20 milligram doses available. An optimal dose may be administered to pediatric and adult patients alike. Each box contains 2 doses of Valtoco. If a second dose is needed, it may be administered at least 4 hours after the initial dose. Our PK studies demonstrating consistent and reliable dosing with 97% absolute bioavailability of diazepam relative to IV, and PK parameters that were 2 to 4-fold less variable when compared to Diastat. When treating seizure clusters with rescue medication, the primary goals are to stop the initial seizure and prevent reoccurrence of seizure activity over time. Valtoco has a half-life of 49.2 hours, which would allow coverage within the expected 24-hour timeframe of a seizure cluster. In an exploratory analysis from a long-term safety study, patients with epilepsy and their care partners reported rapid Valtoco administration and seizure cessation, with a median time to administration of 2 minutes and a median time to seizure cessation of 4 minutes. A separate exploratory analysis demonstrated that in almost 4,000 seizure cluster events treated with Valtoco, 87% of events were treated with a single dose over a 24-hour period. Valtoco has a box warning, as do all benzodiazepines, regarding concomitant use with opioids, abuse, misuse, and addiction, and dependence and withdrawal reactions. The most common local AEs were nasal discomfort, dysgeusia, epistaxis, and in a long-term safety study, the rate of somnolence was 1.8%. For additional prescribing and important safety information, please refer to the full prescribing information for Valtoco. I'd like to thank you for your time and ask that you please continue to allow patients to have unrestricted access to Valtoco. And I'm happy to take any questions you may have.
Thank you, Amanda. Any questions from the board for Amanda? Okay. And any other stakeholders, Nanya?
No, I don't see any other stakeholders. I realized earlier that I was talking while muted, but
And
no one else.
Marissa, oh, okay, thank you. And Marissa, I know you were starting to show us the list on the Apple PDL.
Hi, this is Marissa. Yep. So I will go ahead and go through the PDL for you all. So just going to go class by class for all the anticonvulsants. So the first one is the AMPA glutamate receptor antagonist. The only product in that class right now is Fycompa, and we have that preferred, both the tablets and the suspension. For the benzodiazepine rescue agents, our preferred products in this class are the Diastat. So those are the diazepam rectal gels, various midazolam. I believe these are injections for IV. And then we do have Valtoco preferred, which is the diazepam nasal spray. For the anticonvulsants miscellaneous, the preferred products in this class are, we have Briviact, which is brivaracetam. It looks like that's an IV. We have carbamazepine, which is different formulations, chewables, suspensions, tablets, ER capsules, ER tablets, everything. We have Epital, gabapentin capsules, solutions, those are preferred. Generic glucosamides, solution and tablets. apologize for the scrolling, lamotrigine tablets, and various levetiracetam's. So we have some injection, IV solutions, oral solutions, tablets, ER tablets, and oxcarbazepine, suspensions and tablets, pregabalin, capsules, solutions, primidone tablets. This, it looks like, is a brand name. This is Rowipra. Looks like that's a levetiracetam 500 milligram. We have that preferred. We have Subvinite, which is lamotrigine, different carbamazepines, topiramate, different formulations, sprinkle capsules and tablets. And I think I mentioned everything. Oh, and some zonistamides, capsules and oral suspensions. In the neuroactive steroid GABA modulator, it's Ztalmy. We do have that non-preferred. And then for the succinamides, our preferred product in that class is the ethosuximide products. Looks like it's the capsules and the solutions. And I will go ahead and pause there for any questions about the PDL.
Questions from the board?
So you mentioned that the diazepam for the rescue, there's rectal and inhaled or intranasal preferred, but for midazolam, there's just injectable preferred. I think not the nasal that she was
Correct.
talking
We
about.
don't have the nasal preferred.
Okay.
Yep.
Are there like compelling reasons for that? Is it really new? Is it really expensive? Or do you know?
This is Marissa. Yeah, when we did take a look at it, there were some cost considerations that we took into account. So that's part of the reason why.
Do you know if there's anything like about if somebody fails the diazepam, then they can get that still, or what the details of that are at all?
Yeah, so that would be the expectation. This is Marissa, sorry. That would be the expectation for someone if they wanted to get midazolam nasal spray, is that they would have stepped through, you know, either the diazepam rectal didn't work out or the nasal spray didn't work out. That would be the expectation, and then they would be eligible for the nasal.
Okay, sounds
Yep.
good. Thank you.
And the IV is covered, right? So like if you just had an atomizer, it's probably what I guess you were thinking of. You could just use that.
This is Marissa. I'm sorry, Kevin. Can you repeat that question one more time?
Because if you have an atomizer, you can
Yeah.
give the IV nasally. So I'm guessing that's what you were thinking. And that would probably,
Yes.
I don't know.
Yeah.
Kevin, this is Donna. Yes, that was originally before Nasalam came to market. That was the intention of having the injectables preferred.
Got it.
Yeah. Okay.
This is Laura Bestie. I don't think that the Nasalam product is indicated for children under age 12, and the diazepam it is for I think ages 5 and above. So you've got better coverage age-wise with the diazepam nasal than the midazolam.
This is Donna. Yep, I agree.
Great. Other questions for Marissa? Okay, and if not,
Does the HC ever have or bring like data on, I guess maybe you already use that to decide what you want to propose to us, but like data on which, if there are any things that are like frequently requested that are not preferred, or because like we hear from the stakeholders from the pharmaceutical companies, but we don't always have doctors on our committee who have personal experience with all of these things. And so I feel like the best data would be like, what are people requesting out in the real world? Do you already take that into account before this, or do you ever have that to?
So this is Donna. So,
yeah.
That's a tricky question to answer, Zoe. Sometimes what's being most requested is a result of a very good marketing campaign of the manufacturers rather than what is the more effective or cost-effective treatment. I haven't seen any, I won't say I haven't seen any requests for Nasalam. I haven't seen any provider complaints, get escalated up to our attention about the diazepam nasal spray being preferred over the
Yeah, no
midazolam.
worries.
Yeah, so
I was just wondering if there was like, how that gets brought into account.
we would probably look at, how often are we approving it? And then at what point in time, if there's a rebate on it, are we leaving money on the table? And at what time does it make sense for us to, based on utilization volume, we would adjust, maybe make it preferred because there would be additional savings. But we monitor that, pretty much on a quarter to quarter basis.
Okay, cool. Thank you.
Go ahead, Peter.
Donna, this is just a follow-up to Zoe's question. Maybe one other way of getting at, I think what she was driving at is, do you have a process for tracking outcomes of appeals, overturn rates, and then kind of reviewing that data and triggering something to come to P&T?
We track our prior authorizations and what's approved and denied. Unlike a commercial health plan, we don't have what we call appeals. We might get a request for reconsideration, which I believe gets counted as just another request for authorization in our data. So we don't track it from the client. We got a request for client A, it was denied, provider requested a second, a reconsideration, or what in commercial world would be an appeal, and then it got approved. We don't track it linearly like that. So I can't really answer your question there.
Okay, no, and I guess I didn't realize that you don't follow the same appeal process. What about, you do follow like peer-to-peer, right? Like
there's there's a request for reconsideration. You can request a peer-to-peer. You can go straight to an administrative hearing also. But again, they're not tracked. The hearings are tracked completely separately because that's a JLA, administrative function. But we don't, if you had a peer-to-peer and the decision was to reverse the original denial, it doesn't, like, again, I don't track, it'll get put in as a new authorization. It doesn't get tracked back to that original denial to have that, here's the full story for this person's, request for this drug.
That answer your question, Peter?
Yeah, that's helpful. I guess I wasn't aware of how, like, some of the differences. That's helpful. Thank you.
Any other questions for Marissa or the HCA team? And Marissa, if you can get the motion on the screen in the meantime. Okay, and whenever the board is ready.
This is Greg Hudson. I can read the motion. I move that all products in the drug classes listed on slide 2 are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of the HCA. Products in these classes may require prior authorization to determine medical necessity. All non-preferred products require a trial of at least 2 preferred products with the same indication prior to a non-preferred drug, same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
It's Peter Barkett. I'll second the motion.
All those in favor, please say aye.
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 Any opposed or abstain? Okay, with that, the motion carries. And back to you, Mun, for anti-dementia agents.
Oh, alrighty. So the next subcategory we have are Alzheimer's agents, which are anti-dementia agents overall and anti-myeloid antibodies. So a little bit of background here on the next slide. Dementia is characterized by irreversible loss or decline in memory and other cognitive abilities. Approximately 6.5 million Americans aged 65 and older suffer from Alzheimer's. It is the most common type of dementia, accounting for 60 to 80% of dementia disorders in the elderly, and is the 5th leading cause of death in the United States. Other types of dementia include vascular dementia, dementia with Lewy bodies, mixed dementia, and frontotemporal dementia. It may be associated with HIV, normal pressure hydrocephalus, Huntington's disease, Korsakoff syndrome, MS, Parkinson's, and Creutzfeldt-Jakob disease. Many other conditions can cause delirium symptoms such as thyroid disorder, vitamin deficiencies, but are reversible once the underlying condition is addressed. In terms of Alzheimer's, it is characterized by progressive cognitive decline associated with impairment of activities of daily living and behavioral disturbances. Patients with Alzheimer's eventually lose all cognitive, analytical, and physical functioning. 10 warning signs of Alzheimer's include memory loss that disrupts daily life, challenges in planning or solving problems, difficulty completing familiar tasks, confusion with time or place, trouble understanding visual images and spatial relationships, new difficulties with speaking or writing, misplacements of items or losing the ability to retrace steps, decreased poor judgment, withdrawal from work or social activities, and mood or personality changes. In addition, there are 3 stages of Alzheimer's over the course of the disease, characterized by symptom severity, rate of disease progression, and level of necessary supportive care for activities of daily living. On the next slide here, in January of last year, we have Leqembi. FDA granted an accelerated approval for Leqembi, which is an amyloid beta-directed antibody indicated for the treatment of Alzheimer's. Labeling states treatment should be started in patients with mild cognitive impairment or mild dementia stage of disease, and the population in which the treatment was initiated in clinical trials, as there is not safety and efficacy data on starting treatment at earlier ages or later stages of the disease than were studied. In terms of limitations, pregnancy is one of them. Based on animal data, it may cause fetal harm. And there is a black box warning for monoclonal antibodies directed against aggregated forms of beta amyloid, including Leqembi, can cause amyloid-related imaging abnormalities, or ARIA, characterized as ARIA with edema and ARIA with hemosiderin deposition. ARIA is usually asymptomatic, although rarely serious and life-threatening events can occur. Serious intracerebral hemorrhage greater than 1 centimeter have occurred in patients treated with this class of medications. The dosage is 10 mg per kg that must be diluted then administered as an IV infusion over approximately 1 hour every 2 weeks. And the availability is in an injection form, a single dose vial in 500 mg per 5 ml and 200 mg per 2 ml solutions. Moving onward, CMS Communication in June of last year announced that Medicare Part B will cover drugs that slow the progression of Alzheimer's disease if 1, the drug is granted traditional FDA approval rather than the accelerated. 2, patient is diagnosed with mild cognitive impairment or early dementia due to Alzheimer's. And 3, the patient is followed by a qualified physician participating in a registry that has appropriate follow-up care. And lastly, there was a drug discontinuation earlier this year, Aduhelm in February. Biogen announced a realignment of resources in their Alzheimer's disease portfolio, resulting in the discontinuation of the development and commercialization of Aduhelm. They stated that the decision to withdraw resources was not the result of any safety or efficacy concerns, but allows for reprioritization to focus on Leqembi as well as potential pipeline products. The rights to this will revert to Neurimmune, and the phase 4 post-marketing confirmatory Envision study will be terminated. And just an FYI, this did receive accelerated approval back in 2021. So I will pause there for the committee.
Thank you, Mug. Questions from the committee for the board? For Umang, I mean. Oh, Peter, go ahead.
Thanks, Mung. Yeah, I was curious first about the Aduhelm, and it sounds like, well, you answered it. They just want to double down on Leqembi. But the other thing I was curious about is donanemab. So I know it's not FDA approved yet. I was kind of waiting for the word on that, kind of any time now. And I'm curious if you've seen any of that data, if we're going to bring this class back when there's a verdict on donanemab, particularly if it gets FDA approval, or how you're thinking about that agent.
Yeah, Dr. Burkett. So this is Umang. I haven't seen the data yet. We usually focus on, we kind of dive more into the data once it is officially approved. Once it is approved, we do take a look at it. We do update these clinical review documents that we provide the committee. I may defer the second part of your question to Marissa, or at least have her verify what I'm saying is honest. But I believe based on the scheduling of these disease states, this would be revisited again next year in 2025. And that is when, if it is approved between now and then it would fall into that review cycle.
This is Marissa. Yeah, so just to verify, we typically want to review classes annually, especially the same types of classes. But of course, we could possibly, depending on kind of where it lands in our PDL, if it has to be like a one-off class, we could certainly bring it, not to a April meeting, but maybe to another one. So we do try to keep as closely to that annual schedule as we can. But if we do have to do a one-off, we certainly can for sure.
Yeah, I was anticipating the differential and interval dosing, but I believe Leqembi is also trying to come up with a 1 month interval dose too, so that might be a moot point. But we're kind of I was anticipating there might be a lot of interest in donanemab other than Leqembi based on the difference in interval.
I guess I have a question, probably more for like Marissa and Donna. For something like this, where there's a relatively newer medication and we know it's very expensive, remind me again, when would we look at the policy for that versus just approving it to be included in the PDL?
This is Marissa. So policy-wise, it probably, to be quite honest with you, as soon as if a drug were to come out tomorrow, for example, like get FDA approval, you probably wouldn't see a policy for that probably until a couple months later, depending on the need, or even, the next year, just because we do try to schedule our policies at least for the whole year and try to keep to that schedule. But depending on kind of the urgency that policies need to be created, we do tend to prioritize those, especially those that might have higher utilization than others. So especially for some of, the more rare diseases, they typically tend to get kind of prioritized a little lower, just because one, they're a little rare, so the patient population for them is a little bit smaller, that you wouldn't see very many requests for them. And also the urgency of them may not be necessarily needed because, they're very rare orphan diseases that we typically probably wouldn't see. So I would say it may take like a year for a policy for these products to come out from inception to, really getting implemented.
Okay, so for Leqembi specifically, the policy is not out yet. Is that?
This is Marissa. That's correct. Yeah, we don't have a policy for it, and we don't have a policy that we're working on right now for that one yet.
Got it. Thank you. Go ahead, Kevin.
I just, and Donna and Marissa, correct me if I'm wrong, just for everyone's knowledge, right? Because most of these patients are going to have Medicare, right? So you don't, if Medicare approves it, you don't, like as their secondary, you don't deny it or put any additional requirements on top of it. So there's really not a need for a policy for the vast majority of patients.
This is Marissa. Oh, that's a good question. Donna, I'm not exactly sure how to answer that, to be quite honest with you. I think we can still apply criteria to them if we need.
Okay, yeah, Kavita here. I suppose like because we're reviewing the agents here and the motion we're about to vote on says that we're approving their inclusion on the PDL, I assume that all of those medications that are on the PDL would require some kind of policy.
I just mean practically in terms of like approval, like almost not all, but a vast majority of these patients are going to be of Medicare age. So
yeah,
in terms of like waiting for approval for any new agents that come out, a lot of them are going to, if Medicare agrees to cover them, like by default, Medicaid would just cover the remainder, like their 20% copay, you know, what's left for Part B, right?
Mhm.
This is Marissa. I do believe there is that coordination that you are speaking of. But as far as putting prior authorization on some of them, I do believe we still have the ability to do that. So they can still go through, at least for Medicaid, a clinical review for appropriateness for those. The coordination of as far as Medicare and Medicaid, that's like a whole, I apologize, I'm not privy to kind of how that works. But I do know that we can create policies for those products.
Yeah. Go ahead,
I think
Peter.
on the coordination of benefits, unless Medicaid operates under a different set of rules than commercial, you're not allowed to apply any prior auth as a secondary insurance. So you just you get stuck with the bill. And as far as the kind of Medicare versus kind of non-Medicare insurances covering it, yeah, you're probably right. Well, maybe not most, but a disproportionate number of the patients that would get this medication would likely be of Medicare age at least. But not everybody qualifies for Medicare, and also because the medications are indicated for the very early stages of Alzheimer's dementia, you're going to pick up some patients in their 50s and early 60s before they become a Medicare age. So it's not exclusively Medicare, but overall, my understanding of utilization of this agent is very, very low so far.
And these would be the dual eligible patients too, right? Because we're talking about Medicaid. So, okay. Any other questions before we look at the motion? Okay, Marissa, if you can pull up our motion. And I did see that we did have some written stakeholder testimony that was submitted to us ahead of the meeting, which I'm sure we all had a chance to review. Okay, so here's our motion whenever the board is ready.
So I'm confused about like, do we also look at an Excel of these ones or no, just because there's so few? And is this the same process that we just did with the?
Oh, this is Marissa. Actually, yeah, thank you, Zoe, for pointing that out. I did not show you all the PDL. Let me show you.
Okay, great. Thank you.
What that is looking like. Okay.
Perfect.
Thank you. I forgot myself. Okay, so we will go through the anti-dementia agents. So there's like this class and then a subclass, which I'll show below. So for this particular drug class, this has really the traditional anti-dementia products that you would think of. So the non-Aduhelms, if you will. So the preferred products that we have in this class are donepezil with various types of formulations available. We have tablets, ODTs. We also have rivastigmine, so the patches, memantine, tablets and capsules. Those are preferred. And then it looks like the preferred products are the rivastigmine patches, the memantine capsules, and then donepezil tablets and ODTs. And then for the anti-amyloid antibodies, which is where Aduhelm and Leqembi live, that's where that houses those two products. Aduhelm is still included on our PDL right now, even though I know Umang did mention that it got withdrawn. Just on our drug file of where we get our source of truth for drugs, it hasn't been officially pulled from that database, so that's why it shows up on our PDL. But for those two products, we do have them non-preferred on our PDL, and of course there is PA on them. I don't believe, I haven't heard of us getting, to be quite honest with you, any requests for those two products yet. So I'm just throwing that out there as like an FYI. And I can answer any questions you might have about the PDL.
So by approving that motion, are we saying that all the drugs on this list are safe and efficacious, even the ones that are not preferred?
This is Marissa. Yep, that's correct.
Okay.
Yep.
So it looks like Leqembi is non-preferred, right? And there is a prior auth criteria, but if we approve the motion, Leqembi could be listed as a preferred agent.
Yeah, that's the problem.
Yeah, right. And I think there were like one or
it's
two
not
deaths in the study.
efficacious. So it's not true.
Yeah, like it's just not terribly efficacious, and there are
Yeah.
still some lingering safety concerns.
This is Marissa. And to speak to that, Peter, that's really why we made it non-preferred. I mean, even though there's two products in the class, it's just because the evidence that has been submitted for these drugs is not very strong for them. We didn't feel very strongly about making them preferred. So that's why you'll see that non-preferred positioning on our PDL.
Yeah, I just think it should be part of the motion. Like, this is kind of the whole point of this committee is like when there's a drug like this that's like super expensive, has a ton of marketing behind it, but it doesn't do anything. We're like the ones who say no, right? And so I think even though right now it's non-preferred, which is great, like our motion is still saying like we think this drug is safe and efficacious. So I think, can we change that? Like,
What does the committee think about that? About maybe just having a sentence in there about the anti-amyloid antibody agents that currently exist are not considered efficacious enough to be included in the PDL? And I think Donna and Marissa, I'll have you weigh in on that as well, because I don't think we've specifically excluded medications like that. Go ahead, Laura.
So my question would be, what about making it into its own class? I know sometimes even though it's for anti-dementia, it could be its own class, so then it wouldn't be part of the standard of care classes.
This is Donna. Which drug are we talking? Are we talking about Aduhelm?
Leqembi as well.
Leqembi as well. I think they are in their own classes. Marissa, are they both been pulled from the market at this point in time?
This is Marissa. So as far as I know, it's just the Aduhelm. They are housed in their own anti-amyloid antibody subclass. But I think what Laura was alluding to was, instead of, you know how for the motion we list all the drug classes and say all of these classes on slide 4 are safe and efficacious. What I'm hearing, it sounds like Laura was suggesting splitting it out, so having two different motions for them, and then I don't know, wordsmithing for the anti-amyloid antibodies for that. I think the safe
I
and
think you could just leave, you could the fact that Aduhelm is off the market, just because it's listed here doesn't mean that it's on the PDL. I mean, we can officially make it not covered on the posted PDL, but you could put all available drugs listed on slide XYZ, because that would.
I think Donna, Kavita here, I think the concern is also about Leqembi, that the evidence is not compelling to even include it on the PDL.
Well, we have to cover it. This is Donna. So we have to cover it. So we can either, you know, it's on prior authorization, and so we list it as a covered drug. It could be non-preferred rather than preferred. It makes no difference really in how it's treated, but we do have to cover it. So that's why we list it on the PDL.
Yeah, so my understanding is that the PDL is really about essentially step therapy, right? And regardless of whether Leqembi gets listed as a preferred drug, it's still going to have prior authorization criteria for medical necessity review.
Right.
So until we have like more than 2 agents in this class, which we may eventually have, the PDL is kind of a moot point. But I agree, it feels uncomfortable saying it's safe and efficacious.
Can we say like the jury is still out on that class in general? Like, instead of putting it in the safe and efficacious bucket, can we say like the committee has decided that there like isn't sufficient evidence to like make a decision about. So we
Typically,
go through the.
If you're going to say anything, I would say nothing about them rather than say that they are unsafe or unefficacious when they are an FDA approved drug. Because then you're going against what the FDA has said, and we generally recommend that you don't make those types of statements.
I can get, well, yeah, in the advisory committee, when it came to Aduhelm, like almost unanimously said, no, they're not safe and efficacious, and then the FDA approved it anyway, but Aduhelm's gone now. So Donna, I am comfortable doing kind of what you recommend and making a motion on everything other than the anti-amyloid antibodies and just being quiet on the anti-amyloid antibodies, and they'll continue to get treated the way they are now, not being on the PDL and having medical necessity review.
Right.
Can I just give everyone food for thought? I know all the concerns I think are very valid about the efficacy and the safety with the deaths, but I mean, consider the disease state, and we don't make those kind of statements for like oncology agents, right? Like some of them have very poor data or like incomplete data on their efficacy and like high risk with side effects. And not as acutely for this drug, but definitely, obviously, dementia is clear. We all clearly know the outcomes there. So, I'm just thinking we should kind of consider that too.
Yeah, that's fair.
Marissa, can you get the motion up again? I think that'll also help us think about the wording there and see if slide 4. And so basically, I think the point was from slide 4, do we just remove anti-amyloid antibodies as a drug class? And then we are not commenting on it. Does the committee agree with that on slide 4, removing the anti-amyloid drugs? Yes, no, maybe. I see one head nodding.
Yeah, I would pull the anti-amyloid antibodies and just vote on all the others.
I agree.
Okay. Any opposed to that? Any want to leave it on? Okay, so then let's go to the motion.
As Peter Barkett, I can make the motion. I move that all products in the drug class listed on slide 4 are considered safe and efficacious for their medically accepted indications and are eligible for preferred drug status, grandfathering at the discussion of the HCA. Products in these classes may require prior authorization to determine medical necessity. All non-preferred products require a trial of at least 2 preferred products. With the same indication before, non-preferred drugs will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
This is John McKay, second.
All right, all in favor, please say aye.
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 Any opposed or abstain? Okay, and with that, the motion carries. So I think for our schedule, and this is very well earned at this point, a break would be tidy for 10 minutes. And then we'll see maybe Marissa and team, you can look at the rest of the agenda and see what all we can get through by 4 o'clock.
This is Marissa. Yep, that sounds good.
You
Okay,
guys want to be, sorry,
yeah.
go ahead. Go ahead.
Yeah, that's what I was gonna say. We'll see you back at 3:12.
Okay,
Thank you.
thank you.
All right, let's have our board members come back on camera, please. Wonderful. All right, we have quorum. Next is oh, Marissa, any updates on the topics, or shall we just move forward with the atopic dermatitis agents?
This is Marissa. Yeah, so Kavita, I think we'll just move forward and see how far we get. We may just only be able to.
You cut out for me, but okay, what I heard was we move forward. So go ahead, Umang, we'll get started with the atopic agents.
Perfect, no problem. I did want to clarify, Dr. Chawla, I on the slides have cytokine and CAM antagonists, and that will encapsulate both cytokine and CAM antagonists and atopic dermatitis JAK inhibitors. So this will be an overarch of all cytokine and CAM antagonists. So
sounds perfect. Thank you.
It'll be a bit. So everyone, I hope you have some coffee. Okay. As you can imagine, everyone on the committee, this class has a lot of disease states within it because these are disease modifying agents in here. I did my best to kind of give backgrounds for the relevant disease states that hit the drug updates in this class. So moving right along, very overview here. Cytokine and cell adhesion molecules, or CAMs, are chemical mediators involved in the inflammatory process throughout the body. Cytokines are small proteins secreted in response to an immune stimulus for the purpose of mediating and regulating immunity, inflammation, and hematopoiesis. The actions of the individual cytokines are widely varied, and they contribute to fibrosis, tissue degeneration associated with chronic inflammation, primarily by inducing the proliferation of fibroblasts and collagenase. The pro-inflammatory cytokines, TNFs, IL-1s, are involved in tissue destruction in many chronic inflammatory diseases affecting various organs. So cell adhesion molecules, or CAMs, they are cell surface proteins involved in the binding of cells, usually leukocytes, to each other, endothelial cells, or the extracellular matrix. Specific signals produced in response to wounds and infection control the expression and activation of these molecules. Most of the CAMs categorized fall into the three general families of proteins. We have the immunoglobulin, the integrin family, and the selectin family. And the different CAMs here have been implicated in the inflammatory, fibrotic, and autoimmune diseases. For the committee, I know on some of these backgrounds, there's a lot of information. I've tried to just kind of highlight the key points here. Moving right along to disease background. First, we have hidradenitis suppurativa. It is a chronic condition that affects the terminal follicular epithelium, in the apocrine gland-bearing skin, such as the armpits and the perianal area. It typically occurs in adolescents, generally after puberty, and adults. It's generally diagnosed clinically and affects approximately 1 to 2% of the population in the US. Select signs and symptoms include erythema, raised bumps and lesions, painful lesions, and local arthritis and arthralgia. In addition to non-pharm treatments, pharmacological treatments include anti-inflammatories, antibiotics, anti-androgens, and biologics such as infliximab. Surgery may also be considered in some patients. Again, the guideline for this is very old. It's almost a decade old, so it's here for your referral. But the European Dermatology Forum does have a recommendation that's about 9 years old now here. Next, we have uveitis. So this is a non-infectious intermediate and posterior uveitis, is the inflammation of the intermediate and posterior uvea, while panuveitis is the inflammation of the anterior chamber, vitreous humor, and the choroid or retina simultaneously. Together, these represent the most severe and highly recurrent form of uveitis. The incidence of all cases is approximately 25 to 52 cases per 100,000 patients per year, and anterior uveitis is the most common form. Initial treatment is typically topical corticosteroids. Adalimumab is generally reserved for patients with disease non-responsive to initial treatment, and other treatments include systemic glucocorticoids, immunosuppressives, and intraocular implants. In terms of treatment, on the next page, we have guidelines from the ACR and Arthritis Foundation. It's about 5 years old now, from 2019. Again, just a quick overview. They recommend select topical glucocorticoids in patients with JIA and active chronic anterior uveitis for short-term control. But for those who are unable to control symptoms with short-term therapy, they recommend systemic therapy in order to taper topical glucocorticoids. Regarding specific agents, they recommend sub-Q methotrexate conditionally over oral methotrexate. However, use of a TNF antagonist with methotrexate in severe active disease and sight-threatening complications is conditionally recommended. Abatacept or tocilizumab as biologics and mycophenolate, leflunomide, or cyclosporin as non-biologic options are conditionally recommended in patients who have failed methotrexate and 2 monoclonal TNF antagonists. And for pediatric patients with spondyloarthritis, they do recommend topical glucocorticoids prior to a change in systemic therapy. And notably, the only agent approved for uveitis in this class, back when these guidelines were written, was adalimumab. On the next slide here, we have ankylosing spondylitis. So axial spondyloarthritis is an inflammatory condition generally affecting the spine and can be further subdivided into ankylosing spondylitis and non-radiographic ankylosing spondylitis. The American College of Rheumatology, the Spondylitis Association of America, and the Spondyloarthritis Research and Treatment Network published a collaborative 2019 update on the treatment guidelines for ankylosing spondylitis and non-radiographic axial spondyloarthritis. In general, recommendations for both ankylosing and non-radiographic axial are similar. TNF antagonists, not a specific one was listed, are recommended as first biologics over Cosentyx and Tremfya, which are then recommended over a second TNF antagonist if the first does not produce a response. All the prior mentioned agents are recommended over Xeljanz. Concurrent low-dose methotrexate with TNF antagonist is not recommended. They recommend against a strict treat-to-target strategy. If a patient's disease is stable, guidelines recommend against discontinuing or tapering of biologics. And sulfasalazine provides a viable option for select patients who cannot take a TNF antagonist. Next slide here, we have recurrent pericarditis. So acute pericarditis is the inflammation of the pericardium, and symptoms include chest pain, ECG changes, pericardial effusion, and pericardial friction rub. Typically lasts up to 6 weeks, although symptoms may recur and recurrence may be as high as 15 to 30% in select patients with idiopathic pericarditis. In recurrent pericarditis, these symptoms return after a symptom-free period of at least 4 to 6 weeks. Symptoms of recurrent pericarditis include pleuritic chest pain with fever, pericardial rub, ECG changes, new or worsening pericardial effusion, and/or elevation of markers of inflammation. Patients may feel well in between attacks, and others may have a more persistent disease course. Studies have suggested that many cases of recurrent pericarditis are caused by an autoimmune disorder, although other causes are possible, such as infection, and there are no well-established predictors of recurrence. In terms of treatment, the pharmacologic treatment of recurrent pericarditis is similar to the treatment of acute pericarditis and includes NSAIDs or aspirin, plus colchicine as typical first-line agents. Steroids or combination therapy may also be considered, and other agents that may be used for treatment in late-line therapy include rilonacept and the off-label use of anakinra, azathioprine, or immune globulins. And pericardiectomy may also be considered in select patients. Right. Next, we have polymyalgia rheumatica. So this is an inflammatory disorder characterized by pain and stiffness in the shoulders, upper arms, hips, and neck, and is worse upon waking or after rest. Flu-like symptoms, fatigue, and weight loss may also be present. Onset is typically seen after 50 years of age and most often occurs in the Caucasian and in women. The condition is associated with the development of GCA in approximately 10 to 15% of patients, and treatment guidelines are lacking. The 2015 Collaborative Initiative strongly recommends treatment with glucocorticoids to treat PMR episodes. The addition of methotrexate may be considered, particularly in patients at high risk for relapse and/or prolonged therapy or with comorbidities prone to steroid-related adverse events. The panel also strongly recommends against the use of TNF-alpha inhibitors for PMR. It is estimated that up to 29 to 45% of patients do not adequately respond to glucocorticoid therapy within 3 to 4 weeks, and relapses and long-term glucocorticoid dependency are common. In 2023, the FDA approved Kevzara as the first biologic treatment for treatment of adults with PMR who have an inadequate response to corticosteroids or who cannot tolerate corticosteroid taper. Next, we have atopic dermatitis, which is a chronic pruritic inflammatory disease of the skin resulting in combination of genetic and environmental factors. Often referred to as eczema. It can affect up to 15% of the children and a little over 7% of adults in the US. Commonly occurs in patients affected by asthma and other allergic conditions and is associated with elevated serum immunoglobulin E levels. It is characterized by extremely dry, itchy skin on the insides of the elbow, behind the knees, and on the face, hands, and feet. The American Academy of Dermatology last year had guidelines which only address topical therapies for the management of atopic dermatitis, but they state that moisturizers, topical corticosteroids, topical calcineurin inhibitors, topical PDE4 inhibitors, and topical JAK inhibitors are options with strong evidence for treatment of atopic dermatitis. Systemic immunomodulating agents are indicated for patients whose atopic dermatitis is not adequately controlled by topical regimens and/or phototherapy. Like the guidelines, the American Academy of Allergy, Asthma, and Immunology 2012 guidelines state first-line options include hydration, such as emollients, moisturizers, and topical corticosteroids. And they also recommend careful consideration of risk and benefits of systemic agents in patients who do not respond to topical agents or phototherapy. Abrocitinib and upadacitinib were also not addressed in those guidelines. Next, we also have ulcerative colitis, which is a chronic inflammatory disease primarily affecting the colon and rectum. It affects approximately 1 million people in the US, and the incidence continues to increase worldwide. The CDC estimates the current prevalence is 249 per 100,000 adults, may present at any age, and onset typically peaks between 15 and 30 years of age. The disease is characterized by superficial infiltration of the bowel wall by inflammatory white cells, resulting in multiple mucosal ulcerations and crypt abscesses. The predominant symptom is diarrhea, which is usually associated with blood in the stool. Additional symptoms may include pain in the lower quadrant or rectum, along with systemic features, including fever, malaise, and weight loss. The initial attack may be fulminant with bloody diarrhea, but the disease more commonly begins indolently with non-bloody diarrhea progressing to bloody diarrhea. It can present initially with any extent of the anatomic involvement, ranging from disease confinement to the rectum to the entire large intestine, defined as pancolitis. And most commonly, ulcerative colitis follows a chronic intermittent course with long periods of quiescence interspersed with acute attacks lasting weeks to months, and a significant percentage of patients suffer a chronic continuous course. Next, we have in regards to Crohn's disease. In 2021, the American Gastroenterological Association issued a guideline on the medical management of moderate to severe luminal and perianal fistulizing Crohn's disease, and notable recommendations regarding agents within the class are described below. In adult outpatients with moderate to severe, they recommend the use of a TNF antagonist or ustekinumab over no treatment for induction and maintenance of remission. And the AGA suggests the use of vedolizumab over no treatment for induction and maintenance of remission. In biologic treatment naive adult outpatients with moderate to severe, the guidelines recommend the use of infliximab, adalimumab, or ustekinumab over certolizumab, and suggest the use of vedolizumab over certolizumab for the induction of remission. In adult outpatients with moderate to severe Crohn's who never respond to TNF antagonists, the guidelines recommend ustekinumab and suggest vedolizumab over no treatment of the induction of remission. And if patients had previously responded to infliximab, they recommend adalimumab or ustekinumab over no treatment. And suggest vedolizumab over no treatment for induction of remission. And the group also recommends the use of biologic drug monotherapy over thiopurine monotherapy for the induction of remission. In adult outpatients with moderate to severe Crohn's who are treatment naive to biologics and immunomodulators, they recommend infliximab plus thiopurines over infliximab monotherapy, and adalimumab plus thiopurines over adalimumab monotherapy for induction and maintenance of remission. They do not make recommendations regarding the use of ustekinumab or vedolizumab. The AGA does suggest the early introduction of a biologic over waiting until failure of 5-ASA and or corticosteroids. For those with active perianal fistula, the guidelines recommend infliximab over no treatment for the induction and maintenance of fistula remission, and suggests adalimumab, ustekinumab, and vedolizumab over no treatment for the induction or maintenance of fistula remission. And risankizumab and upadacitinib were not approved for Crohn's at the time these guidelines were developed. And the role of Tysabri and other agents are not in this therapeutic class are also addressed in the guidance. Now, moving right along to drug-specific updates. First, we have Kevzara. Now, up until my presentation today, we've pretty much had new drugs. Just to remind the committee, when there are significant clinical updates, but not a new drug, I try to bold the relevant portions. So here, as you can see, Kevzara, the FDA approved for the treatment of adults with polymyalgia rheumatica who've had an inadequate response to corticosteroids, who cannot tolerate a steroid taper. Previously approved for the treatment of rheumatoid arthritis. So as you can see, it does already contain an additional indication. No changes to precautions or formulations. And with this new indication comes, of course, a new dosing. So the recommended dosage is 200 mg subcutaneously every 2 weeks in combination with the tapering course for the corticosteroids. And for this indication, it can be used as monotherapy following discontinuation of the corticosteroid. Next, Siltezo had a busy year last year. In March of last year, they FDA approved Siltezo, which was an interchangeable Humira biosimilar for the treatment of moderate to severe hidradenitis suppurativa in adults. It did have other indications such as rheumatoid arthritis, juvenile idiopathic arthritis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, ulcerative colitis, and psoriatic arthritis. In May, the FDA approved a new 40 mg per 0.8 milliliter single-dose pre-filled auto-injector pen presentation formulation. And then in July, the FDA approved this medication to have an additional indication for the treatment of non-infectious intermediate, posterior, and panuveitis in adults. Since there are significant, indications, the dosing is stratified by indication, age, and weight, which can be found in the TCRs or the package inserts. Again, no changes to the existing indications and no changes to the existing formulations here. Next, we have Amjevita. So in March of last year, the FDA approved this medication, which is a Humira biosimilar for the treatment of moderate to severe hidradenitis suppurativa in adults. In August, FDA approved the following new high concentration presentations of 20 mg per 0.2 milliliter, 40 mg per 0.4 milliliter, and 80 mg per 0.8 milliliter prefilled syringe, and 40 mg per 0.4 milliliter and 80 mg per 0.8 milliliter prefilled auto-injector. Again, no changes to precautions. And with this new indication for hidradenitis suppurativa, the dosing was on day 1, it was 160 milligrams given in 1 day or split over 2 consecutive days. Then day 15, 80 milligrams. And then day 29 in subsequent doses, 40 mg every week or 80 milligrams every other week. Okay, next we have Yusimry. In March of last year, FDA approved Yusimry, a Humira biosimilar, as a single dose 40 milligram per 0.8 milliliter prefilled auto-injector pen. Previously, it was approved as a prefilled syringe. April of last year, FDA approved a new indication for moderate to severe hidradenitis suppurativa. And in September of last year, they approved a new indication for non-infectious intermediate, posterior, and panuveitis in adults. No changes to precautions and formulations. In terms of dosing, you will see very similar, if not identical, dosing amongst a lot of these medications. So I won't try to repeat it for the sake of the committee's sanity, but it's essentially the same dosing in terms of, for hidradenitis, it's the same dosing as Amjevita in the previous slide. And for uveitis, it was 80 milligrams initial dose, followed by 40 milligrams every week or 80 milligrams every other week, starting 1 week after the initial dose. Next, we have Hyrimoz. March of last year, FDA approved Hyrimoz as a high concentration formulation in the following presentations: 10 milligrams per 0.1 milliliter and 20 milligrams per 0.2 milliliter prefilled syringe, and 40 milligrams per 0.4 and 80 milligrams per 0.4 prefilled syringe and auto-injector. In April of last year, It received a new indication for moderate to severe hidradenitis suppurativa in adults. And in September of last year, it was approved for non-infectious intermediate, posterior, and panuveitis in adults. No changes to precautions here, aside from the formulation changes in March, no other changes. And as you can see, the dosing is identical from its predecessors on this slide deck for hidradenitis and uveitis. Next, we have Euflyma. May of last year, FDA approved this medication, which is a biosimilar to Humira. It contains indications for rheumatoid arthritis, juvenile idiopathic arthritis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, ulcerative colitis, plaque psoriasis, and hidradenitis suppurativa. It contains the same precautions, black box warnings for serious infections and malignancy. Dosing is stratified by indication. And the formulation, you have a single dose prefilled auto-injector and single dose prefilled syringe with a safety guard and a single dose prefilled syringe without a safety guard. Okay. Next, we have Rinvoq. May of last year, FDA approved for adults with moderate to severely active Crohn's disease who've had an inadequate response or intolerance to one or more TNF blockers. It already contains approved indications for a litany of other disease states here. No changes to any of its warnings or black box warnings there. For this new disease dosing, the recommended induction dosage is 45 milligrams once daily for 12 weeks. The recommended maintenance is 15 milligrams once daily, and a maintenance of 30 milligrams once daily may be considered for patients with refractory, severe, or extensive disease. And no changes to formulations. Next, we have Hadlima. June of last year, FDA approved for the treatment of moderate to severe hidradenitis suppurativa. And then in July, they approved it for non-infectious intermediate, posterior, and panuveitis. No changes to precautions, identical dosing to some of the previous medications mentioned so far, and no changes to formulation. Alrighty, in July of last year, there were some new biosimilars to hit the market. Several Humira biosimilars launched per manufacturer press releases. These commercially available biosimilars include Cyltezo, Hadlima, Julio, Hyrimoz, Idacio, Euflyma, and Usimri. All products are available as low concentration formulations except for Hyrimoz and Euflyma. Hadlima, Hyrimoz, and Euflyma are available as high concentration formulations. And citrate-free formulations include Cyltezo, Hadlima, Julio, Hyrimoz, Idacio, Euflyma, and Usimri. Cyltezo is interchangeable with Humira. Next, we have Ilaris. In September of last year, FDA approved for gout flares in adults in whom NSAIDs and colchicines are contraindicated and not tolerated or do not provide an adequate response, and in whom repeated courses of corticosteroids are not appropriate. No changes to any of the precautions. For this dosing for gout flares, it is recommended for 150 milligrams sub-Q. In patients who require retreatment, there should be an interval of at least 12 weeks before a new dose of Ilaris is administered. And the formulation is 150 mg per milliliter solution in a single dose vial. Alrighty, and I apologize to the committee. I'm going to try to pick up my cadence just to be respectful of your time and hope to finish all of this in time. Next, we have Julio. September of last year, FDA approved for the treatment of non-infectious intermediate, posterior, and panuveitis in adults. As you can tell, identical dosing. No changes to precautions or formulations here. Then we have Entyvio. September of last year, FDA approved a single-dose prefilled syringe and a single-dose prefilled syringe in the strength of 108 mg per 0.68 milliliters for subcutaneous self-injection for ulcerative colitis. Previously, it was only available for IV injection. The IV route of administration is indicated for ulcerative colitis and Crohn's disease. After the first 2 IV doses at week 0 and week 2, vedolizumab may be switched to the sub-Q injection at week 6. Beginning at week 6 and thereafter, the dose is 108 milligrams subcutaneously once every 2 weeks. and therapy should be discontinued in patients who do not show evidence of therapeutic benefit by week 14. For patients in clinical response or remission beyond week 6, vedolizumab can be switched from IV infusion to sub-Q injection by administering the first sub-Q dose in place of the next scheduled IV infusion and every 2 weeks thereafter. Since this is just a new route of administration, allowing a transition from one to the other, again, no changes to the precautions there or indications, just a new formulation, as I mentioned. Next, we have abralato. June of last year, received approval for periodontitis. August, received approval for non-infectious intermediate posterior and panuveitis. In October, it was designated by the FDA as an interchangeable biosimilar to the corresponding presentations of Humira, 10 mg per 0.2 mils, 20 mg per 0.4 mils in prefilled syringe, 40 mg per 0.8 mils prefilled glass syringe, prefilled pen and glass vial for sub-Q use, and the interchangeable designation applies to all indications: rheumatoid arthritis, juvenile idiopathic arthritis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, ulcerative colitis, psoriatic arthritis, periodontitis suppurativa, and uveitis. Dosing, identical, as we've seen for periodontitis and uveitis here, and no changes to formulations. Next, we have Tofidence. October of last year, FDA approved this medication as the first biosimilar to Actemra. It is an IL-6 receptor antagonist indicated for certain adults with rheumatoid arthritis and patients 2 years of age or older with polyarticular juvenile idiopathic arthritis and systemic JIA. In terms of precautions, carries the same black box warnings as Tofidence. The dosing is stratified by the different types of arthritis, and it is based on their weight as well. As you can see, it is stratified here. For the formulations, it's an IV infusion injection in various concentration forms in a single dose vial for further dilution prior to IV infusion. Then we have Enbrel. October of last year, FDA approved my apologies. Should be Weslana. This is a typo here. As an interchangeable biosimilar to Stelara. It does carry the indication here for juvenile psoriatic arthritis 2 years of age or older. And it has the 0.8 mg per kg weekly with a maximum of 50 mg per week dose. Next slide, we have Bimzelx. FDA approved Bimzelx, a humanized interleukin 17A and F antagonist indicated for the treatment of moderate to severe psoriatic arthritis in adults who are candidates for systemic therapy or phototherapy. The precautions, infections and tuberculosis, specifically patients who have active TB. The dosing here, 320 mg sub-Q at week 0, 4, 8, 12, and 16. And then once we get to there, it's every 8 weeks thereafter. And for patients weighing over 120 kgs, consider a dose of 320 milligrams every 4 weeks after week 16. And the formulation, it is 160 milligram per milliliter in a single dose prefilled syringe. Next, we have Zimfentra. October of last year, FDA approved a sub-Q formulation under the brand name Zimfentra for the maintenance treatment of adults with moderate to severe active ulcerative colitis or Crohn's, following treatment with an IV-administered infliximab product. Maintenance treatment with Zimfentra, 120 mg sub-Q once every 2 weeks, may begin at or after week 10 of IV infliximab therapy. Zimfentra is the only infliximab product that is given via sub-Q injection and can be administered by the patient or caregiver. This carries the same black box warnings as all of infliximab. In terms of dosing, as I mentioned earlier, it is 120 milligrams sub-Q every 2 weeks, but must be started after week 10. In terms of formulation, it is a sub-Q, 120 milligrams per milliliter in a single dose prefilled syringe, prefilled pen, and prefilled syringe with a needle guard. Marissa, I'm so sorry. Can you go back 2 slides where it said Enbrel? I had a typo earlier and I misspoke. For this, Enbrel received a expanded indication for juvenile psoriatic arthritis of 2 years of age or older. No additional changes. Just please ignore line 2 where it says October 2023. Going back to now, we're at Weslana. In November of 2023, FDA approved this medication as an interchangeable biosimilar to Stelara. Now, it has indications for adult patients. It's moderate to severe plaque psoriasis, who are candidates for phototherapy or systemic therapy, active psoriatic arthritis, moderate to severe active Crohn's disease, and moderate to severe active ulcerative colitis. For pediatric patients defined as 6 years of age or older, moderate to severe plaque psoriasis, who are candidates for phototherapy or systemic therapy, and active psoriatic arthritis. It does carry precautions for infections. Dosing is stratified by indication, age, and weight. And the formulation is a sub-Q and an IV infusion. So the sub-Q is 45 mg per 0.5 or 90 mg per milliliter in a single dose prefilled syringe, or 45 mg per 0.5 milliliter solution in a single dose vial. For IV infusion, and it's 130 mg per 26 milliliter solution in a single dose vial. Next, we have Idacio. October of last year, FDA approved for the treatment of moderate to severe periodontitis. November, it received a approval for non-infectious intermediate posterior and panuveitis. And then in January of this year, a new formulation for 40 mg per 0.8 milliliter single dose vial kit for institutional use only. For those 2 new indications, the dosing is identical, periodontitis and uveitis, as you can see here. Then we have Cosentyx. October of last year, FDA approved first IV formulation IL-17A antagonist for the treatment of adults with active psoriatic arthritis, active ankylosing spondylitis, and active non-radiographic axial spondyloarthritis with objective signs of inflammation. And in November, FDA approved for the treatment of moderate to severe periodontitis suppurativa. As you can see, the dosing is identical, and just the IV infusion update there. Next, we have Orencia. November of last year, FDA expanded the subcutaneous use of Orencia for the treatment of active psoriatic arthritis to include patients 2 years of age or older. No additional changes there. For that, it is still weight-based, so you stratify the dose based on the patient's weight. Then we have Simlandi. We've made it to 2024. March of this year, FDA approved Simlandi, a high concentration interchangeable biosimilar to Humira. Carries the same indications for rheumatoid arthritis, JIA, psoriatic arthritis, ankylosing spondylitis, Crohn's, UC, plaque psoriasis, periodontitis, and uveitis. Same warnings, black box warnings. Dosing is stratified by indication, weight, and age. And the formulation is a 40 mg per 0.4 single dose auto-injector. Then we have Tyenne. In March last month, FDA approved Tyenne, a biosimilar to Actemra. It's approved for the following indications: adults with moderate to severe active rheumatoid arthritis who've had an inadequate response to 1 or more DMARD, adults with giant cell arteritis, polyarticular JIA in patients 2 years of age or older, systemic JIA in patients 2 years of age or older. In terms of precautions, black box warning for serious infections and to monitor signs and symptoms of hepatic injury and hepatotoxicity in patients. Dosing stratified by indication, weight, and age. Formulations include IV infusion injections and subcutaneous injections. And lastly, we have Skyrizi. March of this year, Skyrizi was approved as a 90 mg per milliliter single dose prefilled syringe to allow for a sub-Q self-administration of maintenance dosage of treatment of moderate to severe active Crohn's disease in adults. The patients must be trained on a subcutaneous technique before self-injecting. And for this, the dosing, as you can see, it's 600 mg IV over at least 1 hour at week 0, 4, and 8. And then the recommended maintenance dose is 180 milligrams or 360 milligrams sub-Q at week 12 and every 8 weeks thereafter. And I will pause there for the committee while I drink a little water.
That's a marathon talk there. Umang, well done. Questions from the board for Umang? And while the committee is getting the thoughts together, Marissa, if we could have a look at the drug list, the PDL list.
Okay, this is Marissa. So this is a pretty easy class as far as preferred products. I will make a note that the Sabinco is in its own separate class just because of the way that I believe it came to market. It was before I think some of these other products got FDA approval for atopic dermatitis. So that's really the reason why this lives in its own atopic dermatitis JAK inhibitors class. So for that reason, it is preferred. There is PA on that product. There's PA on pretty much all of these products. For the cytokinin CAMs, like I said, it is very straightforward. The only preferred product that we have in that class are preferred products, I should say, are brand name Humira and brand name Enbrel. For those are really our only two preferred products. We do have the different, I apologize for all of the scrolling, the different formulations for this. The only one for Enbrel that is not preferred is the mini, but the SureClick, the syringe, those are preferred. And then same thing for Humira. We have pretty much all the different formulations of Humira preferred. There is one product I do want to call out that is preferred, but it is nuanced, and it's Spavigo. So that one, that product, we do have that preferred just because of its really specialized indication. The indication does slip me at this moment, but it's not very similar as far as the other products that, the cytokine CAMs of the world treat. So that's why we have that one preferred, because the indication for that one is very specific. So currently, the policy that all of these live in, it wasn't really appropriate, but it is still considered a cytokine CAM nonetheless. But just wanted to call that to your attention. And then for this one, the Simlandi, I'm not sure why this is kind of separated, but it is a cytokine CAM antagonist. I think this might just be some semicolon spacing maybe that got it kind of off of the other classes, but that is non-preferred. But I do also want to call out the adalimumab or the Humira biosimilars. We do make those non-preferred as they do come out to the market. We do treat them like any new drug that has come out, just because right now we are still preferring the brand name Humira and the brand name Enbrel, which doesn't have any biosimilar yet. But I can take any questions the board might have regarding these agents.
So Weslara, is that out yet, or was it just like approved, but it's not actually out yet?
This is Marissa. So it may not be showing up. I don't see it here on our file, so it just hasn't come out yet as far as our drug database that we get approved drugs from yet.
Stelara is the one that I feel like I've had the most trouble with, like even with rheumatology, writing multiple letters with inability to get it approved without trying Humira first, which I don't know the details, but rheumatology has told me they don't think that like makes sense, like science-wise. So I guess I'm just really excited about the potential ability for patients to get Stelara with the biosimilar. So that's just my perspective on the ground, I guess.
This is Marissa. Yeah, thank you so much for sharing that.
Thank you, Zoe.
Marissa, this is John McKay. Are there any JAK kinase inhibitors that are preferred at all?
This is Marissa. No, I don't believe we have any of the JAK inhibitors preferred. Do you want me to stop at a particular, the names of all of them escape me, but I know that we don't have any of them preferred from what I can recall. Besides, this Sabinco, but that was just a caveated type of drug. I will disclaim that we are in the process of revamping our cytokine cam antagonist policy. So you will probably see that policy. It's actually getting broken out. We're planning on breaking them out by their mechanisms of action. So right now, currently, the way that those policies are written, they're really all housed in one policy with all of the products listed. So we are working on currently breaking them out by their mechanism of action. You will see that policy be presented, or policies, I should say, presented later this year. So that's more to come. And then we are considering, at the same time, whether or not we do want to kind of reclassify some of these drugs as far as using their mechanisms of action. But we're still having some internal discussion on that. But this class in particular is getting very big and there is a lot of overlap as far as the indications. So management of it can be a little bit tricky, we're coming to find.
For all the adalimumab biosimilars, is it have you just not made any of them preferred because you're waiting on rebate negotiations compared to the brand?
This is Marissa. Yeah, that's correct, Kevin. Right now, the way that we're seeing the prices for them come out, it's still just more effective or better for us to prefer the brand at this time. We are monitoring it closely to see, when is a good time that, we may want to switch over to the biosimilars, but right now where we are, it's not really beneficial for us right now.
I do know we have 2 stakeholders listed who had signed up to speak. So while the committee is mulling this over, let's maybe bring them on. So I see Erin Nowak from AbbVie. Are you online? And if you are, you can unmute yourself.
I do not see Erin, but I do see Melinda here.
Okay, great. Melinda Turkington?
Yes. Melinda, you can unmute yourself.
Good afternoon. Can you hear me?
Yes, we can. Go ahead.
Wonderful. So I did fill out the form in advance on which I noted that I am not a patient or a provider. I am an employee of UCB, the manufacturer of Bimzelx. My name is Dr. Melinda Turkington, and I am a medical outcomes liaison with UCB. Bimzelx, or bimekizumab, is the first and only FDA-approved humanized interleukin IL-17A plus F antagonist. So not only an IL-17A inhibitor, but both A and F, which is a unique mechanism of action. It's indicated for the treatment of moderate to severe plaque psoriasis in adults who are candidates for systemic therapy or phototherapy. He went through the dosing, but it is available as a prefilled syringe and an auto-injector for self-injection or administration by a healthcare provider. It is dosed as 2 syringes for a total of 320 milligrams every 4 weeks for 16 weeks, with that option to switch to every 8 weeks for patients weighing less than 120 kilograms. There were 3 multi-center randomized double-blind studies, which enrolled a total of 1,480 subjects, which was reviewed early this morning, 18 years of age and older with moderate to severe plaque psoriasis. At week 16, 85 to 91% of patients showed a PASI 90 and 84 to 93% achieved an IGA global score of 0 to 1. Additionally, between 59 and 68% of patients achieved a PASI 100 or completely clear skin. As it was noted in the psoriasis class review this morning, there are 3 head-to-head comparison trials demonstrating superiority to adalimumab, ustekinumab, and secukinumab. Bimzelx most common adverse reactions greater than 1% are upper respiratory infections, oral candidiasis, headache, injection site reactions, tinea infections, gastroenteritis, herpes simplex infections, acne folliculitis, and other candida infections. It may increase the risk of suicidal ideation and behavior, but no causal association has been made. And it may increase the risk of infections, which I already touched on. Test liver enzymes, alkaline phosphatase, and bilirubin bilirubin at baseline periodically during the treatment with Bimzelx and according to routine patient management. Cases of IBD have been reported in patients with IL-17 inhibitors. And then, of course, please refer to the prescribing information for full safety and efficacy profile, or please feel free to ask me any questions as well. Thank you so much for your time and consideration of Bimzelx and our request for Bimzelx to be considered for a first-line treatment for patients suffering from moderate to severe plaque psoriasis. And again, thank you for your time.
Thank you, Melinda. Any questions for Dr. Turkington from the board? And do you see Erin? Nanya?
No,
Erin?
I do not. I do not see Erin. Is Erin on the call? If you are, please raise your hand.
And if there are any other stakeholders who would like to speak, now would be the time. Please raise your hand. No?
No, no one else.
Okay. Okay. So I suppose with that, if Marissa, you could pull up the motion, the board to review.
This is Marissa. Yeah, so DOR board, you will be making two different motions today just because that one product lives in that one class and then all the other cytokine and CAMs are together. So just wanted to give you all that heads up on that.
I can make this one since it's alone. I move that all products in the atopic dermatitis agents Janus kinase JAK inhibitor oral class are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of HCA. Products in this class may require prior authorization to determine medical necessity. All non-preferred products require a trial of at least 2 preferred products with the same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
This is Michael Corsillis. I second that.
All those in favor, please say aye.
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 Any opposed or abstain? Okay, with that, the motion carries. Then the next one on CAM antagonists.
This is John McKay. I can move. Again, I move that all products in the cytokine and CAM antagonist class are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of the HCA. Products in this class may require prior authorization to determine medical necessity. All non-preferred products require a trial of at least 2 preferred products with the same indication before non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
This is Laura Bestie. I second the motion.
All those in favor, please say aye.
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 Any opposed or abstain? Okay, with that, the motion carries, and we are right at 4:01. Great job, board. We covered a lot today. Nanya, Marissa, unless there's anything else, do we adjourn for today?
Yes, you can. You guys can adjourn for today. Thank you guys so much. I don't think there's anything. What about you, Marissa? Anything on your end?
This is Marissa. Nope, we have everything that we need. So thank you so much, DOR board.
Thank you
Thank
so
you.
Thank
Thank you.
Thank you.
Thanks,
You and
everyone.
you.
Thank you. Have a great day.
You.
Bye.
Bye.
Bye.